Good morning, ladies and gentlemen, respected dignitaries and distinguished speakers. It gives me immense pleasure to welcome you all to the National Workshop on Research Methodology, funded in ICMR-Medical Innovations Patent Mitra Initiative for Patent and Technology Transfer Facilitation, jointly organized by the Indian Council of Medical Research, ICMR, and All India Institute of Medical Sciences, AIIMS, New Delhi. Today marks the beginning of a journey of knowledge, collaboration, and innovation, a platform where brilliant minds from across the nation come together to explore technology and innovation, can together drive healthcare excellence. So with immense pride, let us embark on this enriching and intellectually stimulating day ahead. Before we begin with the formal proceedings, we will commence with an invocation, an opportunity to center our hearts and minds in the unity and purpose. Thank you.
Sahanavavatu sahanam sahaviryam karavavahe tejasvinavadhitamastu mam vedveshavahe Shanti, Shanti.
Dais. I'll request Dr. Alka Sharma, Additional DG, ICMR Extramural Research, to please join us on the dais, ma'am.
Namaskar, a very good morning everyone. So I extend my warm welcome to all the dignitaries, to all the faculty members, participants and students to these 2 days of national workshop. So I'm very happy to see almost all the chairs are occupied. So the initiative by AIIMS New Delhi and ICMR is mainly aiming to boost the healthcare innovation and by building a more robust and dynamic ecosystem to provide a more comprehensive patent and technology transfer support to all the innovators who are in biomedical research field, so that their ideas, they get, translated into practical solutions in healthcare across India. So we today, let me tell you, our Honorable Director, Srinivasan sir, he's unable to join us today because he had to rush for an emergency meeting to Doordarshan. But we are honored to have so many, dignitaries and experts and senior officials from ICMR. So I now welcome Dr. Sucheta Markan, Scientist F, Head Innovation and Translational Research Unit and Mission in Charge, ICMR.
So thank you very much for giving me this dais to give a stage for what is this workshop all about. It'll be nice to prize all of you about this workshop. So as you can see that the name of the workshop itself is self-explanatory, that we are trying to cater to this workshop as a national workshop for research methodology, funding opportunities and patent mitra. I'll start my journey. I would like to inform you that how did we design this workshop will be very much self-explanatory from this slide. Start with the basics. If we have with this image, I just want to apprise all of you that the key mandate of this workshop is to help you develop regulatory compliant products to see how you can innovate. So that's the key mandate. And if you have to innovate, this figure of identify, invent, implement, simplifying the technologies in these 3 I's becomes very relevant to all of you to understand and to connect on how we have tried to firm up or plan this meeting. It's the vision of the Director AIIMS and DG ICMR that they thought that we should be bringing all AIIMS together under one roof, that too in AIIMS, to see how you all can get benefited. So if you have to anytime innovate and develop an innovative technology, what comes to your mind as doctors when you look at patients? you look at of products, lot of medical devices, diagnostics. So let us take a very simple example of a BP monitor. It will definitely come to your mind that a sphygmomanometer or a BP monitor, digital, which we have, which every AIIMS, everybody has to use nowadays. It shows lot of variation from device to device. So very simple thing would come to your mind that can we make a sphygmomanometer, a BP operator very, very accurate, so that it is very, very, you are very confident that the reading it gives us. Likewise, looking at biopsy devices, when you look at doing liver biopsies, can it be make more accurate? Can it take a bigger size? So all those things definitely come to your mind. So in the philosophy of medtech innovation, we believe very strongly that medtech innovation pathway starts from unmet need identification, and doctors of our country, the medical professionals, the doctors of our country are the key people working day in and day out with the inventions, are the people who know, who realize that what is needed, what is the unmet need, what kind of products should be developed. Then only they become relevant. So that's why this meeting becomes very important, because doctors are at the center of innovations. That's very, very important. One message I wanted to convey. Of course, even if you have a very good mindset, if you don't have funding, things won't work. So there I have pointed out that innovation also starts with funding. The idea comes to your mind, you write a proposal and try to see if you can get some kind of funding to implement your idea. Once you identify the unmet need, you try to look at the solution space. What are the available solutions in the market? Say you have a sphygmomanometer, you try to see what are the different type of products similarly available in the market, which is more accurate, which can be taken to the next level. Say biopsy device, the example I gave. So identify phase starts with funding, identifying the unmet need, which you believe while working in your clinics that what is needed, talking to doctors, talking to care providers. Then you try to look at the solution space. Once this work is done, then you try to identify how do you develop that technology. How do you try to solve that problem by solving some kind of a solution? So that is the key identify phase. Once that clarity comes to your mind, comes the invent phase, where you try to make some kind of proof of concepts. You try to work upon them in the lab and see whether it is workable or not. You have 5 solutions in your mind. You develop some kind of prototypes. I'm sure people in this room might be aware of the SIB program, the Stanford India Biodesign program. So that philosophy is very clear that innovation starts with identify phase, then you make some initial proof of concept, some prototypes, whether it works or not. Then you try to see whether they work. You do some kind of a demonstration. There is in-house validation, then you will give it to some of your colleagues and say, do third-party validation. Whatever I believe should also work in somebody else's lab as well. So you need to validate them through third-party validation. Regulation, developing prototypes which are not regulatory compliant won't work. Now we have to go to the next level, wherein we need to see how we can develop regulatory compliant products. So getting regulatory clarity, and then developing products is of utmost important. Then the technology, once it is, you receive all DCGI approvals, that product will be launched, and then the dream come true. The product will be used by people at large. But people in this room, do you truly believe that this innovation cycle, the way it looks like very simple, does it actually happen when we work with these innovations? That's true. So generally what we see when we talk to innovators is that they are very good in getting funding, good writing some proposals, but taking it to the next level of technology development, converting proof of concept into a full developed product, which is regulatory compliant and is used by people at large, it's a very beautiful dream in our mind, which everybody wants to do, but maybe we really need to see how to do it. But that dream is there in our mind. So what we see typically is all these proof of concept bucketing down in a bucket and waiting to go to the next level. Why this happens? We as innovators are very passionate about our invention. We want to do everything ourselves. There again we have a challenge. We need to develop those technologies, do unmet need identification, but there comes the role of an industry. You need to transfer your technology to a company who is capable of taking it to the next level. So it's not about giving your technology to somebody else. It is about joining hands with innovators, joining hands with industry, and writing a very good publication is good, but we need to patent such inventions. So if we have to talk about innovations, which is the very, very important thing we are going to talk in these. The key thing I wanted to tell all of you that in these 2 days, we are not going to talk about basic research. We are going to talk about translational research, how these ideas can be patented, wherein Patent Mitra will help you. We'll go deep dive into what this initiative is all about. How do you write? Can we help you as ICMR, as Patent Mitra team to do patentability assessment, file patent applications, do it very methodically. You guys are in a society wherein you know very well how to do research. What are the kind of study design if you have to clinically validate a product? Some of the things we'll try to touch upon in these 2 days, because doing a clinical evaluation of a product for regulatory compliance is something different. That also we'll try to touch upon. How do we transfer your technology to a company is a game, because Patent Mitra is there, it has a key mandate to facilitate tech transfers. So you can just see what we are trying to do. We are trying to talk about funding opportunities in this meeting. We are going to tell you how to do patentability assessment, basically how ICMR team will help you to do patentability assessment, file patents as your friends, as mitra. Then how do you get into regulations, try to understand what is regulations, how do we help you in transferring your technology and joining your hands so that your dreams come to a reality. Plan of 2 days, and it's a very, very well thought kind of a workshop wherein we have dwelled deep into every topic. We have tried to bring in the best of the experts who will justify this topic given to them. So just to summarize what I spoke, the topics that we are going to cover in these 2 days are now been lined up for you in this slide. We will be telling you about funding opportunities, not only about ICMR funding opportunities, the entire gamut of funding opportunities which is being offered by Government of India. Because in this room we are Team India. We are all of government approach. It's just ICMR on the front of it, but the entire workshop will demonstrate whole of government approach, wherein we have experts from DST. They will be telling you about the ANRF schemes. They will be telling about you DST schemes, the D funding opportunities, ICMR funding opportunities, so that when you go from here, DOP funding opportunities, so that you know the holistic picture of different funding opportunities being offered by all government agencies at technology development, which you can explore to use. Then we will be telling very specifically about medical innovations patent mitra, which is the word medical innovation specifically demonstrate it is for you all. We want to, as biomedical research organization of the country, ICMR, we really want to help medical innovators, the doctors in this room, in the entire country, to give them hand-holding support in filing, evaluating their ends for you. Then we will be we will be taking you through the research methodology. Lot of sessions you will see on research methodology. Then again, how can we help you with the technology transfer, which looks like very difficult to doctors, but it's the professionals know their job. We'll be joining hands with you to see how we can help you in transferring your technologies. And Dr. Vel Pandian also leads these tech transfer initiatives, IP initiatives from AIIMS. So to tell you that when AIIMS is doing, every institute can do it. Then we also have one session around MedTech Mitra, wherein we will be, it's a very, very unique initiative. Dr. Vinod Paul, Member Health Niti Aayog, Secretary DHR, they will be joining us tomorrow, and they are the thought leaders behind this initiative. It's very, very unique initiative. They will be telling you themselves about this initiative tomorrow. So you can see that we have tried to make this a very, very holistic, wherein we have best of the visionaries themselves will be coming and telling you about the vision, why these initiatives were started. We'll have Secretary DHR tomorrow. We'll have Dr. Vinod K. Paul, Member Health Niti Aayog, who is from AIIMS himself. The past, he belongs to AIIMS himself, now in Member Niti Aayog. We will have NMC chair coming tomorrow to share his perspective. Director AIIMS will be joining us. And Indian Patent Office himself will be coming tomorrow to give you a talk. Then this is the first line of the vision, so that you get a clarity on the unmet need, the high level vision which is behind these initiatives. And then on the second level, we have the ecosystem enablers, high level leaders at different government agencies leading these initiatives would be coming and speaking to you, sharing their perspective. And then you will, directors also demonstrate some technologies. developed through medical doctors, being validated through medical doctors, which we can put up as a stall outside, which we plan to do as part of experiential sharing, so that you see products and believe that yes, if these people are doing, you can also do it. And we also specifically invited some of the people who have built up innovations and have been able to transfer those technologies. So those startup doctor combination is also there, lined up tomorrow for experiential sharing. That's the USP of our workshops, wherein we always keep some sessions for experience sharing by innovators, so that they tell you about their journey of how they were able to invent, how they were able to develop technologies, and now those technologies, products are being sold. So those very few unique case studies of innovators coming and sharing their experience with you is also lined up tomorrow. So please be with us the entire 2 days. And then because we didn't want to just give you lecture and go away from here, so we have a live counter outside, wherein Medical Innovations Patent Mitra booth is being made outside, wherein if you have something in mind, you want to file your invention, you want to register yourself with Patent Mitra, so that once you go from here, we are behind you to see with this initiative. We have some of my colleagues are there outside in that booth, who will be helping you to register if you have an invention in mind with the Patent Mitra, so that we can actually help you out when you go out from here. And there is an exhibition also waiting for you. So the expected outcomes are very clear. The last line is very important for me, wherein we really want that you should be able to believe in yourself that we all can do it as Team India, as medical engineer collaborators, as government medical and engineering institute collaborators, that together we can make the most relevant innovations and we can take them to the next level, and they can be taken to the level where it actually takes us to the public health impact. People are able to use the innovations coming from your mind to the market. So that mind to market transition, the belief we can do it, is the key mandate. We want to make sure after 2 days of brainstorming discussions, we all are able to come to this conclusion that we can do it. And these are some of the expectations from the workshop, that you will be able to understand the various government opportunities for funding. You should be able to understand the research methodology for technology from technology perspective. What are the effective ways of clinical evaluation? What are the best practices of Indian Patent Office when it comes to patents? Thing is about the networking dinner and the exhibition is that bringing them together under one roof is definitely, we want you to collaborate. We want you to talk to each other. That's very important because our lives are very busy. We have been given a lot of mandates to do in our organization. So whether see how you can collaborate and make some proposals, talk about inventions which can be taken to the next level. So with this, I'll just stop and request you all to have happy learning, enjoy the sessions. Thank you very much.
Thank you so much, Dr. Sucheta, for this wonderful 360-degree overview about this 2 days workshop. So let me welcome one by one. Can I call someone to present a flower pot to Dr. Sucheta Markan? Thank you so much. And now I welcome Dr. Alka Sharma, Scientist H and Additional DG Extramural Research, ICMR. So let me welcome them and then we can have their lectures. Now I welcome Srimati Manisha Saxena, Senior DDG, ICMR. So she was here with us only before ICMR. And we have Dr. Vishnu Vardhan, ex-director, NIMS, ICMR. So thank you, sir, for coming here. And I welcome Dr. Jagdish Rajesh, Director, DDG, ICMR. Thank you, sir, for your presence. And I also welcome Dr. T. Velpandian, Professor and the Member Secretary of IPR Division and Technology Transfer Division, AIIMS, New Delhi. So I'm happy to announce having almost, it is 120 participants across the country from different institutions. We have faculty participants from AIIMS, New Delhi as well, and Bathinda, Bhopal, Bhubaneswar, Bilaspur, Jodhpur, Madurai, Nagpur, Raebareli, Raipur, Rishikesh, Maidangari, and also from JIPMAR and NIMHANS and PGI. A great round of applause for all of you because you could make it in a very short notice. So I'm very happy to say that, this 2 days national workshop is going to be, I can see that, very, very beneficial to all of you. So I request your active participation because your active interaction and, everything will make this workshop more dynamic and productive and successful. My best wishes to all of you. Thank you so much.
Thank you so much, ma'am, for your warm words of welcome. Now to enlighten us about the purpose, theme and expectations from the workshop, may I invite Dr. T. Vel Pandian, Professor, IPR and TT Division, AIIMS.
Thank you so much. Indeed, thank you all of you. We got an overwhelming response from all of you from 12 AIIMS, or something which we feel very proud. In a very short duration, everybody made it. And together we are making around 120 participation. That's really great. So let me just, Professor Srinivas, he is supposed to join, but he could not join because of some other urgent work. He will be joining soon with us. Something he wanted to convey to you, all of you. The main thing is the purpose of this today's meeting, 2 days conclave, I would say, is that I remember that Manisha Madam and Dr. Suchita, when they both of them met director, told about that we have something, a new program coming up that is MedTech Mitra. Immediately, Patent Mitra, immediately he said, call Vel Pandian. And he said, that's a golden opportunity. Why only AIIMS Delhi every time? Why don't we make it possible for all? And he said, we have other institutions which are of national importance. Call them also. So the time was too short. I remember, we had several discussions with Manisha Madam, and she said, what to do? We have to make it possible. And if you require, ma'am, thank you so much for that. And director was very happy. He said that call everybody. See, but AIIMS started in AIIMS Act started a special act of parliament in 1956. Ever since till 2012, there was only one AIIMS. Although at the same time we had many IITs. But when it comes to medical sciences, to promote medical, especially research and teaching, advanced medical research and teaching, only AIIMS type of institution is the only one which was taking a lead. Therefore, the necessity was felt. And through PMSA, they started in 2012. By 2014, we had 7 institutions only in India. Today, we see 25 institutions, and it is growing. When we got the response from everybody, somebody said that, I'm sorry, we have very less faculty recruits. We cannot send anybody, otherwise we won't be able to run the OPD. And today, such a thing, Monday it has happened, so we cannot send people. Therefore, whatever possible, we will send it. So I'm very happy all of you could join here. I'm very sure you will be able to spread the information across. Another important thing is this. See, as my colleague Dr. Suchita explained about it, see, the whole 2 days are power-packed with enough luminaries from all corners, and such a type of pulling people is not possible otherwise. We will not be able to interact with such people anywhere else. So you are free to interact at any time and ask them all your questions. And finally, tomorrow I am going to discuss about things which, at AIIMS, I know your pulse. I know your problem. See, when somebody comes to me every day, one faculty will definitely meet me with a couple of questions. Therefore, over the period of time, how it has happened in AIIMS? AIIMS, the change started happening in 2012. 2012, one of the inventor, along with one of you, I went to the administration. I said, I lost because you didn't process my patent on time, I lost it. So what happened is that I requested permission to file a patent in 2008. 2012, I was searching for that file, where is it? So from one file, one office, it went to another table, another table, another table. Finally, I could find that file bundled and kept in a dark room in Abyss. So when I took it up, the administration said, why don't you take it up and make it? Well, I took the challenge in 2012 for the first patent policy for AIIMS has been brought in 2017, approved by governing body. That is a starting point of medical innovation through a planned approach. Because without a policy, we won't be able to run through these administrative tables. You can cut all the ribbons, but among the clerks. So we could cut that red ribbon with the help of policy. Now the policy, if you just open up AIIMS policy, it's having a beautiful aspect. We open any policies throughout India, all the IIT policies, they're different. But AIIMS policy is different. Here the situation, the ambience and atmosphere is entirely different. It's not like IITs. Till today, yesterday director was informing me that we got the permission to go to Section 8 company. So we are going ahead with a Section 8 company in India. So for the first time, almost some of the institutions, the AIIMS, I came to know, they all started it, but they're all doing it. Everywhere I get a phone call, what is happening in your institution? So now we can proudly say that yes, we are going ahead. Now see, you must understand, there is a difference between you get a doctor, the degree of doctor, very, very catchy. There is a small flip here. A doctor or doctored. So doctored, when you say doctored, there is a suspicion behind it. When there is a doctor, okay, there is some sort of certainty is there in that. When you use this word very carefully, doctored and doctor. So something which is enabling you to stand and change the existing ways doctors are conferred. When you is a doctor of medicine or doctor of philosophy, is always you get the degree for the thesis what you have produced, for the hypothesis you have generated, then only it is being honored. But the same thing can be used for the flip side also, that is doctored. Somebody has doctored a video, then you go to investigation to CBI. So there is always a sort of a response, and the responsibility is the one which makes a normal. What is the difference? Somebody asked me the difference. What is this invention and innovation? There are two terminologies we always use it, right? So you always say invention. Invention, no, it's not my cup of tea. It can happen. The invention means you always take it to such a level of, discovery of penicillin. But it depends upon the necessity to make something possible. And in India, we have lot of opportunities. Because you see the things where what is lagging in our society. Because ours is a very old, 10,000-year-old civilization in a conservative estimate. So this culture is having multi-dimensional atmosphere. You are forced with different situations. How you try to handle the situation. Now, your wisdom is getting transformed into an innovation that is going to be useful for people. And this workshop, what we are trying to do is, the aim of the workshop is activate, inculcate, innovate and succeed. So again it is AIIMS. Activate everybody, ignite everybody and succeed everybody. This is what we are trying to think about it, and that is the purpose of today's meeting. The 2 days you will enjoy it, and you will meet all the luminaries very comfortably in the dinner and talk to them formally, informally, and be very informal. AIIMS is always little, you can talk to people, you can meet people, so all things possible. And what we are expecting out of you is that please take this across your institution and make more innovation possible. By the way, attain the economy what Prime Minister dreams about. Let us join hands with the initiative of our Prime Minister, and we thank the patent mitra for joining us to make this possible. On behalf of all of you, I thank them for allowing this opportunity. Thank you very much. That's what I expected from you. Thank you so much.
Thank you, sir, for setting the tone of this workshop. We are now honored to have with us Dr. Alka Sharma, Additional DG Extramural Research, ICMR, Secretary DHR, and DG ICMR for the keynote address.
Thank you. So, as you heard, the Dr. M. Srinivas, Director AIIMS, New Delhi, who could not join because of some last moment urgency, but he may join us and maybe definitely during dinner today and also tomorrow, the session. My colleagues, Ms. Manisha Saxena, Senior DDG, and also we have Mr. Jagdish Rajesh, DDG ICMR. So team ICMR is very strong today for this event. Dr. T. Velpandian, Professor and Technology Transfer Division, he is a Member Secretary AIIMS. Professor Navadnita Haldar, eminent clinical from AIIMS, and also we have Dr. Suchita Makan with her team. Clinical researchers, doctors, healthcare professionals from various AIIMS across the country, ICMR officials, and all the participants. A very good morning to all of you. Dr. Rajiv Bahel, Secretary, Department of Health Research and DG ICMR, could not join us today as he is currently at Bharat Mandapam for an event which is being inaugurated by the Honorable Prime Minister. My colleagues that I already mentioned, they all are here, an entire team under Dr. Suchita Makan for this patent mitra. They are also here to interact with you during this two-day national workshop. So on behalf of ICMR, I welcome each one of you devoting your valuable time for this important national workshop, which is being organized today jointly with AIIMS on a very important theme, covering research methodology, funding opportunities, and ICMR-Medical innovations, Patent Mitra Initiative for Patent and Technology Transfer Facilitation. So as you know, ICMR, the apex body under DHR, Ministry of Health and Family Welfare, has mandate to formulate, facilitate, and promote biomedical research across the country, aligned with national health research priorities. So accordingly, ICMR is nurturing young minds and supporting transformative biomedical and healthcare research through its intramural and extramural funding programs, fellowships, initiatives, and so on. It empowers scientists and clinical researchers to translate their ideas into meaningful healthcare solutions. So as you know, intramural grant fund high-risk, high-reward projects within ICMR institutions, whereas extramural programs are being supported under various categories such as investigators initiated research grants, intermediate grants for translational research, and centers for advanced research, that is CAR, to strengthen innovation-driven healthcare ecosystem. Most of you may be beneficiaries of these extramural. And in addition, we also support the NHRPs, the National Health Research Programs, and that is aligned with the national research, health research priorities. Through these initiatives, ICMR has been able to put a mechanism in place, but still there is a gap between research and real-world clinical needs in the country. And in the, just before me, two speakers, they have highlighted the importance of end-to-end processes, right from idea generation up to the market ready, Dr. Suchita and Dr. Velpandian. So just maybe you may be aware, and just to recap, that India's rank in Global Innovation Index, that we all are aware, 39, that reflects its growing emphasis and also entrepreneurship-driven growth. And despite these advances, significant gaps persist in innovation indicators, particularly in patent filing and technology transfer, and who all are now in the process of development of proof of concept and early stage technologies. You all are aware that this is not the simple that R&D, which is easy to publish paper, but if you have to develop one technology end to end, is a entirely different journey and an entire different cup of tea. So according to WIPO in 2024, India contributed only about 2.5% of global patent applications in 2023. And the rate of successful technology transfer from research institutions to industry remains modest. Localization of indigenous innovations. As per the IP India Annual Report 2023, within the biomedical sector, patents accounts for only 7 to 8% of total, 3.4% of total grants as against total number of patent applications filed in Indian Patent Office. So this reality emphasizes the need for a structured, accessible and expert-driven support mechanism to foster quality patent application filing. So therefore, as you know, in order quality patent filing and accelerate the translation of biomedical innovations for public benefit, ICMR under the guidance of Niti Aayog has launched the initiative, that is National Initiative Medical Innovations Patent Mitra, which was formally launched by the Honorable Minister of Health and Family Welfare, Shri J.P. Nadda ji, on 8th March this year. This initiative has been implemented in partnership with the Department of Pharmaceuticals under Ministry of Chemicals and Fertilizers and supported by Department for Promotion of Industry and Internal Trade, that we commonly known as DPIIT, we all can connect it, under Ministry of Commerce and Industry, and invites key national stakeholders to ensure comprehensive and effective outcomes in terms of catalyzing innovation ecosystem. So message is very clear here that can work on these kind of initiatives. This should be with the right kind of stakeholders, promoters, interagencies, intergovernmental, whole of government approach. So ICMR is thoroughly now working with all ministries, department concerned just to facilitate this initiative across the country, and there are few. And I'm sure that you are going back to your organization, you will spread this message to other colleagues also, that there is a mechanism in place. Now country is ready to take that innovations further to the next level. So as you heard earlier, that aligned with our government vision of self-reliant, innovation-driven nation, Medical Innovations Patent Mitra offers expert guidance and end-to-end hand-holding filing and technology transfer of biomedical innovations emerging from both intramural as well as extramural research programs of ICMR, and also from medical colleges and research institutions in the country. Through this initiative, ICMR is advancing a people-centric and translational approach to biomedical innovation by providing complete support for patentability assessment, filing, maintenance, and technology transfer. So Mitra bridges the gap between laboratory research and patent and public health application by simplifying processes, strengthening IP support, and fostering translation across therapeutics, biologics, medical devices, diagnostics, and vaccines. And to highlight here that Medical Innovation Patent Mitra follows a zero-cost model. I'm repeating this, a zero-cost model. This was never before in the country like India. And now this is the opportunity for all of you to avail this platform and just take your innovations forward. And this ensures no expenses for patent filing or assessment for the first 5 years. And it's a good time, 5 years time is good time just to explore what is feasible, what can be done, and if it is not working, move ahead with new kind of innovation, and then you have gained the experience. So we never fail actually. If we are not successful to any, just taking any innovation to the next level, we learn a lot. And through that exercise, we can apply to our next innovation, and then we can take that to the next level quicker. So that is a zero cost model is the real, actually in real term, ICMR has taken this initiative, and this is really need of the hour, because many of you are now very young and you are starting your career in your institution, may not be possible all the time to have some kind of grant. In our country, the grants we have under very strict headings: manpower, non-accruing, consumables, this and that. So facilitation thing is maybe in some of the grants there is some flexibility. Normally we don't have. And then if it is zero cost, important, I'm repeating why this should register in your mind that this opportunity, this is something the government is ready to work with you and just facilitate this entire process forward. So this is actually that I said that access to innovation support and empowers India's biomedical research ecosystem. So just it is time to just recap what we have done so far. As I said, that launched by our Honorable Minister this year in March. So I'm happy to share here that under this initiative, in just 6 months period, March to September, because data is up to September. So far over 100 applications have been evaluated, more than 50 patents have been filed, 18 technology transfers deal have been facilitated, and also licensing of 3 vaccines addressing critical national health priorities. So we have a very strong team at ICMR, the entire patent mitra team that is working under the leadership of our DG, Dr. Bahel, Dr. Rajiv Bahel, and also we have Miss Manisha. So everyone is doctor when they just, but Miss Manisha and Dr. Suchita, and of course we have that strong support. Dr. Velpendian is here, and that entire team. So this is the beauty of working together, because 1 and 1, 11. We should always work and we should keep that in mind. Kind of applications, 100 applications, and then evaluation in 6 months time, 50 patents filing and 18 technology transfer deals. That is not possible without a team effort. No one alone can do anything. So entire team is a very dedicated team at ICMR and AIIMS, because this is joint efforts between these 2 organizations. So this is working really well. So this overwhelming response from outreach program at leading medical college reaffirms that the immense potential of this effort to empower clinical researchers and medical practitioners as innovators. So this is the right time for all of you to come forward and just spread this message to among your colleagues and also to your institutions, said that you all have to just think differently and just work towards that, because our country needs your all kind of efforts. So we all would agree that this is a continuous journey to advance biomedical research truly patient-centric and transformative. We must bridge the bench to bedside gap. Now again it's a bench to bedside. We are working right from the beginning, but now again it's a bench to bed, this bed to bench also. So it works both ways. It's not only the unidirectional bench to bedside. So that whatever is your intellectual mind can work, that you are the best judge. You have to just see that how to bring some innovation in your own area. So you all are actually here, and then needless to say that you understand that well, what are the diagnostic gaps, what are therapeutic constraints and unmet needs that influence patent outcomes. So ICMR recognizes the importance of addressing challenges, therefore will continue to promote and nurture indigenous innovations, foster and strengthen academia-industry linkages, and accelerate the journey from lab to market-ready solutions through Patent Mitra. So Mitra is, we just say in English, but it's a mitra, it's a friend. So friend is always there for you, and that is the concept behind this. So I encourage this, I just take this opportunity to encourage each one of you to leverage the medical innovations, Patent Mitra, a national platform designed to support and safeguard your ideas, streamline patenting processes, and facilitate the transition of innovations into impactful healthcare solutions. This national workshop is providing you all the opportunity to exchange ideas, explore collaboration, and our collective contribution to India's innovation-driven future. So with this, I wish all the best, very best to all of you for the fruitful discussion and a meaningful outcomes during this 2-day national workshop. I'm going to see you today evening also, or maybe tomorrow, and I think we will interact more when we'll meet, and after this maybe 1-day discussion today. So that will bring more clarity, and then also we can exchange our ideas and views. Thank you. Jai Hind, Jai Bharat.
Thank you, ma'am. Your words truly highlight ICMR's pivotal role in fostering research excellence and innovation across India. Next, I would like to invite Dr. Jagdish Rajesh, DDG Administration ICMR, for the felicitation address.
Good morning, all of you. Additional DG Madam. Okay. Respected Additional DG Madam, Senior DG Madam, Dr. Nabanita, Dr. Vel Pandian, Dr. Vishnu Vardhan, and distinguished intellectual guests from across all institutes of national importance, AIIMS, PGI, JIPMER, and other eminent institutions. And of course, last but Suchita Markan, our friendly scientist, friendly इसलिए क्योंकि सदैव आपके साथ रहेंगी और मित्रता निभाएंगी. This journey of us started way back in 2020 when I, along with Dr. Suchita, went to different institutes like IITs, IIMs, AIIMS, and everybody, where we started establishing centers of excellence. And then this idea came up of MedTech Mitra and Patent Mitra. तो ये जो जर्नी है जो हमारे सेक्रेटरी साहब डॉक्टर राजीव बहल, डॉक्टर वीके पॉल, इन सब का जो आइडिया था, उनका खुद का एक आइडिया अब जो है उभर के अभी आया है और इसमें काफ़ी कुछ लाभ उठाया है. कुछ हमने ऑलरेडी अचीव कर लिया, कुछ पेटेंट्स का ऑलरेडी फाइलिंग हो चुका है और कुछ टेक्नोलॉजीज ऑलरेडी. तो मैंने जब डॉक्टर वेल पांडेन का एड्रेस सुना था कि एडमिनिस्ट्रेशन के साथ बहुत लड़ाई की उन्होंने अपने पेटेंट वो बनाने के लिए, सर मैं आपको अश्योर करता हूँ कि ICMR में आपका ऐसा कुछ नहीं होगा सर. तो हम आपको हेल्प करेंगे. तो मैं ज़्यादा कुछ समय लेते हुए नहीं वो करूँगा क्योंकि बहुत समय कम है, अभी मैम का भी एक एड्रेस है. सो टू कंक्लूड मैं सिंपल एक शॉर्ट वर्ड में बताऊँगा कि मित्रों, ICMR आपके साथ बार हैं, तो इस इनिशिएटिव का लाभ उठाएं और ताकि पूरे अपने विश्व में इसका लाभ हो. थैंक यू सो मच.
Thank you, sir, for your warm words. To conclude this inaugural session, may I now request Srimati Manisha Saxena, Senior DDGA ICMR, to kindly deliver the vote of thanks.
Namaskar. As you all know, the organization of such events are generally the result of close cooperation among stakeholders and individuals. I consider it a great privilege to propose dignitaries on behalf of ICMR and Department of Health Research, and a deep appreciation for the support and encouragement provided. The successful commencement of this workshop is a testament to the collaborative spirit and dedication to all involved. I would like to express our sincere appreciation to the distinguished dignitaries who graced this session with their presence and insightful contributions. I want to mention about our DG, Secretary DHR and DG ICMR, Dr. who could not join today due to Prime Minister event, but his visionary leadership and unwavering support always support us in such collaborative events. I want to extend my special thanks to Dr. M. Srinivas, Director AIIMS, who also could not join us, but he was actively involved in designing this workshop. He individually see that it should start with research methodology itself, because that is the stone from which our journey for innovation starts. I want to extend our thanks to Dr. T. Velpandian, Professor IPR and TT Division AIIMS, for his continuous guidance and instrumental role in shaping this workshop. I want to extend to Dr. Halder, Professor, Department of Pharmacology, AIIMS, New Delhi, for playing a key role in organizing this meeting of AIIMS, smooth sailing of this inaugural session. I want to extend my thanks to Dr. Vishnuvardhan Rao and Dr. Jagdish Rajesh also for give support to this event. I am thankful to Dr. Suchita Markan, scientist head innovation translation research unit and mission in charge, MDMS ICMR, for clearly articulating the workshop's objective and introducing the Patent Mitra initiative with such clarity and purpose. And I want to extend a special mention about organizing team of AIIMS as well as ICMR for making this. Finally, I would like to thank all the participants for their presence and engagement. Your involvement is what truly brings life to the initiative. I really want to welcome Dr. M. Srinivas, Director AIIMS. His presence makes the difference to this event. Sir. Sir, I want to invite Director AIIMS for his keynote address, sir. Sir, please, sir. Sir.
Thank you. Good morning. Namaskar to all of you. Welcome to Delhi, the polluted city, and the AIIMS. Morning, we had a discussion on the pollution, so I had to go out. Well, first of all, give a big hand to ICMR. Big hand, you know. जितना ज़्यादा चाली मारेंगे, उतना funding मिलेगा आपको. Which institute is going to clap more? The funding is going to be more from ICMR to you. Note कर लेना, madam, please. ठीक है. So that was necessary because thanks to them, because they reached out to us. Generally we reach out to them, but they reach out to us and they said we want to do something. Then I said we will do something which is pan-India based, AIIMS and other INIs. So I'm really to you, ma'am, for the entire team of the ICMR who personally came to my office many times, said कि कब करना है, अभी करना है, जल्दी करना है, as if पैसा कुछ निकल जा रहा है और उसको खर्चा करना है, you know. So that was a very nice initiative, and definitely we are grateful to you, and that thanks to the people who have come, because at a very short notice you have come. That means that you are committed for this cause. This is something where I'll tell you, in a good olden days, you know, as a student, you know, we used to go to that BMJ workshop on research methodology, paying huge sums of money, and this kind of activities outside abroad cost you almost 1,000, 2,000 pounds and euros, you know. So the same thing which has been cut and paste here, you know, the entire structure of the program for the research methods, ethics, under ICMR and other funding opportunities for all of us, the patent mitra, and the various initiatives which the ICMR has come out today, you know, with respect to the patent, with respect to the equipment and all that, you know. This is something which we wanted to share with all of you, and looking forward that you have a wonderful interaction with each one of you here. See, it is not that you have come here to understand research methodology. All of us are doing research. They do. What is important, two components in the research, what we lack at the moment is, I always tell the people, you know, please come out of the syndrome of my syndrome. This is the problem. It is my equipment, my lab. Some people claim it's my student, you know, as if, you know, I don't want to go beyond, you know. So this is not right. See, this is a time where if we want to do high-end the research, we need to collaborate. First, collaborate intra-department, you know. That's a problem. You know, we don't talk to each other. Our machines don't talk to each other. Our reagents cannot be exchanged with each other. We don't sit with the people and say, what are you doing, and am I duplicating and doing the same thing? And it is very embarrassing, and sometimes the same fellows are presenting the conference and somebody says that the results have been like this, somebody says the results have been like this, you know. That is very embarrassing for the institutions and the country also. When you go out and present this kind of result somewhere, people will say, well, the country is what? Unity in diversity, where it is visible, you know, the department doesn't speak to each other. Please come out of this silo syndrome, my syndrome. After thinking of the intra-departmental collaboration, please go beyond the departments. Today's research cannot be done in one department. Let us be very honest. At least clinicians, at least they should come out of this ego. I don't know how many of you are clinicians here. The so-called pure clinicians, very less. That's good. That is good. That is good because we carry a huge ego. मैं surgeon हूँ, वो तो मेरे पास आना चाहिए. And you know that, I wonder that sometimes the people are standing in the operation theatre for 3-4 hours and saying, I used to shout on them, ki we will send the sample to you. The scientist fellow standing in front of the operation theatre to take a sample, small sample bit of what surgery we perform, वो 3 घंटे वहाँ बैठा रहता है, ठहरा रहता है, ऐसे ही वेले जैसा. That's not that. That's not the kind of respect which we have to give to our JRF, SRF, scientists, and everybody. Let us come out of that again. Remove that ego, and then we have to reach out to the people. Now, the most important thing is the collaboration between institutions. There are many institutions in our country which are very good in the basic sciences. They are doing only publication-driven research. वो अपना impact factor दिखा देगा. ICMR is very happy. We have a center which is given so many crores of hundreds of crores of rupees. They have given so much of publication, so much of good work. That's all. Beyond that, no. And there are people who want to do something, but they don't have the equipment, they don't have reagent, they don't have scientists. The clinician people, they're all, the waiting list of the institutions, that, it doesn't end. So, somewhere we need to hold together and then collaborate with these basic institutions and the clinician institutions. In fact, I should not be saying this, one of the proposal which I am giving to the people is that let us dismantle the entire ICMR and give it to the nearest medical college, whatever research institutions you have. People have to work together. The DG ICMR will hang me. But instead of dissolving, let us dissolve those silos and reach out to them. That's all we can do. Easily go to them, utilize wherever we are, the nearest ICMR institution, DST institution, DBT institution, CSR institution, medical institution, research institution, regional centers of cancer and everything. Please reach out to them and then sit together and say that, we have the strength in the clinical side. Our strength is number of patients. This is a particular disease which is very common and exclusive, unique to this particular region of the country. Is there anything which we can do together? Please focus on that, collaborate with the people, and then bring the picture. Whatever we do, the beginning of that is a collaboration. What is the end? End result is the social impact. See, publication will come. When you impact, when you do a policy change, when you give advocacy, when the Niti Aayog listens to you, when the ministry listens to you, when the ICMR listens to you, when everything happens, the publications will definitely follow you. So let us not go into just publication and say that मेरा publication हो गया सारा खत्म बात एक ICMR को report दे दो publication done look for one more grant. Not. Please look into the social impact. What is that? Look into that. The kind of money. The money is going to go down and down over the period of time. In fact, ICMR has done a wonderful thing again of the ICMR where they have cut short on those individualistic projects now. They are more focused into the one which are collaborating. They are more giving funding to the projects which are going to address the needs of the country. What are the problems of the country? Which is going to have something to do with either prevention or diagnosis or treatment or something like that. That you must have seen it over a period of last at least few years. This team has done a wonderful job of giving the importance to that. Gone are the days where, people used to get the it. And you must have also seen that, let us be very clear to that. There is a clear change in the ICMR focus where it is not driven by the name and fame of the institution. You must have seen it. You look into the website and then look into the people. 15, 20 years back, some people were getting more money and today everybody is getting that opportunity is there. So all of you have opportunity if you have strength in the research, strength in the collaboration, strength in addressing, strength in the social impact which you are going to give. All this is something which is there. Then coming to the specific thing, we all the doctors, we are not interested and we don't know much about the patents. What is this going to happen? IPR, who is going to do all these things? And then how are we going to get those ideas into the commercialization? That's where these people have come now. The wonderful work done by the ICMR in the last 1 or 2 years, where they have given where the doctors will do only doctors' work, rest all they will handhold you or they will take care of that. That's a wonderful thing they have done it. So it is a time that we must understand and then take the help of that. So whatever the ecosystems and the helping hands and the teams which they have created in the ICMR, let all of us utilize this and take our ideas onto the commercialization. Otherwise, we will end up in TRLs of 3 or something like that, or we don't go beyond that. And then the other thing which they are doing now, of course, ICMR and the AIIMS, other day we had a CEO of AIIMS here, sitting here, and saying that the connect between the academic institutions and the industry, that is something which we are lacking very much. It's a time that, we utilize these people, ICMR, AIIMS, and everybody, and say that, well, here is an academic institution, has got robust minds, best of the minds, best of the equipment, lots of bubbling ideas, young people, but we cannot we can't handshake with the industry partners, especially the private people. The country cannot become a developed country. Viksit Bharat is what? Viksit Bharat is we have to be part and parcel of it. How are we going to get that particular equipment which are on ICUs and HDUs, operation theaters today? 80 to 90% of equipment is still imported. The best of the brains are there, equipment access we have. Let us go back to these organizations and tell them that, we want to bridge that gap between the academic institutions and the industry partners. Give the feedback to the people. The best of the equipment in this country cannot come on day one. We will have to give feedback. Who is the best person to give the feedback to our industry partners, the government institutions, private industry, to tell them that, your equipment is at this level of the international quality benchmark and standards. You need to go up, do the testing, do the validation, give the feedback to them, do the different versions, newer versions, international standards. And that's how we can build this nation. Viksit Bharat is all our responsibility. We have to work together for that, especially on the medical devices. That's a big area, because most of our dependency is from abroad. So with this, I once again thank you from the bottom of our heart, from the AIIMS, and for all the people here that for having brought all the people at a short notice, and then giving us opportunity for everything, right from research methodology, going up to the ethics, funding opportunities, what kind of have it, what is patent mitra, what about the devices which we are doing it, how are the various organizations, MIDAS and everybody is going to work. All this is something which they are going to give us. And then my request to is that the people who come here is not just we are going to take for ourselves. You are the brand ambassadors. When you go back to your institution, when you go back to the region, you spread this message that what you have heard here and what are the opportunities which you are seeing, parcel of building this nation. Please spread that and then come back. They are willing to do the similar program again after some time. That's what I have been told. So take these people again and then bring more and more people and then be the brand ambassador of a change. The change to say that, we break the silos. The change to say that, we are going to collaborate. The change to say that, we are going to do a high social impact led research and innovations and be in part and parcel of decreasing our dependency on the imported equipment and the devices and the equipment and everything. And that's where you are the brand ambassador. Please, when you go back, reach out to the people and say that, what you heard today is something what needs to be practiced there. Again, thank you very much, ma'am, and welcome all of you to the institution. Thank you.
Thank you so much, sir, for your very warm and encouraging words. As a mark of gratitude, we would like to present mementos in appreciation of their gracious presence and insightful words. I would like to invite Mrs. Manisha to present a memento to Dr. Srinivas.
ICMR.
Next, I would like to invite Dr. Suchita Markan to provide a memento to Mrs. Manisha. I would also like to call Dr. Suchita, mentor to Dr. Alka. Thank you so much, ma'am, for expressing gratitude so explicitly and for acknowledging everyone who made this event possible. That end of our inaugural session. Ladies and gentlemen, we now invite you to join us for the inauguration of the exhibition. You are also warmly invited to enjoy high tea in the dining hall following the inauguration. We also request you to be back by 11:15 a.m. when the first technical session starts. Thank you. Welcome back, everyone. We now begin with our first technical session of the day on strategic intellectual property, technology development and technology transfer from idea to impact. To guide us through this, we are pleased to have with us Dr. Suchita Markan, Scientist F, Head Innovation and Translation Research Unit and Mission in Charge, MDMS ICMR. Dr. Suchita Markan is a scientist and mission in charge in diagnostics, mission secretariat, MDMS and head innovation and translation research, ITR, unit at Indian Council of Medical Research, ICMR, Ministry of Health and Family Welfare, Government of India. Dr. Markan is an expert in biomedical research, public policy and innovations in the area of medical devices and diagnostic sector with over 18 years of experience. She has led development and implementations of strategies and policies for promoting innovation and entrepreneurship with wider public health impact. She is leading the National Mitra and Medical Innovations Patent Mitra. She is a thought leader and has represented country's health agenda, particularly from medical devices, diagnostics, innovation and entrepreneurship perspective at national and international forums. She also serves as an executional committees in this sector. Recognizing her contributions, Dr. Markan was selected as an emerging leader for biomedical innovation and entrepreneurship under International Visitor Leadership Program, IVLP, of the United States Department of State. She has also been nominated for Leadership Exposure Program in Health Innovation Management of Cornell University, New York. Dr. Markan holds a technology from PGI-MER, Chandigarh, and completed her Executive General Management from IIM Lucknow and Executive Program in Public Health Policy, Leadership and Management from AIIMS, Jodhpur. She is a registered technology transfer professional certified by AUTM, USA and STEM India. She has numerous national and international publications to her credit. Thank you.
We'll have to make the introduction short maybe. So thank you for giving me this opportunity. So with this we'll start with the first technical session and I'll take you through medical innovations, patent mitra and other things which we have been talking in the inaugural session in depth so that you understand how to leverage this opportunity. So you can see in the topic it's too detailed, lot of things to be covered. So I was thinking in the morning how to cover it. So one way was to tell you about different forms of IP, how it works, what is this, what is that, what is IP, what is infringement. I'm sure that will become very, very difficult for you to understand. Lot of words, lot of jargons are involved. So then I thought of the ultimate objective of the program wherein we need to make you understand how to develop a commercializable technology lead, how to evaluate those technologies, how to protect them and other things. So to make it relevant, I thought I'll take you through the technology development cycle, which I showed you in my introduction. At every stages tell you what forms of IP are relevant, how do you develop a commercializable technology lead. so that it is patentable, industry is finding it interesting, because there are many nuances involved when you go through that journey. So I've tried to build that in the form of a story wherein I'm able to take you through the technology development cycle, this cycle again, and every step give you some examples on how some innovators have appropriately protected an IP, how they have been able to look at various aspects so that these technologies they have developed, they are commercializable technology leads. They are not academic projects, they are not research leads. They are the things which had the potential to become a product, and ultimately they were transferred, and some of them are being sold as well. And some of those examples are from A, so that you can correlate much better. So I've tried to build my presentation around these 4 sections, wherein the nuances you need to keep in mind when you are at ideation stage, team building stage, when you are at ideation stage, when you are identifying technologies or the ideas, the products that you should develop, invent phase, and then implement phase. So these 4 steps, and what are the things important during these stages, I have also tried to some words that during team first phase of the technology development cycle, when it is time for team building, collaboration and inventor engagement becomes important. At the identify phase, broadly, if we have to see, you don't need to reinvent the wheel, you need to do something new. How do you identify what matter is what I'll try to cover in this aspect. In the 3rd stage, I'll tell you more about technology transfer and how Patent Mitra can help you with tech transfer, and then translation and sales. So let's get started. So first of all, let's start with the basics on what is an intellectual property. As the word says, intellectual property means something which is coming out from your human intellect, and which is not something plain science. It is basically something which has a commercialization potential, so basically that commercialization protection, or something which can be used in commerce, which can be sold, is very, very important. And generally we evaluate these technologies through two, three pillars of strengths we have to see. One, of course, any invention, it is not always patent. An invention or an IP can be of different forms. It can be copyright, it can be trademark. So all these things also I'll be telling you. But something which is coming out from your human intellect, it is novel, it is inventive, and it has, it can be something, literary artistic work is also one form of IP, but something box thinking which comes to your mind and which has a commercialization potential, which has something to do with commerce, is broadly is an IP. First part of technology development cycle, it starts with collaboration and inventor engagement. People in this room, we will definitely feel that inventor engagement, you are not very particular when you start an invention, because we are more used to of working in an academic setting. Who work in your lab, you feel, okay, I mean, these 2-3 people, we have written a project, so we are inventors. But when you have to develop a commercializable IP, you may have some engineers, you may have some doctors, you may have people as inventors from different labs, different departments of an institute as well. So other part of it, but institutes also, you might have multiple institutes. So what we have seen, that any invention always starts with in good faith. Fine, we are friends, so let's start building something. But when it comes to developing a technology and you have inventor engagement, you need to be very, very clear on who are the inventors. It's not typical researchers. That is the time you need to be very clear that who are the inventors, who are the applicants, who are the collaborators, because they will have ultimate stakes in the IP when you file a patent application. So this is one point I wanted to tell you, that when you do interinstitutional collaboration, 2-3 institutes coming, develop a technology, you must have some interinstitutional agreement in place. While you have inventor engagement, you should have clarity on who is doing what, so that when ultimately you file a patent, you should be able to understand that they have significantly contributed to the invention. Therefore, they deserve to be inventors in your patent, because these things do matter when it comes to patent or forms of IP that we are going to talk about. Other important aspect is IP ownership clarity you should have. These 2 things are too much. I mean, licensing clarity, inventorship clarity, this is important, because I'll give you an example of a real life example which came to us from ICMR-Medical. This was a team of inventors who came to us and requested us, can you help us in filing an IP for this invention? But when we dwell deep into this invention, we asked these inventors that what kind of funding you have gathered to develop this product to the stage where it is right. So then they told us that they received some kind of a grant from Department of Biotechnology. This technology was developed in an institute in Mumbai, but inventors were, the work started through the time. So the PhD happened in a CSIR institute, then it when the faculty, that student became a faculty at ICMR Institute, then stakes of ICMR came into picture. And so therefore there was a CSIR co-inventor, there was an ICMR, DBT had stakes because they had given a funding, so there was a grant agreement which came from DBT also. So we had to, since the technology was ready, we were ready to file the patent, but we also had to think from tech transfer perspective. So we requested the inventor to get these things cleared. You need to get an NOC from DBT BIRAC because they had, as part of the funding agreement, they had those stakes. The inventorship clarity, we requested the ICMR Institute to get NOC from the CSIR inventor, because if ICMR will be doing tech transfer, maybe through patent mitra, nobody should be coming back to us and say that it was a CSIR technology, inventors were involved, DBT had funded. So we had to get all these things cleared. So after getting the NOC, after getting the go-ahead from DBT in email from them, we were able to file IP, we were able to transfer this technology, and recently in Bharat Health, we did expression of interest through Patent Mitra, and we were able to transfer this technology. But clearing these out, these took us more than a year to clear all these things. If we would have clarity in inventorship, ownership, all those things early, basically if Government of India from DBT had given grant, but they would have given licensing rights to ICMR already, because revenue sharing will be equal, that's not a challenge. But two parties coming together, who has the licensing rights also becomes important. If we do EOI, we should from DBT also. So clearing those things took us more than a year, and now we were able to file a patent, we were able to transfer this technology. But for you guys, if these things are made clear early in advance, then things become very easy. We thought that these instances should be dealt by through policies. The institute should have very clear-cut IP policies in place who defines very well, wherein the rules are well defined. What are the stakes of ICMR if it is an intramural research? What are the stakes of ICMR? Who owns the IP if it's an extramural research grant? If it is a funding agreement. So we have a very clear ICMR IP policy defining different case scenarios and the IP ownership clarity as a policy document. So that is the base centers and our extramural grantees to refer to. Likewise, AIIMS also has a very clear-cut IP policy which defines various rules of the game. So that's important that the institute should have an IP policy in place, which is a Bible for the institute inventors to refer to when they develop an invention. Coming to this of the invention, wherein that team building is clear, inventorship is clear, ownership is clear. Now coming to the real work on how do you develop an IP, how do you file a patent application. So if you are very sure you want to develop a product or an invention, do it like that. You should, this is an ideal case scenario, you should try to just Google and see, maybe Google Scholar through Indian Patent Office website, that what kind of patents have already been filed in that area. Is that domain too cluttered to invent in that area? Although it's the job of the professionals, but still by doing a basic search, you'll be able to know and see that how cluttered is that domain, whether it is a good area to put your energies in developing. For 5-6 years, you'll be trying to put your energies to develop a product. So that's something interesting you can look at. From patentability perspective, there is a very specific search that needs to be done, which is called prior art search, that ICMR team can do for you in a very professional manner. So how do you come to know that this is something you should file a patent for? So these are some pointers I thought I'll share with you, which you can try to evaluate on your own, whether it is a subject matter of patent filing or not. There are 3-4 things you need to see. One, your invention, the work that you're trying to do should be absolutely novel. This is done through prior art search and patentability assessment that Patent Mitra will do for you for at no cost to you. It's a very professional search that one does through paid databases, through free databases, which we have hired. So we can do it for you. Believe that you have some invention, we can do patentability assessment for you. Second, the invention should have an inventive step. By inventive step, whatever you try to do, it should not be obvious to a person skilled in the art. I give a very simple example of, say, for example, you are going to go through a canal and it's very dark, and it has started raining also, and you're carrying an umbrella. So while going through the canal, you have an umbrella to protect yourself from rain, and it's dark. You can think of putting maybe something to some bulb below the umbrella so that it lights, and you are able to sail through the canal. Do you think it is inventive enough to be granted a patent? An umbrella and a combination of a bulb while enabling you to protect yourself and as well as from show you the light when you go through a dark place. Do you think it's inventive? It's not, because it's obvious to a person skilled in the art. Somebody not very thorough with the patent, with the IP, but who believes that mixing two things is very obvious. It's nothing very new, nothing like great science. So this is how you are able to evaluate that what you're trying to protect, whether it is inventive or not. Industrial application is very clear. Whatever you try to develop should have a commercial value, is something which is a IP or a patent worth considering. Again, the Indian Patent Act very clearly defines what is a patentable subject matter. There are few things which are not patentable subject matters, like you might be aware, mere admixture of two things. These have been done so as to protect the interest of the public, because we do not want that there should be a mechanism of evergreening of patents, so that some industries take some right on some invention and we are from using those medicines who are patent protected. So there are some things which are non-patentable subject matter defined in the Patent Act. You can go through it. So these are some examples which can help you to evaluate whether your invention is worth patenting or not. One very thing I want to tell all of you, I'm sure Dr. Vel Pandian would have been telling you day in and day out, that doing a publication of your invention, your work, before filing a patent application kills your own invention. Because you might have seen slide that novelty, absolute novelty is the key thing for your patent to be filed. If you publish before filing a patent, your own invention become a prior art. If it becomes a prior art, then you will not be able to file a patent, because your own publication will come in the search of the Indian Patent Office that it's a prior art. Which means, it does not mean that you cannot publish, because publication is our first love. I know, all of us, we really want to publish in good journals to do so. So I would advise that you file a provisional patent application. For filing a provisional patent application, it's like a very small document. You don't need to completely develop your invention and then publish. And filing a provisional patent application through Patent Mitra won't take much time, 15 days, 20 days, much less than what you can expect for a publication to be reviewed. So you file a patent application, provisional patent, and then publish. Then it's fine. Then it's not a problem. So this is something I really wanted to tell you, and you need to take into consideration. With Indian Patent Act, Dr. Unnat Pandit, when he comes, he'll share with you, there is a provision of anticipation which has come in Indian Patent Office, by virtue of which you get a window period of 1 year. If you publish by chance and later on you think that it was a patentable subject matter, you have a provision of anticipation by virtue of which, if your publication has been done in the form of an abstract or a poster in a learned society, you are able to prove that any of these 3-4 points, you will be able to still get a patent. But still, I would urge that you first file a patent provisional and then publish. Coming to the third part, you have now developed, now team has been built, the collaboration has been streamlined, you have clarity on ownership, you have been able to file a patent as well, and then you published maybe. This is the time your invention has been developed, this is the time you need to see whether one patent is good enough, or you need to develop a strategic IP portfolio around your invention. This is also a time to see whether you need to engage with an industry, because even if you have to do clinical validation of a product, prototypes can't be tested on patients. I was telling director sir also that the products which can go into patients are the products which are developed into ISO 13485 compliant facility. If it is a drug, it has to be developed in the GMP batch, test batch has to be developed before the dose can be given to a patient or a subject. which means you need to tie up with an industry. This is the time for tech transfer. Just to give you an example of a patent portfolio, how do you develop an IP portfolio? This is an AIIMS invention. It came out from the SIB program. This product is called NeoBreath. It's a neonatal resuscitation device, foot pedal operated device for resuscitating newborns. You can see how smartly this innovator has developed an IP portfolio around this invention. The technical features of this product were protected through patents. You can see a portfolio of patents around this invention. This product is named as NeoBreath. It's a trademark. So NeoBreath as a trademark was protected through other form of IP, which is called trademark. The external aesthetic features of this product, it is very unique. Somebody who just looks at this device, this can also be protected through an industrial design application. So this innovator filed an industrial design application to protect the external aesthetic features of this product. You know that every product has a product leaflet, and sometimes the product also has a software inbuilt into the product. Software is a matter of copyright protection. The softwares in Indian Patent Act, as per the IP of our country, can be protected through copyright. And this product leaflet can also be protected through copyright application. Again, if you make some kind of a visual demonstration of this product, this can also be protected through copyright. And still something in this product is always kept as trade secret with the company, which is generally protected through agreements, confidentiality agreements. So this is how you try to smartly develop an IP portfolio around your invention if you are very sure that your invention is a product which has a commercial potential and you believe it can impact the people at large. Another example of a product, very simple product, Oral-B brush, many of us electric brush that we use in our houses. Again, this has a complete patent portfolio, IP portfolio around this invention. Oral-B IO, this is protected through trademark. External aesthetic features of this product, how bristles are arranged, very differently they are arranged. You can see there is a left turf as well in these bristles, is protected through industrial design application, the look and feel of this product. Some of the advanced products do have some software aligned with it, which is protected through copyright. And it's not only one patent, there are around 250 patents which have been protected. A huge patent portfolio nationally, internationally has been built by the company to effectively protect the technical features of this invention. So this is how industries work. I gave you an example of a startup. I gave you an example of an industry trying to build an IP portfolio around your invention. So this job we cannot do. We can just do basic thing. Then let's transfer the IP to a company who is able to think very deeply and see how they can build a IP portfolio around the invention. Another important point I wanted to tell you is that the IP patent rights are jurisdiction specific. If we file a patent application in India, we are able to gain protection in India only. So therefore, you also need to think from global perspective, where all your invention will have potential. If your product has a potential to be sold in America, US or Europe, then you have to file separate patent applications there in different jurisdictions. So again, I would recommend that at one stage we need to give it to a company wherein they already have some marketing channels, they already have their established markets there. It's their job to do a strategic IP filing in different countries. I'll give you an example. These are hands-on experiences and examples which keep coming to ICMR, and they generally come from medical fraternity. So they become relevant for you all in this room. This invention for a kit for Kala-Azar diagnostics came to us, and the inventor had filed application in India and US from neural research. And when we reached out to company, nobody was interested to take this technology. Why? Because you know Kala-Azar is most relevant for Africa. So we need to think strategically on where to file. Application filed in India will have no value for an African country because territorial rights are there as when we talk about patent. Likewise, there was a therapy which was developed for obesity and patents were filed in India, China, Japan, Europe. While you know that Western countries have the most major problem of obesity. No application was filed in US, Australia. So the companies which came up had more interest to look at these jurisdictions, but the timeline had gone. So this deal we were not able to materialize. I'm sure people in this room would know that the technology developers, even from IITs, they can develop technology till a stage. Then you need to engage lot of other maybe service providers, consultancy firms who have to be engaged for developing the regulatory compliant test batches which can be taken for studies. So this is a AIIMS example, and I think Sandeep Singh is involved in this product, an intraosseous device which was developed by a startup company coming out from SIB program. This product, we engaged in this product would not work very well during its initial stages because there were many components in the device, so scalability was a challenge. We engaged Lunar Group USA for industrial design finance product. We engaged a contract manufacturing agreement with them so that the inventorship and everything is secured with the inventors of this invention. We engaged Uttejana company for engineering design and testing. Again, and for regulatory guidance, we engaged UL Group Bangalore. Again, we had some kind of a confidentiality agreement with this team. So we were able to engage multiple partners to do different tasks for this product, but we were saved from our IP being lost because we had different kind of agreements being executed with different parties to protect IP ownership and inventorship. And nobody was able to reverse engineer the product. And since this was a startup driven product, lot of patents were filed in different jurisdictions and they have been granted as well. So this is a name story through a startup. So these are the kind of agreements you need to sign, and you can refer to ICMR guidelines for technology development collaboration. We have put up the key documents of ICMR and AIIMS pertaining to IP in your pen drives. So pen drives which have been shared with you all, they include IP ICMR, AIIMS, technology transfer guidelines, and at the end of one document, the technology development collaboration guidelines, you can see 4 standard agreement templates. So those standard agreement templates you can, when you try to engage with the companies, maybe some contract manufacturing firms, so that you don't have to do it for yourself. So we have tried to standardize some of those agreements and shared with you as part of our booklet. Coming to the third part of tech transfer, now you have developed the technology, maybe we have some strategic IP protection also done around your invention. This is the time when you need to join hands with the industry and do transfer. This tech transfer business is also done by ICMR, National Medical Innovations Patent Mitra. So what is tech transfer? In very, very basic language, technology transfer means the transfer of a technology from an academic institute, an institute who is the key technology developer, to an industry. The industry is the one who is able to commercialize. The parties from the party or the people who are good at developing invention to the party or company who knows their business well of technology commercializing the invention. So it's about joining hands. And here again I want to tell you that tech transfer is from an academic institute is always collaborative. We will never be able to fully develop a product which when given to a company they are able to sell. So there are a lot of things we have to do even after tech transfer. You should believe in yourself because we do tech transfers as part of ICMR. So you need to do lot of post-transfer monitoring. Lot of engagement happens between the inventor and the industry. Then only the industry is able to take those technologies to the next level. Don't wait for the situation wherein your technology is fully ready and then you do tech transfer. There are different forms of tech transfer, but we will be telling you only about the vertical tech transfer which is done from academia to industry. And it's a very diligent process. These are the steps you need to follow: preliminary screening, evaluation, valuation, marketing. So those, just to summarize, we have gone through the entire pathway of technology development and commercialization, and you can see that IP is inbuilt into the entire process. So it's not that you just file a patent and you are free. You are diligent at every step of technology development and commercialization, and different forms of IP tech transfer, they will become relevant as you keep developing your technology to the next level. So that's one message I wanted to give you. Now, changing gears, ICMR can help you out. I understand when we talk to the doctors that they are, in fact, all biomedical researchers filing patent, doing evaluation, all these becomes very difficult for you guys because it's very understandable. You are very good at doing what you have been made to do at. Very comfortable. We are not very comfortable in that. So filing patents and tech transfer, when we talk to the innovators, they said they are very difficult jobs, we can't do it. So there came Patent Mitra, wherein we will try to make your journey easy. I'll take you through Patent Mitra, although madam has told you in the morning, Dr. Alka Sharma told you in the morning, but I thought I'll still tell you about why we started Patent Mitra initiative. In my few slides now, I'll be telling you about the W's and H's of Patent Mitra, so that after 20 minutes or 15-20 minutes of our discussions, when you go out from this room, what is Patent Mitra all about, why we started this initiative, how to use the portal, and what we will do for you. All these things will become very clear in these 15 minutes from now. When we started this journey in March 2025, we looked at some of the statistics and came to know, we started with the good news that yes, India is doing very well when it comes to Global Innovation Index. We rose up from 91 to 39. It's a great for our country to feel proud of. But if we look at the numbers of how much we are contributing as a country vis-a-vis other countries when it comes to patent filing and technology transfer, friends, our country contributes 2.4% of the global patent application filings in 2022, as against China, which contributed to 46.6% of global patents, and US, which contributed to 18.7% of global patents. So when it comes to things, we are relatively low in numbers, and when it comes to tech transfer, it is even lower. So these are some of the things we referred to before we launched this initiative. These two very, very important for you to ponder upon. This is particularly relevant from biomedical innovation perspective, because ICMR is a custodian of biomedical research. People in this room also bother only about biomedical research, which involves all therapeutics, vaccines, device, diagnostics, molecules. So if we look at the number of patents which have been filed in the biomedical sector vis-a-vis for all the patents which have been filed in Indian Patent Office. So this is an Indian scenario. We contribute only to 7 to 8%. The biomedical sector contributes only to 7 to 8% of the total patents which are filed in Indian Patent Office. This is the last 5-year statistics, which means the number is very less. The percentage of being filed in the biomedical sector is very less. And if we look at the percentage of biomedical patents being granted by Indian Patent Office in the last 5 years, it's even less, just 2 to 3%, which means maybe we are not filing quality patent applications. One, we are filing less numbers. Second, the applications being filed are not very good in quality, because that's why they are not being granted as well. So these statistics we looked at, and Honorable Nadda ji, Dr. Paul, our secretary, they thought that we need to really increase this number. And this can happen only if we provide hand-holding support to innovators. Innovators don't have to do a job of thinking how do we do patent evaluation, how do we file patent application. So as Dr. Alka told you in the morning, additional DG madam, that we also looked at the different government schemes of DPIIT and other government agencies. What are the kind of IP support they are giving? So most of these schemes are reimbursement schemes. There is no fully supported scheme. This is a fully supported scheme of ICMR, wherein you don't have to spend even a penny. The patents that we facilitate filing, the evaluation that we do for you is fully ICMR supported. You don't have to spend even a penny. That's one point. And of course, we want to build that innovation ecosystem. We want to increase the numbers of patent filing in our country. I'll not go through the challenges, because I think we have dealt it quite a bit in our previous slides. That I'll just focus on what we offer. We will be providing hand-holding support. Let's focus on this slide. What Patent Mitra does, what we have covered so far is why we started this initiative. The statistics is clear, and our visionary secretary wants that to 10-folds, maybe in a year or two. Because we need to experience, we need to do those experiments in the biomedical sector, innovation sector. What we offer is all these 4 things. One, the Patent Mitra will do a detailed patentability assessment for you. The 4 pillars of patentability we will do for you. Whether your invention in the form of invention disclosure, which comes to Patent Mitra through an online portal, whether it is novel, whether it is inventive, whether it has industrial application, or whether it is fine as far as Indian Patent Act is concerned. There are no in that domain of not to go ahead as per the, it's not stopped by the Indian Patent Office. So that patentability assessment in the very professional manner we'll do for you, and we'll share that report with you. Second, we will do patent filing for you. We have a panel of attorneys, 10 panel of attorneys we have. I'll show you in my subsequent slide. We will do prosecution for you for 5 years, and we will also do tech transfer. That's the very, very important part. It's not about increasing the number of filings, it's about increasing the number of quality patent filings. Only the inventions which pass the patentability assessment will be filed by us. Anything and everything won't be filed by us. We will do patentability assessment. Those which are able to pass that test will on filing. So that job will do. And who all can avail this facility? The name itself says Medical Innovations Patent Mitra. It's for medical institute innovators only as of now. It is for all 700 plus medical colleges across the country, ICMR intramural grantees, our 27 institutes, all ICMR extramural grantees. And this is the plane that we built during the launch of this. This was Dr. Paul's vision that the people should believe in you. It should not be a group of scientists sitting in ICMR doing it for you. People should believe that this patent drafting and filing is the job of professionals. It cannot be dealt by anybody and everybody. So we have a panel of attorneys, 10 panel of attorneys have been empaneled with ICMR, and out of which we counted there are 350 plus, 340 plus registered patent agents. We have registered tech transfer professionals who will be doing this job, IP attorneys. Fine, and you can just be very sure, and so I just want to convey that it is a group of professionals who will be handling your work. And you can see that this plane is running on the runway of Indian Patent Office, which means Indian Patent Office is a very, very strong collaborator to this initiative, and we have Dr. Unnat Pandit coming here and sharing his thoughts with you. How to reach out to us is just a click of a button. Everything is fully digital. You just need to go through this website and click here. While you click here, you will see that you will see two sections. First, you need to first of all register like you do in any application. You need to do registration, and you need to give a registered email ID wherein you will receive a password. It's a very basic thing. You need to register. With those login credentials, you need to enter into the portal. This is how you will receive the email. You need to use this user ID and password to enter into the login page. Once you do a entry into the login page, this is the main page you will see. You will have very simple patent facilitation cell, and you will have a tech transfer facilitation cell, kind of a section. Going to the patent facilitation cell, this is the sheet you will see. It's a very simple invention disclosure, very standard aspects which you would see in any invention disclosure. You need to tell us about the inventors, you need to tell the applicants, then you need to tell us about the technical details of your invention, how your invention works. And if you have gained any kind of funding, because we want to clear that applicant part of it, that whatever we do as Patent Mitra, we are very sure on who are the applicants. That's why we'll ask you about if you have received any kind of funding, if you have received, enter into any agreement with the government or any other party. Since patent filing is a serious business, it's not like a publication, we need to also, this Patent Mitra is innovator centric. Everything has to be done by innovator. But you can't leave your director as well, the authorized signatory of your invention. You also need to take him on board. So therefore, we have developed some kind of an undertaking or NOC you need to take from your director. But that will be taken by the inventor himself. So inventor will show this standard format to the director's office or maybe Dr. Vel Pandian's office, and he will facilitate this NOC or undertaking from the director's office, so that he is also informed that this patent is being filed by the inventor of the institute. So that's it. This undertaking, once you get signed and upload, that has to be uploaded, and then you submit the invention disclosure. Once the invention disclosure form is submitted, this email will come to your registered email ID that your invention has been submitted. And then somebody from our team in some days will approach you and try to understand. We'll fix a meeting with you to understand your invention. Then the real work will begin. We'll go through that invention disclosure and then discuss with you to understand what is your invention, bring an attorney on board and do a three-way meeting to understand your invention better and do patentability assessment. So that patent filing part is done. Now how do you become very sure that your invention is at a stage you should approach us for tech transfer? Another point I want to tell you that patent and tech aspects, both these windows of Patent Mitra are alone. I mean, they are not fully joined. You can come to us only for tech transfer help, and you can come to us only for patent help. If you have already filed patent, you want to take help from Patent Mitra for tech transfer, that is doable. It's not mandatory. It's not a bureaucratic kind of a framework wherein if you file patent through us, only then we will facilitate tech transfer. They are standalone. So that one point I wanted to tell you. How do you evaluate that your patent or your IP is ready for tech transfer? There are two important things you need to keep in. The IP filing is done for inventions. This can be something early stage as well. Technology transfer is done for technologies, not for patents, not for a piece of paper. Your invention should become a technology when you come to us for tech transfer. So for example, you think of developing a kit. This is how the kit will work. You are ready for patent. Now you have to develop the kit and evaluate it in-house and party validated also. Say if AIIMS Delhi sends the kit to AIIMS Raebareli or some other institute to evaluate this invention that it works very well. We want to be very sure that from the biomedical settings, if we do some tech, should be working technology. Some industry should not say that it didn't work in their facilities. So that these are the two important things we wanted to be very clear, that patents you do for IP, but tech transfer. So the invention should be at an advanced stage when you come to us for tech transfer. The second part, again, it's very basic. This is the form you'll see once you come to us for tech transfer request. The information which I told you, that is only being sought through this form. We would like to understand whether you have filed a patent, whether this invention is validated in-house or whether it is third-party validated. This is the information you need to put in this form. For us to evaluate that it is ready, some information you need to give to your director. And this is how we keep reaching out to different places. We went to PGI, we went to Mohali, and many institutes have taken help from this initiative. We are proud to say in last 6 months. And we keep reaching out to various institutes so that they can take use of this facility. So just to summarize and break this presentation is this is the wheel of IP and tech transfer which we facilitate through Patent Mitra. And why we were able to do such a good job in 6 months, we are very proud of facilitating all this, is because we had made our base very strong. You can build a strong building only if you have a good base. We worked for more than a year to develop these guidelines, policies. ICMR built on, we developed these technology guidelines, we developed IP policy. Dr. Vel Pandian has been there throughout with us. We developed CSR guidelines. All of them were formally launched by the minister, by the state minister, and then we launched the Patent Mitra initiative. So we did this groundwork for 2-3 years. Then we launched this initiative, and as madam told you in the morning that these were the pre-launch efforts, behind the stage work that we did before we launched this initiative. And this is in 6-7 months our outcomes. We keep on presenting our report card to all the innovators wherever we go to make you believe that we mean business. We really want to help you out. So that's all from my side, and with this I would like to welcome you all to leverage this opportunity and use this opportunity, talk to my team sitting outside who will help you out and even take you through the portal. With the portal live. It's there outside, and thank you very much for your patient listening. If you have any questions you want, we can give them 5 minutes, or you want to take them? Okay. So maybe I'll be around only. Any questions you have, I'll be very happy.
Thank you, Dr. Markan, for that powerful and informative session. I would like to invite Dr. Vel Pandian to please provide Dr. Markan with the memento. Moving on to the next topic, framing research questions, objectives and hypotheses. We are privileged to have Dr. Anand Krishnan, Professor, Center for Community Medicine, AIIMS with us. Dr. Anand Krishnan is a professor in Center for Community Medicine, AIIMS, New Delhi. He has more than 350 publications. He leads Minerva Network, former million death study, which assigns using verbal autopsies done under SRS Office of the Registrar General of India. He is a member of WHO. He heads the WHO Collaborating Center for Capacity Research in Community-Based NCD Prevention and Control. Can I please invite you for the talk, sir?
Thank you. Once we are running late, I think this one way to cut time is to stop long introductions of the speakers. Okay. First, I will start with an apology, but before that, I think it was an excellent presentation. Even I learned a lot of things in the previous presentation. And the apologies is that, and this is a request for Dr. Vel Pandian, that I come from a public health background. So I think this question should be more oriented towards the way of technology development and device. So I have not done that. So that's why I'm apologizing in advance, because I didn't. Yeah, so but I think in future it might be good to even organize the workshop around those phases of how this the process of development occurs, right? So I will see if I can how we can change and give some examples which are more appropriate for this situation, but apologies in advance in case I fail to do so. And I have not patented anything, so I'm not even sure I'm the expert for this workshop, but since he asked me to come, I am here. Okay. But ultimately, all of us are researchers, and my point, question would be that when we start a research, in whichever area that you are working in, we should start with a very clear question. So my whole effort is how to have that very good research question, whatever that question may be. It could be a clinical question, it could be a business question, it could be a public health question. But having a good question is the first step in doing research, right? So that is very critical. And if you don't have a good questions, rest thing will, even if you do a very good job, they are not going to be very useful, right? It may not get published. Obviously, it will not get patented and other things. So that's I'm very fond of Alice in Wonderland, Lewis Carroll. So it has very lot of good sayings. So if you remember, it says the Alice asks the Cheshire cat, which road should I take? It says, where do you want to go? She says, I don't know. Then he said, doesn't matter which road you take, right? So exactly, so it's like a research question. You have to know where you want to reach. If you don't know where you want to go, it's going to be waste. So have a very clear idea of what you want to achieve at the end of this project is the most important thing for any researcher, right? And whenever we evaluate a research proposal, a research question and hypothesis is a very important decision in whether we decide to fund you or not. How well you have arrived at your research question, what is your research question, and how well it is framed. That's a very critical point for decision making in whenever you apply for grants. So we all are trying to, research is basically process of generating knowledge, and one of the things at least we believe is that there are multiple truths, especially philosophically. Scientists may not necessarily always agree. They sometimes feel that there is only one truth and that's what I am going to discover. I am going to be the, but ultimately the world has multiple truths, right? But the way the science works is that we have to prove something. Otherwise, unless proved, everything is, it does not exist, right? Knowledge exists only if it is proved. So that is the way that the system works, the science works, and we need to follow. So we start with an hypothesis and we try to prove it or disprove it, whichever way. And as I say later, whether you prove it or disprove it, science still gains. It is not that if you only prove an hypothesis, science gains. Even if you disprove an hypothesis, science still gains, right? So all research studies start from a question, And the main difference between hypothesis and research is hypothesis is something is predictive, and I will come in more detail later, whereas research is more inquisitive. So starting with research question, as I said, it should be clear, concise and specific. The scope of your project. So if one of you wants to say, give a research hypothesis or objective or research question, we can take that as an example which you think is relevant for discussion here. I was talking to one of your gynae oncologists from JIPMER, trying to develop a device for diagnosing cervical cancer, right? We all know, anybody in public health knows that cervical cancer is a major problem. We also know that diagnosing, because the current methods of diagnosis, especially if you do, it requires a room, a privacy, doing a pervaginal examination, collecting a sample, testing it, it is too cumbersome. Our cervical cancer screening rates are below 5%. So definitely the country could do with a technology which solves this problem. So clearly an identified need, right? So that's the first thing. You have to clearly identify a need that is required. What is the problem you are trying to solve? And you should be able to explain it in simplest way. Like I said, it's a very simplest way. Anybody will agree, yes. If you have done this, your homework well, you should be able to do it. There was another person, Dr. Hari Prasad, he worked with me a long time back. He has his patent kept outside. He's trying to develop a point of care device for dopamine measurement. Because he's trying to see whether in patients with Parkinsonism, which is dopamine deficiency, if you overdose, they move towards schizophrenia. If they have schizophrenia, which is a dopamine overdose stage, and if you treat them, they move towards dopamine deficiency, towards Parkinson's. So you have a narrow range to treat. Currently, we don't monitor. Most patients, don't get monitored for dopamine. It is too complicated for a routine clinical measurement. So he's trying to, it is work. So again, a very clearly identified problem, and you are trying to solve. They both are trying to develop a technological solutions to either a public health problem or a clinical situation. So that's the first. So based on that, you will define your first question. In this case, since I come from public health background, my question is, you can say this is quite similar to the example. This one, I say, how well is the disease controlled in patients of hypertension in India? And what are the reasons for poor or good control? I'm keeping it open. It could be good control. I should not assume without measuring it first, right? My first will tell you whether it is a good or a bad thing. Second, I will try to know why it is, whether it is doing good or bad. So it's a very generic question, but it's again a relevant question which we can answer. And this will, first thing we are looking at it is this. Obviously, again, you will have to say hypertension is a problem. It kills so many people. Our treatment rates are this much. And again, hypertension control rates in India are about 10-15% of people with hypertension in India are actually controlled. Again, a serious problem, and we could do with doing a better job of it. But not necessarily every problem is solved with the technology, but definitely. So this is a good question that, again, one of you wants to take an example and wants us to discuss that. I am very happy to do so. So in all the examples that they all are grounded in very good, sound knowledge base. We know the problem that is. Second is how researchable. Is it even possible? Or it is just, not all problems are possibly researchable at this stage. At some point of time, they might become researchable. But in the point of time that you exist, is it possible, if somebody will believe I am going to do this, will they think that it is actually researchable? And how important results will be? How will it change? For example, my question to Hari Prasad after listening to him was, has it changed any clinical decision making? So you can develop a point of care for dopamine, but my question to him was, okay, has this been used? And has this improved clinical treatment of patients, or had improved clinical decision making? Have people titrated? That technology is not yet reached that stage. It's still not gone there. It has just been patented and these things. But so that's how we have to make a guess of that. We may not necessarily know, but there are three ways of doing a good research question, but almost all good research questions come from the first box. If you want a good research question, you have to be a person in the field. If in public health, you have to be really engaged with public health. If you are a clinician, you have to be really engaged with patients. If you're in basic sciences, again, you're. So all really useful questions come from, as a first step, come from a actual lived in experience of either patients or doctors, right? And nothing, and if you're a good science, then you will definitely face clinical situations in your clinical experience, which will make you, why is this drug not working? Oh, this patient I lost because of XYZ. Could I have done something differently? Could there have been a solution for this? So if you start thinking like that, you have already moved towards making those corrections and identifying a good research question. You can do some literature review and then do a brainstorming with people who have faced similar problems, or you think people who have such kind of, who are thinking in the same way. So all these things are required before you really frame a good research question. They complement each other. So again, I will give you some research questions. Generally, again, this is a generic one, not specific to this. Is your question descriptive? Is it analytical? Is it interventional? Right, again, when in descriptive, you are just describing, estimating what is the level of control, risk factor or an association, and intervention obviously an impact of a treatment, right? So again, the same thing. How well is the disease controlled in patients? That's a descriptive question. So I will end up with a percentage. X number of percentage of people are. Is it more analytical? Okay, why are some people not controlled? Why are some others controlled? What is that determining factor? So that looks like an analytical question. Is it age? Whatever. There you could put age. You could put any other risk factors. And then some. If one of the factors is that people are being treated with different treatments, then the introduction of a standardized treatment protocol, hypertension management. So that's the intervention level question. In general, when we discuss this question, we generally ask you to put it in a pie chart format, which is very specific. P is populations or patients in whom you are interested. What is the intervention? In many analytical studies or descriptive studies, E, which is exposure. What is your exposure of interest? What is your comparison group? And what is your outcome of interest? And time in which you will do. So you can take examples later on. But remember, pie chart does not help you tell whether it's a good question. It only tells you that it's a good framing of the question. You have put all the matters. It does not necessarily mean that your comparative group is right. You could do a good citizen group, but yet you would satisfy pie chart criteria. So pie chart criteria is not a criteria for deciding whether your question is good. It's a question whether you have addressed all the key variables which are important in deciding whether your question is good, right? So that's why I say it does not need any special. I just have to see, okay, is there a P there, is there I there, C there, I can make that. Even if in an area where I have no interest, I have no expertise, I can still decide whether you have followed pie chart criteria. I have no idea whether it's a good question, relevant question, whether it is really a useful question. But it is pie chart. Judging how good it is, this is a better criteria, right? Because it tells you the quality of your question. And so this is done by subject in ICMR is that, and this is true across globe. Your question, your proposals, grant proposals are not always only looked at by subject expert. I am often asked to, because I am a part of a committee, that committee gets all kinds of proposals. I am also asked to look at some biochemistry proposal. I have no idea how many of them, right? But I am supposed to give marks to them. So, but that's our, so it's not, because I cannot decide whether a question is innovative or interesting. I am not a neurologist. How do I decide whether a neurological question posed? A neurologist might say, oh, this is nothing, everybody knows this. I might find it very innovative. Oh, बहुत बढ़िया काम है. लेकिन person working in that area may say, no, there is nothing new. Everybody knows this. So that's the challenge of this thing, right? So how novel, interesting is something that only experts can tell. I can tell about maybe others, ethical, feasible, but not really interesting and novel. That's a subject expert has to do. So the challenge for people applying for proposal is to, your target audience for this is a non-expert. So remember always, when you make proposals, think always, in any communication we should think the worst case scenario. So worst case scenario is that your proposal is being evaluated by a non-expert. So your proposal should be written in a way that that person is able to decide this is innovation, right? Hypothesis. So hypothesis, I say it is primary intent of statistician, but actually hypothesis is the scientific way of looking. It is the way a scientist will look at it, not a clinician or a public health person or a, right? So and when I said earlier that it is the predictive in nature, so this is a question which is statement which is predictive. Children with a high body mass index are more likely to undergo renal transplant than children with normal. So I am predicting that, right? Obviously this is not very specific, right? I need to put more details, especially if, statistical detail to make it. I have to define what is more, right, especially. So research question is inquisitive. What is happening? Why are some people, those are more inquisitive questions. Where hypothesis, what you just saw, is more predictive. Research question guides the research projects. Research question could be qualitative also, but hypothesis is invariably a quantitative way to look at things. Research question starts definitely with an open-ended approach, and it's inductive. Whereas hypothesis is more of deductive. We begin with a theory, formulate hypothesis, and then prove it or disprove it. I'm not going to go too much into the statistical part of an hypothesis, but when I say directional hypothesis, it means that I'm only looking at one being one way. In a statistical way, is it only higher or lower? Or non-directionally, it could be higher and lower. They are just different. I have no hypothesis which says it is better or worse. But directionally, I choose one of these two. They have statistical implication which I am not going into. So we start with a null hypothesis that there is no relationship, and then we collect data and apply it and see whether that hypothesis is proven or not proven. Just as a scientist, you should know that absence of evidence is not the evidence of absence, right? So again, as you can see, there is no difference is a two-sided hypothesis. One-sided hypothesis is clearly, I'm taking it to shorter hospitals, say so. Based on these, we actually frame the study objectives, especially in a thesis and all. For example, in the example that I gave, I would say to estimate the control status among the adult patients of hypertension in India. That's my, how I would frame the objective based on what I showed to you earlier. And maybe to compare the control status of hypertension between younger and older adults with hypertension. So that's how I converted my research question into an objective. So again, an example where same thing. What is the effect of a robotic assisted surgery compared to traditional laparoscopic surgery on long-term outcomes in children with pediatric renal carcinoma? Converted into research hypothesis, which is children with renal carcinoma who undergo robotic assisted surgery have improved long-term survival compared to those who undergo traditional surgery. And an objective I write to evaluate the long-term survival rates and disease-free survival in children with renal carcinoma treated with robotic assisted surgery versus traditional. So again, say all the three things are related to each other. They are just different way of telling the same thing. So again, just to repeat, identify the knowledge gap, which is the most critical. Based on then, formulate an appropriate research question. Do a good review of literature, refine your question. Based on which, draft your objectives and ensure that your framing is in PICOT format and it satisfies the finer criteria. So again, just a point of view, again, from grant manager's point of view, this is my last slide. Ultimately, wherever you do, you are going to managers look at it. First is that obviously the granting agencies should be interested in what you are doing. It is aligned to their specific needs. Ultimately, it demonstrates the applicant's client. So that's what I said. That's the most important thing. And the third one, it should demonstrate larger benefit to public and feasible. So ultimately, whatever you write, it should satisfy these 4 criteria for any grant agency to seriously look at your proposal. Examples, but as I said, those are not necessarily the correct examples. We can take some examples from the group if you want. That's all for my talk. We can take examples. If anybody wants to share their examples, if you have come to this workshop with a research hypothesis in mind, then we could work on that. So any volunteers? Yeah, please. First is very good. In many workshops I conduct, most people are very hesitant to volunteer. So thank you for volunteering. I'll tell you why you are attending this workshop. Because the question you are posing is not really related to the patent mitra initiative.
I am Dr. Sajid Dunia from PGH Chandigarh.
Okay.
I'm a pulmonologist and I work in interstitial lung disease and sarcoidosis. So that's why my question pertains to sarcoid. It's just an example of a descriptive research question where we're just asking about the prevalence of the disease in a particular.
Yeah, absolutely. So that's a very straightforward question. What is the prevalence of ILD in the Indian? Okay. Anybody else wants to go for a more ambitious research question? This can also be linked to your next session. I don't see Sumit is here. No. So he can also take it up these questions when he takes up it on your study designs. Yeah.
I'm Dr. Rupa. I'm a neurologist at AIIMS. So, something related to my work that I mentioned by IIMS. So, it could be what is the diagnostic accuracy of a particular tool that one has devised for in screening for, say, Parkinson's disease with a
Right. So this is an important question which most people will have to answer. What is your validity? How good is your technology in doing whatever it is supposed to pick up, whether it is a Parkinsonisms or ILD or cervical cancer, right? We often develop diagnostic technologies. They all have to ultimately answer how good they are in diagnosing, right? Okay. I think there's something happening from the back. Yeah, okay. They really want me to leave now. Yeah, okay, so anything else? No problem. We are running late. This is perfectly fine. So anything else you want to discuss on this question? Anybody wants to give an analysis? And any other questions related to this session? Otherwise, I will hand it over to Sumit. Okay, thank you.
Thank you, sir, for offering such a structured and insightful overview of how to formulate meaningful research questions. I would like to invite Dr. Namanita to facilitate the memento to Anand Krishnan, please. Our next session is on study design, conduct and utility, to be delivered by Dr. Sumit Malhotra, Professor, Center for Community Medicine, AIIMS, Lead Innovation and Learning Center, Comprehensive Primary Healthcare Implementation in New Haryana, Lead Implementation Research Project, Strengthen Regulatory Care for Common NCDs, Faridabad, Member Secretary, Research Integrity and Publication Ethics, Areas of Interest, Operational and Implementation Research, Monitoring and Evaluation, Primary Healthcare, Recipient of many research grants, Health Literacy, Adolescent and Young People's Health. I would like to invite sir for the talk, please.
Thank you for that kind introduction. I was just saying, let's straight away go to the talk, because I'm standing, now between, the lunch, and I'm being told that already we are delayed, right? So, and already I have got the signal that, don't be speaking too much, okay, so that I can curtail the session. I just want to ask, do we have the moving mic so that I can move hair hooked up with this laptop? Do we have? No? No. Okay. Fine. So let me start by asking, what do you know about study designs? Let us give chance to someone else before we'll back to you, but since you answered the last one, I want to pose this to others. Yes, ma'am, please go ahead. So there is one classification coming up. So that's observational and experimental. Okay. Okay, we have another, keywords, retrospective, prospective. Okay. Somebody else now wants to contribute. Yes, ma'am. You can sit and talk. Yeah. Yeah. So study design is nothing but to collect the data and to answer your question. So always in research, remember, the question is the king. Okay, if you have got the question right, everything flows from there, right? And your question decides how you are going to answer that question. So the first fundamental thing is get the right question. Once you get the right question, then which study design to use, how to solve the puzzle of answering the question is going to flow automatically. And there are plenty of designs that we have. Okay. Now suppose there is a new technology and you want to find out G is taken by general people. What are their experiences in using that whatsoever? Whatever technology you are concerned with. You have introduced it, some people are using it, you want to find out experience of using that device or technology or whatever. So how are you going to go about it? But live streaming should also capture me live. Yes. Yes, sir. Okay, case and control group. Okay, what happens there in case and control? What is that you are trying to answer for? Okay. Okay, so now we have another thing, that how many cases were benefited from using a device. So maybe your case is one who used it, and what will be your control then? What will be your control? Those who haven't used the device. Is that what we start in case control? Yes, sir. We come to you.
We need to divide it into a diagnostic device and a therapeutic device.
Okay, diagnostic device and therapeutic device.
So therapeutic device will be tested in a controlled trial where the efficacy of that device and safety will be tested between the control group and the intervention group. If it's a diagnostic device, we need to test it amongst cases and controls to get a sensitivity and specificity and other performance characteristics from it for a particular aim or objective, maybe diagnosing it.
Okay, thanks. So what all we have heard so far, we have case control, we have heard now RCTs, we have therapeutic, maybe some participants are going to be subjected to some entity. We see their performance compared to someone who is not being subjected to that or subjected to something standard that's going on in that space. and then we kind of evaluate it. So this is an experimental study, a randomized controlled trial. The hallmark of this is going to be randomization, right? Okay, you can do this also in a quasi-experimental way, wherein we don't have the step of randomization. Rest everything is the same, except who gets the exposure to device and who does not get is not very randomly decided. It's a non-random allocation or assignment that is there. Okay, right. So we have RCT. Okay, and we have discussed. We have heard about the diagnostic evaluation accuracy, wherein the test performance is being assessed, and our intent is to know the measures of validity, and the measures of diagnostic accuracy or validity are the test characteristics in terms of sensitivity, specificity, positive predictive value, negative predictive value. You require a gold standard, you aim on plotosy curves and things like that. So I think most of you are familiar with that. Yes? Yes? No? How many not familiar? Raise your hands. So most in this hall are familiar. Coming back to the question that I asked, I haven't got the answer yet. The question was different, and I got a different very answer and a different flavor. Okay, sir, you want to answer? Yes, sir. Okay, fine, good. Yes, yes. Yes, so my question was, I want to find out, what has been the experience of people in using some technology, some device, their interface, whether they are using it, not using it, if they are using it, what is happening to them. So there, as rightly been, shared by our friend, the question is not how many are using it. How has been their experience is the intent that we are interested to capture. So there, a classical quantitative survey-like approach doesn't fit in. Okay, it's a very personalized experience that we are bothered, and that design is, qualitative approach. And therein you have more of these open-ended, and you go in-depth, and you try to find out. And we are interested to find out how people are interacting with the technology or device, something new. It is their experience. Maybe you have found out a new way of delivery of insulin, and somebody who is injecting insulin knows what's happening with the new method, and that person and only can tell you the difficulties that that person is facing, or maybe if that person was using another approach, how do they compare in his or her experience. So there we are not interested in using that approach. We are interested in how is that being perceived by an individual who is using it. What is the experience? What is the user experience? And that is another kind of a study, approach, and that's called as qualitative study designs. So the first fundamental classification that we have in study designs is qualitative questions, quantitative questions. So all your questions largely were how many. That how many and a numeric answer, your approach is going to be quantitative. And all your study designs that you elaborated were actually quantitative study designs. Okay, so the research question, is it starting with how and what? Why? Then you have a qualitative approach. If the fundamental question is how many are new technology compared to an old standard methodology, it's how many, it's a quantitative, you'll get a number. So wherever the expectation of an answer is in terms of number, it's a study design. Is that okay? Is that point clear? There is something qualitative versus quantitative. Often we are resorting to mixed methods now, wherein we incorporate both qualitative and quantitative. Right? Now then comes your quantitative, the basket of designs that you have and the recipe, and madam shared that we have observational and experimental. So observational and experimental, what is the basic difference? Yes. So where you observe and where you intervene. So in an interventional study or an experimental study, you are creating an artificial environment because of intervention. You are exposing people to intervention, and in that artificial exposure of new technology and device, you are making measurements which is under your control. So that becomes, the question for experimental studies or the interventional studies. Sometimes you cannot randomize patients. You are not allowed to do certain experiments wherein you have to just resort to normal what is happening. Like for example, you can't say that I will expose people to air pollution and study their lung health. You can't say that, right? We already know it is toxic, it is deleterious to health. Right? If you are living in Delhi, you have to live with it. So all of us are exposed. So anybody who wants to examine our lung health is welcome to do. Is that right? So this is I can't expose anyone to do, smoking and then say, I'll measure your cardiorespiratory fitness after smoking. It's an unethical question. Right? So we can have, for certain things, observational. Okay? So some questions can only be answered by observational. So when maybe OT decides or an hospital decides we are having this kind of a device, you don't have a choice, everybody will get this. So there's no randomization, everybody is getting exposed to that device. Whatsoever surgery or operation is happening with that device. So you can observe what's happening after the device is being used in all of them post-surgically, right? So study long-term outcomes, short-term outcomes, whatever outcomes you are interested in, right? And that makes the case also of having a cohort studies. So what is a cohort? Cohort is a group of statistics. Maybe all people getting operated inside one kind of OT by one surgeon, the cohorts of patients operated by that person. Is that right? Okay, so cohort is anything. I mean, all faculty in an institute are cohort. Whatsoever the institutional policies happen, everybody is exposed to those policies, good or bad. Right? Okay, so cohort. Right, so observational studies, largely we will have, cohort, case control, cross-sectional. Now, there's another very important thing that you have to understand is there are certain designs or studies that will be hypothesis generating and some study designs that are going to be hypothesis testing. In previous talk, Professor Anand was talking about hypothesis and formulation of hypothesis. So if you have hypothesis in mind and you are testing hypothesis, usually we use analytic study designs. So there's another word, analytic. Okay, so we use analytic study designs, and the analytic study designs primarily will always have a comparison group. So the fundamental thing that goes with analytic and descriptive, so the opposite of analytic is just descriptive. So most of the descriptive study designs that you have are going to generate hypothesis, to test hypothesis. Is that clear? So we have hypothesis testing studies, hypothesis generating studies. Is that clear? Whenever you have hypothesis testing studies, you have some hypothesis, prior hypothesis, this device is going to be better in these set of patients compared to the standard whatever is in use. This is an hypothesis, this is directional hypothesis wherein I know that this is going to perform superior. This, whenever you have an hypothesis testing question in mind, you need to have an analytic approach, and analytic study designs will always have a comparison group. A study that has got a comparison group is superior to a study that does not have a comparison group. So your RCTs have comparison group, your cohort studies have comparison group, your case control studies have comparison group. These are all analytic study designs. Is that okay? They have a comparison group. We want to see what is happening in one group compared to the another when that exposure is not there. Is that clear? Right? So we have analytic designs, the opposite is descriptive study design. So descriptive study design will not have comparison group traditionally. Now cross descriptive purely can also, you can create within the group comparison also. So as our friend mentioned, that if I want to find out prevalence of a particular disease, so that fits traditionally into the purview of cross-sectional study. So that is a survey. It can also be done in a hospital setting, if suppose you have a question, but traditionally you have to go to a population, true prevalences. Okay, any study that is done in hospital comes with some bias. Yes, no? So any study which is done purely in hospital, you have to know that there are limitations of that sample. Anybody who is coming to that hospital will have some set of characteristics. Not everyone will come to hospital. Maybe if one is coming to AIIMS Delhi, maybe those are unique people who land up here. Maybe more complicated, or maybe referred, right? So you get so many cases that are not being treated elsewhere, and they are coming for referral opinions here. So you get a different kind of environment in each hospital and an institution, and that also governs what kind of patients you are treating, managing, and what is happening. Maybe you are not offering therapy for that particular disease, patient won't come to you. Only certain set of patients will only come to you for their management, right? So hospital always, any sample that is purely based on a some inherent biases. So what is a bias, by the way? What is a bias? Okay, right. So there is an error and there is a systematic error. Now what does that mean?
Not corrected randomly. There is some problem in the design, say, of a study, which causes that bias to appear.
Try this. Yes, that's perfect. Answer is right. Yes. So there is a random error. There is a systematic error. There are two errors. Random error, systematic error. Random error happens due to chance. Okay? You can't do much about it. Maybe if I have 100 people in my sample, I do another study with 100 different people, my result are going to be these 100 set were different and those 100 were different. Right? And when I measure the characteristics, it becomes different, right? So that's a random error. Largely the sample characteristics and larger the sample, the random errors are going to be less. Systematic error, on the other hand, are not random errors. They happen because of someone's fault. Okay, now it may be intentional, it may be unintentional, but these are going to affect the results. Validity and accuracy of the study is going to get affected. They will underestimate or overestimate your findings. They can happen in the design phase, they can happen during the conduct phase, they can also happen in the analysis phase. So at any phase of the study this can happen. So any research study will have design, conduct and analysis. Is that okay? So all your studies, whatever technology you are developing, you have to first think about the design. You will have to conduct that, execute the study, and then you will have to analyze the findings, right? So all the studies you need to think about design, conduct and the. Is that right? Okay. So we discussed about analytic and descriptive. In analytic we said we have cohort, we have case control and RCTs in the experimental. In the observational we will have cohort, case control, cross-sectional also, right? Now cohort studies, the defining thing is there is a group of people, cohort, they share some common characteristics. It's a longitudinal design, or you can say you will follow up them. I'm not using the slides because I don't, if I use the slides I'll take another hour. I don't want to take another hour, and that's the reason I'm not referring to the slides, okay, and I'm going like this, right? Otherwise you will have to sit and bear with me till 2 o'clock. So I don't want to do that, okay? Yes, so the cohort is, there's a group of people, and the group of people are to be followed up, and so it's a kind of follow-up study. There are three varieties: prospective, retrospective, and ambidextrous. So prospective study design is wherein you start with a group Some are exposed, naturally to an intervention. Some are not exposed, to something, whatsoever that exposure is. So that exposure is not in your hands, it is happening naturally. जैसे मैंने मेंशन किया, एयर पोल्यूशन आप ऋषिकेश में या दिल्ली में है, ये नेचुरल है. आप जहाँ रह रहे हैं वो डिसाइड कर रहा है कैसा वातावरण होगा वहाँ पे. Yes or no? Right. So it's a natural division of exposure, and that's the basic difference between RCT and cohort. Are very similar. Okay, if you see in RCT also, you will follow up people prospectively. You will make them exposed to some new device. You will observe after a period of time some outcome, but this has happened after randomizing them. In a cohort, you don't do these randomization. People are naturally, getting exposed to something, and another group is there that is not exposed to the same thing. Over a period of time, you measure their outcomes and you compare the outcomes between both of them. One exposed group, another non-exposed group, and we calculate relative risk ratios. Is that clear? Cohort. There's another design which is called as retrospective cohort. And a retrospective cohort would basically mean there is a cohort group in which both the exposure and outcome has occurred, but you are going back in time to see who was exposed and who was not exposed. And what is very important for doing a retrospective cohort study is you should have a good quality data availability. So all your registries, all your records, all your hospital records, you can do retrospective studies. If you have good, accurate data collected similarly for all the people who are attending your clinic or whatever, right? If you have exposure data, if you have outcome data, so this could be a record-based study also, right? For example, in a particular hospital, there are deliveries that are happening, and you have both the details about the outcomes, that is the babies that have been delivered, and also the antenatal records, and there was some kind of risk exposure, some non-risk exposures. You can do all that and then see, babies had some characteristics and kind of do a lot of analysis, right? So you do calculate relative risk, you do calculate risk ratio, and an ambidextrous cohort would basically mean that you have a mixed retrospective and prospective. Is that right? Okay. Now, what is the difference between cohort and a case control study? Yes?
So basically, cohort can be prospective or retrospective. The essential difference from case study groups. If we are dividing study groups based on the exposure, it's a cohort study. If we are dividing study groups based on the outcome, it is a case control study.
Yes.
Both can be retrospective, both can be prospective.
Yes. So cohort, as I mentioned, there is an element of retrospective cohort also. That design is also there. But what is important and that fundamentally differentiates between case control and cohort is your starting points. So usually in a cohort you start with exposure division, exposure, non-exposure, and then you basically measure the outcome, right? Or exposure and outcome both have happened in retrospective, okay? Whereas in a case control study, largely our starting point is with outcome, diseased, non-disease. So somebody has got a rare adverse event with your technology. Your starting point is that particular event, right? And you take another person who did not develop that event. And then you basically, see what has happened to them previously during their periods of exposure to that technology or device or whatever. Is that okay? So that is a case control study. Your starting point is selection of cases and controls, cases with a particular kind of an event that you were interested in. And that defines your case. And a control, the basic difference between case and a control is somebody who does not have that event of interest. But if being exposed, that person also has the potential to develop that event. Is that clear? That's a case control study. It's a retrospective design. You go back in time and then you find out what's the exposure in the past and you calculate what is called as odds ratio. Is that clear? So that is the case control study. Lot of biases operate in case control is not that easy. It becomes very difficult, right? And there are various ways of, finding out, from where these controls have to be selected. Is that clear? So cross-sectional, you all know, it's a kind of a survey design. You find out, if you want to find out a prevalence, you want to also find out how many people are using a particular kind of technology. Again, you can do a survey and find out that percentage and proportion. Is that right? So you can do those, surveys. It's a cross-sectional, which basically means in a particular snapshot cross-section, what is happening. Now, out of cohort, case control, and the cross-sectional, which is the superior design. Which is the superior design? Cohort. Why? Prevalent. Suppose it is, suppose it's prevalent and it's not very rare. So rare exposures, and yes, you do basically cohort study. Within cohort also there are more study designs like nested case control. Let's not go there. Yes. So cohort, one of the fundamental property or a characteristic is, as you go from exposure to disease, the causal inferences, the causal association, the temporal association, the ability to ascertain temporality of events that exposure led to outcome is best in cohort, and more than this is in RCT. Is that okay? So RCT, you are sure that before you have not, till the randomization, people are not exposed to your technology. Once they are exposed, once you have randomized, there is an exposure to technology, the outcome of interest will occur sequentially only. Right? So whenever in any study you are able to establish that exposure occurred first and then comes the outcome, this gives us temporal association, and this is one of the very important attribute to a certain causality, right? So cohort and more than that, in RCT you have the causal inference properties. Is that right? In case control this won't be there. In cross-sectional again there are some set of limitations. Okay. Now often when you do health technology assessment and when there is so much available evidence, what kind of evidence you have to look at? What is my question? So everybody is hypoglycemic now. Yeah, so this will be my last point. This is the last study design that I am talking about, which is not there in this slide. I am telling you something extra. Okay, so now you have to pay attention to the question. Okay, the question is, in health technology assessment, there is HTA methodology. Okay, often we have to find out how good that technology was, how effective that technology was. So what is the kind of evidence you will be looking at? Cost से पहले तो effectiveness की बारी आएगी ना, फिर cost के बारे में बात करेंगे. Health economics is the next step. In health technology assessment, we do exactly cost effectiveness and health, economics. We do a lot of modeling things there. We will not go there for this session, but for effectiveness. So RCT. Now bigger than RCT, what is that you are looking for? Systematic reviews and meta-analysis, right? So this is another kind of the gold standard or a bigger evidence beyond RCT. So a systematic review and meta-analysis of RCTs gives you the best available evidence. Okay, so in evidence hierarchy, and what is systematic review? From a traditional review. So what is that systematic? Okay, so one is the systematic search. So there is a way how you are going to define your search strategy. You are going to look for available evidence. So that has to be very, very explicit and transparent. Systematic. What else? Yes, you will also ascertain the quality of evidence. Okay, so these are the two very important things. And if you just do search systematically and don't do quality assessment, that does not make it systematic review. It will be a rapid scoping review. We have now so many terminologies for review, right? But systematic would mean that yes, you have to find out the quality of evidence and the risk of bias assessment has to be done. And then you see what kind of evidence is coming out from the about systematic reviews. Some systematic reviews you can do meta-analysis, and meta-analysis does basically mean that you can quantitatively find out the pooled estimates. You can pool them, quantitatively. And then you can get, whatever the pooled estimates for, say, RR or 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 whatever you want to give. Is that okay? So you have systematic reviews and meta-analysis. Then comes RCT. With RCT, I also mentioned, is a difference between RCT and quasi-experimental study? Yes, presence and absence of randomization. Okay. Then comes your observational cohort. Then your case control. Then cross-sectional. And you can also have descriptive studies. So these are the variety of study designs that you have. We also have validation studies which can be done in a cross-sectional way also, right? Which are cross-sectional, and you compare the characteristics, and you can find out, performance of tests by sensitivity, specificity, and PPV, NPV, and ROC things. Clear? We also learnt about analytic, descriptive, observational, experimental. When you have hypothesis in mind, which study you? Analytic, experimental, depending upon whether the exposure has to be by you or it's natural. ठीक है? Is that right? I think this is sufficient. Go to questions. Yes, sir. हाँ जी बोलिए सर, क्वालिटी इंप्रूवमेंट क्या है? Okay. Yeah, so that's a very, very good question. Quality improvement studies are increasingly happening, and quality improvement studies come under largely the categories of implementation research. So there's another kind of research which is implementation research. So in quality improvement, we want to improve the quality, okay, of whatever is your interest. Maybe it could be as simple as blood pressure. You want people to measure blood pressure with good quality, because there is a lot of variation. If you don't measure it good, the results may also not be accurate, right? So the quality improvement studies come under the purview of implementation research studies, and usually they don't fit into all of these traditional epidemiological study designs basket that is there available, but they are very, in an ICU you can do a quality improvement. So wherever we are operating, wherever we are working, we can do QI studies. How we can improve quality of whatever, and there is a different approach there. You started with a question, you basically form a quality team. So it's a quality. The QI study is a kind of a quality approach study. You do plan, do study act cycles, PDSA cycles. You record an indicator and you track the progress in the indicator. So it doesn't fit traditionally into this. So yes, but not in a traditional RCT way, right? And it will be very local and contextual, right? Okay. Yeah. Any other question? Yes, sir.
Can there be, there's a difference of opinion among RCT and the feasibility of RCT?
Yeah. So any RCT, before going to a full-scale RCT, you can do feasibility small-scale studies. Whether this will come, for a full RCT or not, there are certain questions that you don't have answers for. So feasibility studies are done, and any big studies you can do feasibility studies. Yeah, so feasibility studies is a smaller sample. You can have 2 groups, lesser number of patients, see how they are performing. Sometimes you don't know how to calculate sample size, so these feasibility studies give us, one is your population or the sample characteristics. You will get some figures, this can help you in designing your, larger studies. The second, feasibility studies are also very important in telling, how you have to conduct a study and where all errors can happen and how you can prevent them. So it is given as how things need to be done. You don't have best answers available before a large trial. So you do a small trial and see, how best you can do that study. And these...
In case if you don't have a value through which you can calculate the power, there you can do it.
Yes, and also feasibility of different procedures that are involved in trial. So
Always statisticians say that there is no RCT. Feasibility of RCT cannot be called as RCT because it is
So small RCTs are there. They are feasibility trials. Yeah, you can do that. Yeah, and even like within RCT, you have a lot of spectrum. You have explanatory trials. You have pragmatic trials. The setting is different. So each is a one week workshop on its own. Right. So if case control study, if you have to know everything, you have to spend 3-4 days. Then only we can cover. If it is RCT, there are many nuances and analysis in RCT. There are different kinds of RCTs. There are superiority trials, non-inferiority, step wedge design, cluster randomized trials. There's a lot of cancer trials will have different kind of things. So each one is a very different area. But I think for an overview, this is okay. Yeah. So yes, so we do systematic reviews for observational studies also, and for evidence synthesis, sometimes we don't have RCT being done. So it depends, whether the question can be answered by a question cannot be answered by an RCT. Obviously, you will have only observational studies. So for observational studies, you can pool it by systematic reviews. But if there is a possibility of doing an RCT or an experimental study for that question, then evidence coming from RCT, even a single RCT, if done right, could be considered superior to what is the evidence coming from observational study. An observational study, lot of biases, confounders, all those happen, which have to be taken care in an RCT design. And that is why it is superior to an observational study. So if you don't have an RCT available, then you have to do systematic review of observational study. And that's also a clue if it is, researcher should also venture into that area. Yes, madam. Yes. Okay. So a hybrid study again, comes under the purview of implementation research. So increasingly we are going, in implementation research studies. Implementation research, those who do not know about it, they are studies which actually help us in knowing answers how to do something better. Like you have a technology, but if that technology is not scaled, not utilized, there are barriers that exist for utilization of technology. How can we make our routine real world settings adopt this technology will be coming under the purview of implementation research. There you have one kind of design called as, the studies, some studies are referred as hybrid studies. Now hybrid study basically is telling us that you don't know best how much is the best utility of this, or there is an intervention we don't know that is effective or not. So any technology you can scale it if you are convinced that it is effective. If at first you are not sure that it is not effective, you won't like to scale it, isn't it? So hybrid is you are also ascertaining effectiveness, along with that how to get that adopted by working on some of the barriers that can hinder its adoption. So you want to ascertain the effect, along with ascertaining the effect, how to basically take it to larger people. So that makes hybrid. Sometimes there are a variety of hybrid designs also. Sometimes which question is more important? Is it the ascertaining effectiveness, or is it how to scale it? So if suppose both are important, then you have to give equal importance to them. But so there are type 1, type 2, type 3 hybrid designs. But the hybrid basically is to ascertain the effect. So you will do, randomized trials to ascertain the effect. But when you want to take it to routine settings, you will have a different approach. And when you want to do both, that is the high. Yes, there was some other hand I saw. So some aspects mean that design will be in totality there. So if it is an RCT, you will have to design a good RCT, right? Implementation research studies are done wherein you have established evidence that this particular intervention works. How to make that work in larger setting is the question that you have to answer in implementation research. In hybrid, when you are not sure that it works, you want to answer that question also. Okay, so there you will have, experimental designs along with the other things. Clear? Shall we end? Yes. Okay.
We'll take it forward in the evening, right?
Okay, thank you. It was very nice. Yes. Okay. Yeah, you want to come?
Thank you, Dr. Malhotra, for sharing such practical insights on research design and methodology. I would like to invite Dr. Suchita Markan to please the memento to Dr. Malhotra. Two, right? It's time to recharge and refresh. Lunch will be served now. Please rejoin us by 2 p.m. Thank you.
Those who came from outside,
Welcome back. We now have Dr. Kalpana Luthra, Professor and Head Department of Biochemistry, AIIMS, speaking on adopting cutting-edge techniques in research. She completed her PhD at AIIMS. The main focus of her work has been towards understanding the immune responses elicited by HIV-1, infected adults and children, generation of human anti-HIV-1 recombinant monoclonal antibodies as potential therapeutic reagents against HIV-1, and to map the neutralization determinants on the HIV to identify Indian clade C specific epitopes for immunogen design. Can I please have ma'am for the talk?
So thank you very much, Punya, for your introduction. A very good afternoon to all of you. At the outset, I would wish to thank ICMR, Dr. Suchata Markan, and Dr. Veli Pandian and the other organizers for having me here. So on to the various techniques that are used in research. I would like you to just raise your hands. I know that most of you are clinicians, all of you are faculty. How many of you are into research? That's good to know. So I did have a few slides put in, thinking that because the theme of the conference was on pharmaceutical advances and so on. So a few slides I had on what are the recent advances and how it can help in drug screening and designing and later on taking it on to clinical trials. Very brief slides. Following which I'm using this, the research that we undertook in our laboratory for the past couple of decades and how that research has evolved with the advancements in the techniques. So I would like you to be very interactive and stop me and ask questions wherever you have any doubts. So what are the recent advances? All of you are aware. There is artificial intelligence, big data analysis, computational analysis, and within artificial intelligence, basically there are a lot of machine learning tools available. There are a lot of websites available in which you have the data. Earlier, like if you wanted to identify any molecule for inhibition as a drug, right, or for therapeutic purposes or so on, you had to screen a large library of compounds. Either you begin right from the crude extract, purify. So it used to be a humongous task. You already have databases available, and you screen for the desirable properties, pharmacokinetic properties and so on. And also they are QSAR, that is the quantitative structure activity relationship softwares available. So they will even predict the PK and how you can take them forward. Likewise, you identified your molecule of interest, purified, tested it and characterized. Then you want to take it forward for clinical trials. Ultimately, if 100 molecules are taken, phase 1, phase 2, phase 3, by the time ultimately even one, if it reaches the market, that may take a decade-long period. So therefore, it is better to resort to these kind of AI technologies wherein such methodologies are applied across various stages of the drug development, discovery, lead compound optimization, right from high throughput screening, prediction of the efficacy and toxicity. Like say for example, you had some drug targeting choriocarcinoma, adenocarcinoma, right? So initially you would test it on Caco cell lines, right? So nowadays you also have databases based on studies conducted in these cell lines and what is the output of those molecules. So you can skip the in vitro studies and take a lead and go on to validation. So this comes by machine learning. So these kind of deep learning algorithms can swiftly identify lead compounds with therapeutic potential from vast libraries. Like you have more than 10 to the power 60 molecules which can act as drug targets, right? So therefore it's better to use these kind of approaches in the current days where it is available. Further, such machine learning models can predict the pharmacokinetic properties and the toxicological profiles based on the structural features of the compounds. So like say even in our lab where we use we generate monoclonal antibodies against HIV. So in order to understand which are the epitopes recognized by these antibodies, we need to do a cryo-EM analysis. So we send our complex of the protein antigen along with the antibody purified through size exclusion chromatography and then send it at Indian Institute of Science Bangalore, and there it takes some time to do a cryo-EM analysis, and then that's how we come to know the epitopes for mapping for immunogen design of a vaccine. So all that can be cut short if you already use these kind of machine learning models which will predict based on the structure that where your compound is going to interact with. And this will help in guiding optimization and selection of promising drug candidates. So this is just an overview. I'm not going to go too much in detail. As you can see, you can use these kind of AI tools for drug discovery, pharmacological product development, pharmaceutical manufacturing, quality assurance and control, and clinical trial design also, and monitoring can be done using this information. And there are a number of artificial neural networks which are available which give you information. And these are the various AI tools that are used in drug discovery. I've shared the slides, so if anybody is interested, you can just take it. So what you machine learning, which is one of the component of artificial intelligence, you have training set on which you train the particular algorithm or the software, and then you use it for the test set and see whether you're getting a similar observation. And then the best models are then taken forward, again determine the PK/PD of that particular compound. So this can be used to screen any new chemical that you are researching. And finally, hybrid machine learning output, wherein continuously it learns from whatever data comes out of the various chemicals being screened and any new compound that you want to test. Of course, ultimately you have to do validation of these are all just to cut out the time for the screening identification of those compounds. Just like, for example, in the omics technology. So in research, all of you are aware that these days, like if you earlier, what used to happen is like if you want to do identify whether a particular gene is expressed, mutation present, so then you will try to clone that particular gene, express it, and then characterize it. Nowadays, what you do is you do a next-gen sequencing, RNA sequence, so you know, okay, this gene is expressed or not to begin with, whether there are any miRNA targeting this gene. So all that information you will get from the omic analysis, because you have not only next-gen sequencing for the RNA expression, but you also have metabolome, microbiome, and so on. You also have Jiva studies, which will entirely sequence the entire genome complement of the cell. So once you do this, identify your protein of interest or the DNA protein interaction or the RNA which is expressed from a particular gene. How do you validate it? So if it's a protein, then you have to do a Western blot analysis, right? So if it's an RNA which is being expressed, so whether you do not know whether it's going to go on to expression or not. So that again needs to be tested in. And whether, yes, this particular mutation is affecting the phenotype or not, how will you validate? By silencing. So if you silence a particular gene, if it is still continuing to alter the phenotype of a cell or an organ or an animal model, then you know, okay, it is a functional mutation. So these kind of validation experiments in the wet lab are mandatory, but yes, kind of tools will help you in getting large amount of data from which you can select out based on the existing literature. So my preceding speakers, I'm sure, would have emphasized the importance of how you arrive at a very specific research question. So you extensively review literature, identify what is a lacunae, and then come on to the research question. And if you are spending enough time in posing the correct question and identifying what are the suitable methods to address that question, half your work is done, right? So therefore, it is not that, like often we find that not only ICMR scientists would agree with me in most of the funding agencies, now that you have these genomic tools, many of the investigators, they mention in their proposal, okay, we are going to do the genomics, we are also going to do the transcriptomics and ultimately the metabolomics. But what is the question are you asking? Whether you need to do all those omics. So that is what is important. We have a lot of tools these days, but you need to know why you want to use it. So that you must keep in mind and then only go ahead. So basically, using these omics technologies, it helps in discovery of molecular markers for diseases and biomarker identification, right? So biomarkers are nothing but any molecule which is present not only in disease, the individuals. So when you're looking for a biomarker, what you're looking at is alteration temporally, right? Whether it is being altered in disease, whether it has any prognostic potential, or whether it is increased or decreased significantly compared to the normal levels, and therefore it can be used as a diagnostic or a prognostic biomarker. So even for biomarker identification, if it is at the protein level, you'll do a proteomic analysis. If it is at the gene expression level, you can do an RNA-seq, or if you want to quantify certain mutations or gene alteration, gene duplications and so on, then you can do a GWAS study. So now I will come to the topic of research in our laboratory, which we have been doing for more than a decade. All of you are aware that HIV is an enveloped RNA virus with 2 non-complementary strands of the RNA, and every individual, be it an adult or an infant, infected with it, is able to generate antibodies against the virus, and majority of these antibodies are targeted against the virus enveloped glycoprotein. So these are the envelope spikes which you can see here, and most of the known broadly neutralizing antibodies are identified against this. So you may be wondering, what is the purpose of generating antibodies, and so why is this research question being addressed in our lab? More than 50% of the global HIV infections belong to clade C viruses, and in South Africa, both these regions constitute more than 90% of the clade C infections. And also we had an Indo-South Africa project in which we looked at what are the types of antibodies and viruses, whether they are different in the Indian and South African clade C infected individuals. Again, they are very different, the viruses circulating. Although they are clade C, within them also there are differences. Therefore, the need of HIV antibodies from Indian clade C infected individuals, and primarily we are focused on pediatric HIV-1 infection. So these kind of antibodies, when they are generated, what are their potential uses? So you can see here this abbreviation BNAbs. This is nothing but broadly neutralizing antibodies. So what do we understand by this? Nowadays, a number of antibodies are used for therapy. I'm sure each one of you can list a few, 1 or 2. At least can I have something from the audience? You must be aware of antibody which are used in clinics. Ibalizumab, and then for rituximab, right? for multiple myeloma or non-Hodgkin's lymphoma. For a number of autoimmune disorders, you have a number of antibodies used in the clinics. So antibody-based targeted therapies are very, very specific, and they are preferred over the other anti-cancer chemotherapeutic reagents to avoid the side effects, right? So potential use of these, so why broadly neutralizing antibodies? These neutralizing antibodies not only prevent infection against a particular strain of the virus, but are effective against a large number of strains, and therefore they are broadly neutralizing. So now this is a whole IgG1, entire antibody comprising of 2 heavy chains which are identical, 2 light chains, right, linked by the hinge region. So you can use these antibodies to design vaccine. So anybody quickly, if you can tell me how an antibody can be used. Any one of you. Any method by which you have the antibody, like now during SARS-CoV-2, right? There were a number of antibodies generated in many of the individuals. If they are purified, many of those antibodies have been published, right, as potential. And so based on that, if you want to design COVID-19 vaccine, so how would you use that information from an antibody and go back to vaccine design? Any thought on this? Yes? So direct antibodies can be used for passive therapy, right?
Yes, for various antigens that are available, whether the antibody binds.
Yes. So as he rightly said, antibody, is elicited against a particular antigen, right? It will bind and recognize a particular antigen. So if you recognize what are the epitopes or the amino acid residues of that antigenic protein, then that antigenic protein can be used to design a vaccine. So it can either be an epitope focused vaccine, or it can be an entire envelope based vaccine, like in the case of SARS-CoV-2, you know that covaxin was basically the entire envelope of the SARS-CoV-2, right? It was inactivated. Or you focus it like the mRNA vaccine against a particular spike region or so on and so forth. So antibody can be used for vaccine design. It can be used for pre-exposure prophylaxis, as it is used in exposed individuals at risk to develop HIV. Yesterday only it was in the papers that, yesterday or today it was in the Times of India, that many of the children receiving blood transfusion for thalassemia, 9 of them have developed HIV infection due to blood transfusion. So screening is very important, and this pre-exposure prophylaxis is also a very important component. So again, mother to child prevention. So there are many women in the 3rd trimester, if they are found to be diagnosed to be HIV positive, it can be prevented by a single dose of giving Zidovudin, right? So that is an antibodies can be used as an HIV vaccine alternative, as you rightly said. They can directly be used for therapy. Then HIV combined antiretroviral therapy alternative. Now, in most of the West, HIV is not that much of an issue. Why? Because antiretroviral therapeutic reagents are available, and if they are taken as per the regimen, they can, to a great extent, delay disease progression. However, none of the antiretroviral therapeutic drugs can prevent HIV infection, right? Why? Because whatever virus has already in flea in our peripheral blood T lymphocytes or in macrophages, those cannot be targeted by antibodies, because antibodies are in the circulation and they cannot see a virus which is residing inside the cells, because HIV is an intracellular pathogen, right? So they are rendered ineffective. However, if you use these antibodies in combination with ART, then together they are helpful in significantly reducing viremia if they are given immediately on detection, and they can viral latency. So that is the advantage, and as HIV functional cure. So this is basically a reverse vaccinology strategy which was proposed by Dennis Burton right in 2002. So reverse vaccinology means what? Individuals who are infected with a particular disease, be it like say in the case of COVID-19, SARS-CoV-2 infected individuals, or in the case of HIV, you screen HIV infected individuals, adults or children, for the presence of plasma antibodies. If those antibodies are effective in blocking the viruses that you test in your assays, then such antibodies can be used to map the epitope specificities and design immunogens or to generate broadly neutralizing antibodies and test them for their protection. Now, there are a number of these available for monoclonal antibody generation. So, I'm sure all of you would be aware of the most ancient technology for antibody production. Hybridoma technology, right? So, there what you do is you just simply fuse a myeloma cell with the B lymphocyte of which is specific for a particular antigen, and then those hybridomas are then cloned and they are clonally diluted, and you get single hybridoma cells which will secrete only antibody of one epitope specificity, and that's why it's known as monoclonal antibody. So that was the most simplest way or traditional way that was available. However, that technology is very cumbersome because as you go on to select the hybridoma, at every step you have loss of cells. So at the end of which, when you start with 10 to the power 6 clones of hybridoma which are fused, when you come down to generating the single cell, you characterize it by clonal dilution, you land up with 1 or 2 antibodies of 10 to the power 6, and it takes about 6 months to 1 year even if it is done in a high throughput manner. So now there are nowadays very recent advanced technologies for antibody generation. One is a combinatorial display libraries. So you may have heard about phage display library. So a whole and 2 heavy light chains combined together, whereas you can also have SCFV libraries, that is a single chain variable fragments. So ultimately in an antibody which is Y-shaped, so in that antibody it is only the complementarity determining regions which are recognizing a epitope, right? So what you do is by simple PCR you amplify the variable region of the heavy chain and light chain, right? And then clone it in a phagemate vector and infect the phages. So any plasmid will infect the phage, and you have a large library of the phage clones, say 10 to the power 9 diversity. And then what you do, that those phages which are secreted in the supernatant of the library, those you screen using an antigen, whether which of these phages is specific for that antigen, and then you do the cloning and antibody expression. The other recent technique is development of humanized mice. So for any purpose, you can generate humanized mice by recombinant technology. So here in this case, if we want to screen whether antigen is able to infect a mouse, right? So then what you do, you replace or knock out the mouse antibody producing genes, heavy light chains of the mouse, and then you knock in human antibody coding genes for the heavy and light chain. So at the end of it, this humanized mice is expressing human antibody B cell receptors, correct? And then when you immunize with HIV, for example, so you have a lot of antibody forming B cells, that is a plasma cells, which you will fuse with the myeloma cells and you get a hybridoma. So earlier, all of you are aware that mouse monoclonal antibodies used to be used for therapy, right? So why do we need to go through this cumbersome procedure of, say, hybridoma, human hybridoma technology, or say having humanized mice to screen the antibodies? Why not use mouse monoclonals for therapy? Say again. Precisely. So either the body will recognize it and as foreign and immediately clear it, or it can have immunogenic reactions in the body, as you very rightly pointed out. And therefore, it's preferable these days to have human antibodies, and if not feasible human, then you do humanized antibody. So humanized antibody is that in the mouse antibody, whatever antibody you raise in the mouse, out of that entire region, only that portion, the complementarity determining regioning of the mouse antibody is retained. Rest is replaced by simple PCR with all the human antibody encoding genes. So therefore you humanize it, right? So these are the various technologies. Now apart from that, the most recent technology is single B cell cloning. So what you do is that from the human peripheral blood, you just take 10 ml blood and you take the, by FACS sorting, flow cytometry based sorting, you take the memory B cells of the humans who were infected with a particular disease, like say with SARS-CoV-2 or with HIV, right? And then you either take the plasma blasts, and ultimately what you do you do an antigen-specific B cell sorting, see which of these memory B cells are against HIV, memory B cells are against SARS-CoV-2, and then based on those cells, you can use the antibody to be generated. So apart from this antibody generation, you need to, once you generate an antibody, you need to test whether it is able to protect, right, against that particular disease. So if you use live HIV virus in a laboratory, that will not be very biosafe, right, unless you have a BSL-3 laboratory, and all experiments cannot be done in that because it's a very time-consuming process. And therefore, you have these pseudoviruses which have the envelope of the virus, but they do not have the infectivity component. So they are very biosafe. They can be handled in a BSL-2 laboratory, right? So I'm not going into the detail. So by generating these pseudoviruses and a luminescence-based, luciferase-based reporter assay, we are able to see that if whatever antibody we generated, if that is very effective in preventing the pseudovirus from infecting the cell, that means what? The luminescence will not get activated, and therefore you get less readout when you read it. Whereas if the antibody is not protective, this virus will infect the cell and the cell will give out luminescence. So this is a simple luciferase based reporter assay which can be done in a laboratory. So way back in 2014, one of my PhD students, Rais Andrabi, presently he is an associate professor at University of Pennsylvania and continuing with HIV vaccine research. So he generated by the traditional hybridoma technology in 2012, patented this neutralizing antibody which was granted patent both in India and South Africa, and this antibody was directed against the V3 region of the envelope. I'm not going into those details, it's all there in the publications. Then I talked to you about the phage library. So this is a very simple technology wherein, unlike the hybridoma, where you get one antibody at the end of 1 year, here you will get 10 to the power 9 clones. Right, so you have a large repertoire of antibody clones secreted by phage library. You can see here, this is the phage, bacteriophage, which has the code protein 3 on the surface, which is a minor code protein. So even if you replace it with your antibody coding gene, the phage will continue to replicate. It is not affected. So this is how you have your SCFEs expressed on the surface of the phage, and you have a large library which you can screen by biopanning, whether it is specific to a particular antigen or not, and then you can express that particular SCFE. So this PhD student Rajesh, presently faculty at IIT Roorkee, so he developed this novel strategy of developing SCFEs by phage display library. So currently you must be aware of CAR T cell therapy, all of you. So that is basically what does CAR mean? It is a chimeric antigen receptor. So expressed on the T cells. So every T lymphocyte has a T lymphocyte receptor. So what you do is, by PCR, you express a gene coding for your SCFE of interest. So if it is against, say, placental antigen, so you will have anti-placental antigen antibody, only the SCFE part cloned into the T cell receptor. So what will happen? That will target that T cell against the placental surface antigen and not against any other organ in the body. So SCFEs have a lot of applications these days in CAR T cell generation. So FDA approved ibalizumab for treatment of those patients who failed both first and second line therapy, and since then the antibody generation took impetus. So this was an antibody which was directed against the CD4 receptor on the T lymphocytes. So again, one of my students screened the HIV pediatric patient plasma for the presence of SCFEs against CD4 binding site, and identified 2 of these SCFEs which had protective activity. So this was my student Sanjeev who published this work, and ultimately it is seen that such SCFEs not only can also be used for producing bispecific antibodies. So what do we understand by bispecific antibodies? See, especially there are viruses like HIV or SARS-CoV-2 which have spikes, envelope spikes on their surface, which are at a distance. So if 2 spikes are at a distance, you have a bivalent antibody against one site, so the other arm of the antibody is loose. So the antibody will fall off and not be effective in blocking that virus from preventing another cell. But if you have bispecific antibody, means one arm of the antibody is against one antigen and the other arm is against the other. So they are recognizing two, like you can see here, this is one antibody of one specificity. The light chains are light pink, right? Now you have another blue chained antibody against another epitope. So you have antibody again by PCR based experiments. So such bispecific antibodies demonstrated synergistic and also enhanced in vivo protective activity in HIV infected human eyes. This was shown in Rockefeller University, and there is a large application of such bispecific antibodies for therapy. So now with the advent of single B cell sorting, and if you have a good purified antigen, you can also do that antigen specific sorting. So from all the plasma, B lymphocytes, other mononuclear cells in the blood, you can identify only those B cells which are active against HIV. So using this single B cell cloning, we generated this particular antibody, AIIMS-PO1, which was very effective in neutralizing 76% of the pseudoviruses. And this antibody again has been filed a provisional patent number, and now presently it is being filed for the full complete specification. Now, very recent development, I told you, the omics technology. So what we did was, this donor 330, from whom we isolated this antibody AIIMS-PO1, we had say 2 million cells of this donor. It was a pediatric HIV infected donor. So we took 2 million of this PBMCs of this particular donor and did B cell receptor sequencing, bulk B cell receptor sequencing. So what we got thousands of B cell receptor sequences from this particular donor, and we identified, that whenever an antibody is generated in the body, due to repeated infection or exposure to the pathogen, the antibody undergoes affinity maturation and somatic hypermutations. So by doing bulk B cell receptor sequencing, we identified 21 lineages of that wild, found to have much higher specificity and better neutralizing activity than the wild type AIIMS-PO1. So this is how the large data can help you in getting more high affinity antibodies within a short period of time. If you do it by hybridoma or any other technology, it will take years to develop. So then what we did was, this particular high affinity lineage antibody mapping at IISc Bangalore, our collaborator, like in the morning, our director was also mentioning, no research can be done in silo by one investigator or one institution. So everything happens by collaboration. So this was our collaborator from IISc, Dr. Somnath Datta, who did the cryo-EM analysis, and we were able to identify certain epitopes at 4.4 angstrom resolution. So the major focus in our laboratory is how the virus and the host antibody responses are co-evolving. So in order to understand the pathogenicity and how it can be targeted. So here we followed up these two twins. They were genetically identical twins. We did HLA typing in our HLA laboratory with Dr. Uma Kanga, and we found that they were genetically identical twins. But however, you see in both, if you see the left panel and the right panel, just by the color code you can see one of the twins is developing darker, more antibody responses as compared to the other twin. So we went on to see that why they all, despite being genetically identical, did they elicit different antibody responses. So we went on to further characterize the various viruses circulating in these individuals, and to summarize it, we found that both these twins elicited antibodies against both V1 and V3 region of the envelope, suggesting that presence of viruses in these children is associated with the persistence of early neutralizing activity with time, which improves, right? Then there was a report from Julie Warbo from the United Nations showing one infant who developed a neutralizing antibody within 1 year. Normally it takes at least 3 years, even in adults, to develop protective antibodies. So we screened 49 HIV infected infants less than 2 years of age who were not then initiated on ART in collaboration with our pediatricians. And then we found that multivariant infections in these children led to very good protective antibodies. And then we went on to characterize the viruses in these infants and found the proteins for exploring polyvalent vaccination, because these children who had only multivariant infection were able to elicit good protective antibodies. So, as I told you, reverse vaccinology is a very good strategy for combating any particular infection to design vaccines. So, this is a model of neutralizing antibody development in humans. So in all humans, during health and in disease, initially whenever a single virus for the first time infects, right, that is known as a transmitter. That will bind to the naive B cell receptor, which all of us inherit. Once that B cell is activated, it will undergo a number of clonal proliferation, activation, and elicit protective antibodies. So in vaccine also, in a say lineage vaccine, what you do is you initially vaccinate with the simplest form of that immunogen and then add more complexity to it to mimic this particular natural action of antibodies in the hosts. So by way of this, we have developed in our lab from the 330 donor, as I told you, pediatric donor, we developed a trimer which was native-like. We engineered it to act as a good HIV vaccine and also tested it in preclinical models, that is the rabbits. And currently, this is now filed for complete specification. So during the pandemic, what we did, we had anti-HIV1 antibodies, that is also an RNA virus. SARS-CoV-2 is also an RNA virus. So we tested one of the various known broadly neutralizing antibodies. Are they cross-reacting with SARS-CoV-2 infections? Why we tested this? Because you can have a pan-RNA virus antibody or a vaccine which will prevent against a large number of RNA viruses. Cut this short, we tested these antibodies of HIV against the pseudoviruses of the SARS-CoV-2 that we generated in our laboratory. That was done by my PhD student Nitesh during the COVID pandemic. And we found that there is a cross-neutralization of the SARS-CoV-2 but by one of the HIV-specific broadly neutralizing antibody and also the polyclonal plasma of these SARS-CoV-2 patients. So all this work was possible by a large number of multiple collaborators and various funding agencies who have helped this work happen. So I think at the end of this, the take-home message what I would like to give you is, although you may begin to answer your research question using a very simple technology, constant review of literature going back to what is the most recent available and then important. Otherwise, our work will never reach that stage which will recognize recognition. So wish you all the very best, and if you have any questions, you can please ask. Any questions? I know during postprandial time, so difficult. I hope at least.
One thing is very interesting is, when she filed the first patent, when she came with the neutralizing antibody, the concept was so novel. Unfortunately, we have other side. Those days, we didn't have something like a contact between us and the company, that kind of thing. So it's academically we developed so much, but when it comes to transfer, we had some issues. Her case was one among that. In fact, the first one we filed through TIFAC or something.
Through DBT and TIFAC.
Yes.
And now DBT has also stopped.
Yes. So now I think with the help of mitra, I think we'll be able to take it further. That's a great advantage.
Yes.
But I must appreciate that she was one of the pioneers.
I think there's a long way to go. This is a way forward. Thank you.
Good afternoon, everyone. I just want to inform that we have a patent mitra booth outside. So today morning we have heard about Patent Mitra initiative from Dr. Suchita. So the participants who are interested in filing patents and understanding the portal, so you guide and register there and have a practically look at the initiative and how to register, so that when you go back you have ease in doing registering the portal and using it. Thank you.
Thank you, Dr. Luthra, for that fascinating session on how technology continues to redefine medical research in the 21st century. I would like to invite Dr. Vishnu Vardhana Rao to please present the memento to Dr. Luthra. Up next, we have Dr. Geetha Menon, Scientist F, ICMR, HTAIN, DH, who will speak on methodology and interpretation of data and systematic review and metadata analysis. She is a trained biostatistician currently serving as Scientist F at the Health Technology Assessment India, Department of Health Research, DHR. She also holds the position of professor at the Academy of Scientific and Innovative Research under the Faculty of Medical Research at the Indian Council of Medical Research. A gold medalist in her master's program from Delhi University, Dr. Menon completed her PhD at the All India Institute of Medical Sciences with specialization in her meta-analysis. Her core areas of expertise include the analysis of data from large-scale surveys and epidemiological studies, statistical modeling, and conducting systematic reviews and meta-analysis. Can I please invite Dr. Menon for the talk?
Good afternoon. So I was just looking at the agenda today, and I found that this talk that Madam gave, that was sandwiched between two simpler topics. One on research methodology and the other one is again on systematic reviews and meta-analysis. The topic that I got was a huge big one, and I couldn't make the meaning of it because then I thought let it be simple, and I'll talk about systematic review and meta-analysis. Of the group here, many of you have done some research work. How many of you have actually done a systematic review? Good. At least there are a few. So this topic is mainly on an overview of how to do a systematic review and why systematic reviews are considered to be the highest quality of evidence. So we all know that when we are talking about healthcare decision making, it is a multi-dimensional approach, and there are a lot of challenges. The challenges could come from the medical fraternity, where they are more interested to look at the clinical outcomes and the patient safety. It could be challenges coming from the technology itself, because the question is how the technology is scalable, whether it is reliable, scalable, usable or not in terms of devices, diagnostics, and then the next dimension could be the social dimension. How many of the people, what social sector will be actually using that technology, and how this decision making will be useful to them? Is it acceptable by them or not in terms of culturally being acceptable or not, affordable or not? Then the ethical dimension looks at the fairness, the prioritization of the decision, and then the economic aspects, where the decision is cost effective, does it provide you the value of money or not, and whether it is harmful to the environment or not. So it is a multi-dimensional approach, and with conflicting objectives. The objectives could be on one side to improve the outcomes, on the other side you have to have a reduction in the cost of the decision. Similarly, whether there is an increase in access, at the same time you should have a high quality standard of the decision. Similarly, it will promote innovations on one side. Some of you are innovators, so does it promote innovation? At the same time, is it affordable or not for the patients and the community? So, decision making has multiple stakeholders. It could be the patients and caregivers who are only interested at looking at the significance of the decision in terms of their safety and their health issues. The clinicians, the policy makers who are more interested to look at the public health impact. The payers and insurers, again looking at cost, sustainability and affordability of the decision, and the innovators and industries who are helping in promoting the innovation. Ultimately, it's a multi-dimensional challenge, and it cannot be answered just by based on one or two studies. So we need to rely on a body of evidence that comes from a way of taking stock of existing evidence. So how do we take stock of existing evidence? We'll see that. And that ensures reliability. It ensures a balanced approach in terms of benefits and harms, and also it is more evidence based. So this is done through evidence synthesis. So what is evidence synthesis? Evidence synthesis is a process of bringing together information from multiple sources and combining the results in a manner that is useful for decision making. And it helps in identifying the gaps. It establishes an evidence based for best practice guidelines, and in the end it informs the policy makers, the decision makers, the practitioners, the clinical practitioners, and it helps us in producing clinical practice guidelines. It helps us in producing policy briefs for the for the policy makers, and it also helps in producing systematic reviews for researchers. So we have all seen this many times when we do our research, that this is an evidence pyramid where the at the base is the background or the expert opinion, which are only very few in nature, and they are anecdotal evidence, single studies or from case reports or so, and then gradually but the quantity of information of these types is very large. So if you look at the base, you have a lot of information, background information coming as expert opinions, as case series and case reports. But gradually as you go up the pyramid, you see that the number of studies coming out are very few, which are of good quality evidence. So as you go up the pyramid, the quality of evidence increases, whereas as you at the base of the pyramid are those studies which have lot of biases, and we cannot take those studies as evidence informed studies. Then systematic review is one method of evidence synthesis. There are many other methods like umbrella reviews and are also there, but systematic review is one method which is commonly adopted by the researchers. And what is systematic review? It is a method of using explicit and unbiased methods to identify studies, select those studies, synthesize the information from these studies, and summarize these results in a meaningful manner to inform the policy makers and the decision makers. So systematic reviews and meta-analysis are used as similar terms, but we must remember that systematic review is a larger group of review studies, and meta-analysis only talks about the quantitative pooling of the studies from these from this review. If you do not have quantitative information from the primary studies, you can still do a systematic review and publish your results. So systematic review, there are 8 broad steps in a systematic review. One is to formulate the review question. Second is to establish a team, which is very important. Develop and register a protocol. Fourth is to conduct the research. Select the studies, the primary studies that goes into the review. Next is to extract these primary studies, synthesize this information, and then disseminate them as a scientific paper or as a report. So in the morning session, you would have already learned how to formulate a research question, so I will not go into the details of that. Let's go to step 2 here. The step 2 says that you should establish a team. So whenever you are planning to do a systematic review, remember that you should have a domain expert in your team, expert in the team, in addition to the project manager who is actually conducting the review process. So the subject matter expert, if not one, maybe more, and a statistician who is a methodological expert and who will help you in doing the quantitative synthesis of primary studies should be part of your team. The second is, the third step of a systematic review is to develop a research protocol. Like any research study where we start with writing a protocol which actually outlines the methodology, here also in a systematic review we should start by writing a protocol which includes the rationale for the review, what are the key research questions, and can you break them into PICO components, then the inclusion-exclusion criteria, the literature searches that will be done in the unpublished and the published arena, and then the data management procedures, the data extraction, and the methodological assessment of methodological quality of the primary studies, including the risk of bias of the individual studies, the data synthesis methods, and grading the evidence. Once you have done all that, how to grade the evidence from this study needs to be also written up in the protocol itself. And remember that a systematic review protocol, as far as possible, there is a mandate by the Cochrane review that if you are planning review, please write up the protocol and register the review in a good portal. And I'll show you what are the different websites where one can register the systematic review protocol. So the first and the commonest place where you can register your protocol is the Prospero. So most of the times Prospero registration is considered to be mandatory, because just like clinical trial CTRI number, Prospero gives you an ID number, and this number needs to be quoted when you publish your systematic review protocol, and as well as the systematic review itself. So Prospero is free, it is available, and it helps you in registering the protocol. Recently there has been an update in the Prospero, and it makes your protocol very easy for publication purposes. If you are able to follow the steps of it is a ready paper for you to publish. So you can use this Prospero as a website for registering. Then not only that, it is not only not mandatory, although it is not mandation, but many of the funding agencies also mandate that the research review protocol needs to be registered. Then some of the journals like PLOS and BMC also strongly endorse Prospero, and this is very useful for funding organizations, for guideline developers, for journal editors, and for peer reviewers. This is another website where you can register your protocol, and that is INPLACI, which is available from 2020 onwards, but it charges a small amount for submitting your protocol, and every update it adds a cost to it, and it accepts systematic reviews and scoping reviews. So this can also be used as a registry registry for protocols. Then the other one is the OSF, which can also support your registration. It is free and it is available, and it is accepting all study designs, and all of this provide you a DOI also. So if you are interested to publish, it will give you the DOI value, DOI number, which can be used later for your publication purposes. Then the last one is the protocols.another website where you can submit your protocols, and it accepts all study designs, and it is free also. It provides a DOI also. So the next step is to conduct the research. So once you have decided the top of the review and you have written up the protocol, the next step is to conduct the search. The aim of conducting a search is to generate a comprehensive list of primary studies, which includes both published and unpublished, that helps you in answering your research question. And as thorough your search is, as valid would be your review. If you are including all the right search terms in a review, then the validity of the review increases, and therefore you need to develop a very comprehensive search strategy that includes all synonyms, all terms that are used by different authors who published their primary studies, and that can be included as terms in the search strategy when you do the search on the website. So what are the different ways of systematically searching primary studies? One is using the electronic sources, which includes all the databases, the MBase, the PubMed for medical research, the electronic libraries, and the internet. Sources could be, you can use journals, you can use textbooks, or it can be done by hand searching also. Sometimes there is a need for us to also look for grey literature, because not always that published information is available to answer your question. In such cases, you will have to go to databases which publish grey literature, conference proceedings, and sometimes funding agencies also have their own repository of studies that they have funded, which can be used to look at the grey literature. So when you're searching electronic databases, then it depends on the research question. If the research question is related to healthcare interventions, then Medline and MBase are the most common databases. Then Cochrane can be used if you're searching for randomized controlled trials. For psychology and psychiatric studies is a database. For nursing and allied health professions, Sinahal is a very common database which helps you in providing you the primary studies. Similarly, you have the research question that you have in mind. And for internet searches, today in today's world, we have AI-driven search engines like Semantic Scholar, then the medRxiv and the archives are available, Google Scholar is there, and these all have search mechanisms by which you can search this database to identify the primary studies of importance. Then you can look at dissertation abstracts from this website, http://library.dialog.com, where dissertations are available, which could probably help you in identifying the primary studies. Or there are some thesis websites also which can also be searched. For other sources, we have to look at the primary studies first and go to the reference list and then do a research again. And you can do hand searching using the journal's content, conference proceedings and abstracts. You can look at commentaries and letters from databases which are not indexed in databases, and CTRI and clinicaltrials.gov are definitely there to identify primary studies from the on randomized controlled trials. Literature, Google Scholar is there, OpenGray, SSR is a common website where they do not, the paper wouldn't have been peer reviewed, but it goes into SSR for anybody to see. And then ProQuest and Shodh Ganga for theses from Indian universities. So once you have searched these databases, you have to first, to search these databases, you have to construct a search strategy. So what are the steps for constructing a search strategy? First of all, once your review question has been framed, then you will have to break down your review question in the PICO format. Do you know what is PICO format? Patient intervention, comparator, outcome. So the PICO format has to be there in the review question itself. If a review question does not have a PICO format, that means it is not a well-framed. So with the PICO format in place, then you will have to identify synonyms or common terms for each of these components, the P, the I, the C and the O. So there could be multiple terms or synonyms for the same thing used by different authors when they publish their papers. So you will have to do a pilot search first and try to get as many synonyms as possible for each of these components, P, I, C and O. Then independently you will have to run a search for the P first, then the I first, the O first, C first and the O. Together, when you put all of them together, the terms that you use for each of these components are combined by the Boolean operator called OR. What is OR? I'll tell you in the next slide. And when all the P, the I, C and the O has been searched, then you do a single search using the Boolean operator AND. So that is how the stepwise search procedure is carried out. And how do we look at how do we get these keywords and synonyms? From title and abstracts from published studies. So we'll have to do a pilot study first, pilot search first, and identify all keywords for each of these synonyms, and then you can use various wildcards methods, like if you want to look at women, so you can have WOM #N. So hash includes either A or E. So this way you can use wildcards and truncated procedures to search for the keywords, and sometimes you will have to use index terms also, the mesh and the mtree, which is usually used for searching the databases. And then this is one example where the author has searched for data coming from data on physiotherapy and management of adult complex regional pain syndrome, which is a health condition, and for this health condition he is trying to do a systematic review. So you just look at the number of words and synonyms that different authors have used for the intervention, for the comparator. So we'll have to look for all these words. If you need a comprehensive search to include all existing information about a particular topic, then we'll have to include all these synonyms in your search strategy. And then the Boolean operators are these. The AND operator tries to only get results that contain the words that you combine with AND. So here is an example. Suppose you are doing a search on older adults and look at influenza vaccine and pneumonia. So if these three words, if you want all these three primary studies which include older adults also, those include influenza vaccine also, and with pneumonia, then you will have to use the OR word as the Boolean algebra operator. And when you are combining these words with AND, you see then only the middle portion, that is the only number of studies that you get. When you use OR, you get an exhaustive, comprehensive list of primary studies. Got it? So you will have a lesser number of studies when you combine it with Boolean algebra and Boolean operator and, but when you are OR, you get a huge number of studies. So that you will have to see. So the OR operator is used for combining the synonyms of individual components of the PICO, whereas the AND operator is used to combine P and I and C and O. That is how you do it in a stepwise manner. So please remember that whenever you are doing a systematic review, you have to document whatever you have done. Document your search strategy, document the databases that you have followed, all the literature sources that you have used, any individuals or organizations you have contacted, any journals or conference proceedings or abstracts or whatever sources you have used, please document all that, so that if at all you are not able to complete, or if you are done only a review on current number of studies and somebody wants to update that review, they don't have to start from scratch. They can use this information and do it. So the next step is to select these studies. How do we select the studies? Suppose that you have done a lot number of studies. So primarily when we do a research, when you do a search in systematic review, you don't draw the full texts. You only look at the title and the abstract. So as I said before, team of systematic reviewers, you should have more than 2 subject experts. The third expert will be there only to adjudicate the resolve the disagreement between the first 2 reviewers. So the first 2 reviewers, the title and the abstract in the first stage, and they will say whether this article or the title goes into the review or it doesn't go to the review, or is there a maybe not very clear, the information is not enough for them to decide whether we should include that paper in the review or not. So there are three categories of decisions that could be taken, and accordingly the title and the abstract screening is done. And wherever there is a maybe, or people who are sitting in the studies which are sitting on the fence, then the third reviewer decides whether it should be included or not. Once you have done that and some of the topics go out of your list, from the primary list, then you pull out the full text for each of these studies, and then the full text is again reviewed in a similar manner, and it will be included or not included based on whether it meets the inclusion criteria or not. And then you will have to develop this PRISMA 2020 flow diagram. Very important that for any systematic review you have to follow PRISMA 2020 flow diagram, which has 3 domains: the identification, the screening, and the included domain. So the graph very clearly gives you all the boxes, and it's very self-explanatory. Anybody who wants to do a systematic review should follow this, and you can fill up the boxes that is given here. Then the next step is that once you have included these studies, it is not that you include all of them. There could be studies coming from multiple regions, from various geographies, on a different population or patient characteristics, having some specific subgroups of populations also. So there could be heterogeneity in the population groups, there could be heterogeneity in the methodological approaches that these primary studies have adopted. It could be the clinical definitions that they have followed. So there could be differences in all these when we do a review, and it is necessary that we look at these studies very carefully and critically appraise the quality of the studies that go into the review. And questions like, are the findings reliable? What do the results mean in the context of the decision that we are making? Able to replicate the same findings in our kind of settings. All those questions are important, and whenever we read an article, we'll have to ask these questions and check whether these primary studies are able to address these questions or not. And that is what happens in a critical appraisal. When we are doing a Cochrane kind of review, then they talk in terms of risk of bias. What are the different biases that a study could have? The selection bias, the performance bias, the detection bias, the attrition bias, and the reporting bias. To talk about it a little more, I think we need time, but for the sake of this, I'll just mention that these are some of the biases that a study could have, and one has to address these. Look at them, look at the primary studies and see whether these studies, do they address these biases, do they talk methodology or not. If they do mention about it in their methodology, then we say that the study has low risk of bias. But at the same time, it doesn't talk about this in their methodology. We are not very sure whether that addresses that primary study addresses that bias or not. So we categorize them as studies with high risk of bias. So this is what a Cochrane RCT review would look like. We don't have to go into details of how to develop a quality assessment tool, because quality assessment tools are already available. The Cochrane risk of bias tool is the most standard format for randomized control trial. And then the critical appraisal skills program, the CASP checklist, has about 8 checklists for different study designs, like the systematic reviews, the case control studies, the cohort studies, all these types. The Newcastle, Ottawa is mainly used for case control studies and cohort studies for reviewing their quality. And the JBI critical appraisal tool has 13 checklists for all study designs. Not necessarily that you do a systematic review of only RCTs, you can have studies also. So, but then again, you'll have to look at the quality of those studies, and these tools help you in doing that. So once you have appraised the study, and the next step is to extract data from these studies. And what data do you extract? You extract the data on the study characteristics, the study design, whether it is an RCT or where the study was done, what settings was it done, was it in a hospital or was it in a community, what was the sample size of these studies. the population characteristics, what was the age of the patients, what was the gender, what was the ethnicity, what was the health condition that was being studied, baseline characteristics of this population, weight, height, comorbidities and all that. And the intervention and the comparator details. What was the intervention, what was the dosage, what was the frequency of this intervention, and for what duration the intervention was given. Same way, the comparator has to be also described, and any co-intervention that will also come in the data extraction. And the clinical outcome measure, the secondary outcome, any other clinical outcome of interest, and whether it is patient reported or self reported, is it clinician reported, all that needs to be extracted, and the units of measurement. In addition to all this, you also have to extract from the primary studies the numerical data, statistical outcomes, which are ultimately going into the meta-analysis. The means, the standard deviations, the proportions, the odds ratio, risk ratio and all that. So that will also come, the sample sizes, the events in the binary group, the number of events for the binary outcomes, and any subgroup analysis that goes into the primary study. And once we have done that, we should remember that when we are doing a primary study where we collect participants, we are actually drawing the raw data from them, and then we are trying to get an overall effect estimate, say the mean or the standard deviation. But when we are doing a meta-analysis, we do not have the raw data. We have only the published data coming from the published articles. So we need to quantify the effect for each study. So the effect size is what measures the magnitude and direction of the any treatment's outcome from a primary study. So combining 10 studies, then you will have 10 effect sizes and their corresponding variances also. So remember that when we do statistics, we should have the magnitude and the direction and also the uncertainty attached with it. That's how statistics is usually done. The central tendency and the discrepancy. So we all know this, but if it is a binary effect measure, then the effect measures usually defined are the odds ratios, the risk, right? So odds ratios and risk ratios are drawn for each of these primary studies, and they are written up at one place along with their standard deviations or variances. And how do we define the standard deviations and the variances? I have another slide for that. And if it is a continuous effect measure, then the commonest measure that we use is the standardized mean difference, and it is also called the Cohen's D. Cohen's ratio of the difference of the means in the numerator and the pooled standard deviation in the denominator. This is a statistical formula for that. You need a statistician in the group so that it explains to you all this. Otherwise it will be difficult. You do the clinical part and he will do the statistical part. So all that the statistician will explain to you how to calculate, and depending on the different types of scenarios, you can have a pre-post scenario, and if you want to do a review on that, again it is possible to do that. These are the formulas for that. And if you are looking at a response ratio of some physical measurements, then the ratio formula is also given, and we can pool them all along with the standard variation, standard error and variance. For odds ratio, the standard error and variance is defined like this, and for the risk ratio also it is given here. So once you so you have now the all studies in place, all the data has been extracted. How do you extract the data? You have to create a data extraction form. So you create a data extraction form and then put all the data in the form of Excel sheet into that data extraction form. It is ready. Now you hand it over to the statistician. He will do the analysis. So there are two types of analysis in systematic reviews. One is the fixed effect model and the other is the random effect model. Fixed effect model assumes that all the studies, all the patients, all the measurements that are done from the primary studies, they are identical in nature. But is it identical in real life scenario? No, because studies are coming from multiple countries, from different settings, from different geographies, right? So assuming a fixed effect model is not very realistic. So the other types used for analysis purposes is the random effect model. So random effect model says these studies come from different places, so definitely they are going to be different, right? And we are again trying to combine them and see how much of difference is there. We will quantify the difference between them in terms of variance of the effect estimate, and then that is how we use the random effect model. So meta-analysis is actually as I said, it is a statistical way of quantifying or synthesizing the data from the individual studies. It is very simple. You just do a pooled effect. What is a pooled effect? It is a weighted average of the estimates. Suppose you have 10 studies and you have odds ratios from these 10 studies, then it helps you in combining these odds ratios using a weight. And what is a weight? Weight is actual studies, because how precise each study is what is used as weight. And in statistics, precision is defined as the inverse of the variance, right? So we are actually weighing each of these individual studies by their weights, and that is what is done in meta-analysis, as simple as that. So if you are using odds ratio, use the same formula. If you are using risk ratio, but you will have to define the variance in a manner which is appropriate for that particular effect. So if you are looking at standardized mean difference, again the same formula, but the values will be different. The difference between fixed effect and random effect is that the fixed effect estimate uses the weight as the inverse of the variance of each of these studies. So every study has a variance, an effect estimate and a variance. So take the inverse of that and then you pool them all, weigh them with their variances. But when you are looking at random effect model, then you are not only using the weights of the individual studies, the inverse of the variance of the individual studies, but you are also adding one more component, that is a variability between studies. So that is the difference between random effect model and the do it hands on. I think you will be able to understand it better. This is a forest plot. This is a forest plot that we commonly use as a graphical way of depicting a systematic review. And if you look at the two analysis, the graphs, what difference do you see? What is that diamond in the middle? That is a summary effect estimate, right? That is a summary effect estimate, and we are all looking for that diamond only in systematic review. Diamond. And if you see the diamond here, which diamond is bigger or broader? The random effect. So what happens is, in a fixed effect, the diamonds are precise because we are assuming that these are the only studies in hand, we don't have any other studies. But in reality it does not happen. So random effect model covers a larger population of studies, and therefore the fixed effect has a compared to the random effect, which has larger uncertainty. But in reality, random effect model is the one which you should use. So these are the differences of fixed effect and random effect. It assumes that the effect size is the same in all studies, whereas random effect assumes that they are different, and the weights given for fixed effect is just the inverse of variance of individual study, whereas the weights given in random effect model is not only the weights, also includes the weights of the between study variance. Okay, and when we do a fixed effect analysis, what happens is larger studies get more age. So if you are pooling a small study with a large study, the entire effect measure is actually borrowed from the larger study, which is not in reality true. Whereas in fixed effect, in random effects are more balanced. So that is the difference between the two. Which model to use? Generally the random effect model is the one which is most appropriate, which captures heterogeneity. But if there are very few studies and you know that these studies have been done in a similar manner using the same protocol, like you have task force projects of ICMR where every site is doing the same, following the same protocol. If that is so, then fixed effect is a better alternative. One more thing that we need to look at is the looking for heterogeneity. So when we look for heterogeneity, what is heterogeneity? Heterogeneity again means that there is variation between the studies. We can't do anything about it because we are handling published studies, right? Data from published studies. What we can do is that we can look for heterogeneity and see whether we are able to identify reasons for heterogeneity and then report them. That is our job. As a reviewer, we can't do anything about it because we have the limitation of only looking at published studies. We don't have the raw data with us. So there is heterogeneity between the studies and we report the systematic review, how much of heterogeneity is there. So if you see the graph at the back, there is something called I-square there. In both the graphs we have I-square. So I-square actually quantifies the proportion of variation between the studies. Larger the I-square, more heterogeneous the studies are. Then can we combine apples and oranges? No, we cannot quantitatively combine data from studies which are highly heterogeneous. So we'll have to look for heterogeneity, and somebody has tried to categorize the heterogeneity and when to combine quantitative. So if the heterogeneity is 0 to 40%, it may not be very important and you can combine them. If it is 30 to 60%, then they say it is moderate heterogeneity. But if it is 50 to 90%, that is substantial heterogeneity. And even if you combine and give a single statistical measure, it may not actually be the right or most appropriate summary measure. So, and when there is considerable heterogeneity, definitely you should not go for a meta-analysis. You just do a systematic review and publish that. Okay, do a descriptive review. And there could be many reasons for heterogeneity. We've already seen it could be the difference in studies, it could be the individual characteristics, it could be the various follow-up procedures that the studies have followed, or it could be various reasons. Another important thing is to look for publication bias. When we do a review, we are assuming that all the studies that we have gathered for the review has been accounted for. Okay, but actually there are reasons for some studies to not getting published. It could be because you have got a negative finding and therefore the researcher decides not to publish it, or it could be that the funding agency deciding, so we will not publish it. So there are n number of reasons why some of the research that you do doesn't actually go for publication. Many times, even when I was in ICMR, the researchers or the principal investigators, they do a study and they submit a report and they don't publish. So it's actually whatever research that they have done, it goes unnoticed in the scientific group. So we should publish of doing a publication, looking at the publication bias, and that is called the funnel plot, where you plot the effect sizes on the x-axis and the inverse of the precision of these effect sizes on the y-axis, and then if all the studies that you have in hand follows an inverted funnel like shape, then we say that it has no publication bias. But in reality you will never get an inverted funnel when you do an actual systematic review. But one, because you don't have enough studies. You should have at least 25 to 30 studies to get a shape like this. You may not be able to get, and if the points are spread all across the funnel, then definitely there is heterogeneity and publication bias, and we need to report that. And the last step is to report the review. How do we report the review? There are guidelines for reporting, and if I click on this, I'll leave these slides with you. You can click on this and there is a website where you can, that gives you the guidelines on how to report. Achha, there is no web, this one, no? Wi-Fi. So there is a way of reporting, and you can go through this website to actually see how to report your review. And there are, if you are only trying to publish your systematic review, there are templates for that, and this link actually gives you the template in which you can publish your systematic review. These are the sections of a systematic review. Like any article, you will have title, abstract, introduction, methods, results, discussion, all that you require for a scientific paper, and you can publish your systematic review. Thank you.
Any question for us? some people have done it. Great.
Any particular software you would suggest for conducting a systematic review?
Not conducting a systematic review. Ryan is there for screening. For every step there is a software. There is no software that does a complete systematic review for you. There is one software which is AI based that is called nested knowledge. Nested knowledge. That supports screening. That supports screening. That searches for you. That does a PubMed search for you, and it will list all the articles also. And if you are alone, actually you should not do it alone. But if you are alone, there is an AI person with you, will be there with you as a reviewer. So you are one reviewer and the AI is another reviewer, and together you two review the articles and include it in a research question. And then it helps you in data extraction. If you are able to master nested knowledge, I think your half of your work will be done. Then Cochrane RevMan is already there, but that is only for RCTs. But if you are planning to do an observational study, again, the RevMan will not work. Then the best software that I know of is comprehensive meta-analysis. If somebody can purchase it, CMA. It's called CMA. It's a very good software.
Most of these softwares are very costly.
They are costly, but if you can get some funding.
Otherwise, you keep a statistician with you. At least they would pay us.
So we have a Center for Guidelines in Department of Health Research. So there are experts in that. You can always approach them if you need. Don't do everything yourself. Leave some for the statisticians.
Thank you, Dr. Menon, for such a clear explanation of data interpretation in research. I would like to invite Dr. Vail Pandian to please present the memento to Dr. Geetha. Our next session is on electronic data keeping and management by Dr. Vivek Gupta, Additional Professor, Community Ophthalmology, AIIMS. Dr. Vivek Gupta is a graduate of University College of Medical Sciences, Delhi, and MD from AIIMS, New Delhi in 2005. His work spans AI-based glaucoma screening pathways, integrating glaucoma and diabetic retinopathy screening pathways, epidemiological glaucoma research, national blindness and visual impairment surveys in India, verbal autopsy-based cause of death, ascertainment systems, SAND implementation, research for prevention of blindness and visual impairment. He has over 100 PUB MED and 40 plus non-indexed publications. Can I please invite Sir for the talk?
Thank you. Thank you for this opportunity. First of all, how many, I am assuming everyone here would have done data entry at one point of time or another, right? You have all been doing research and that is why you are here. So let us look at some of these basics about data management and data governance issues, which become important when you want to actually share your results, when you want to instill confidence in others that the work that you did was genuine and completely authentic. So how do you put faith in naysayers, so to say, about the data that you have collected being genuine? So that is what, in a very, very broad shell, data management is about. So if you look at the NIH data management sharing policy, what they say is data management is the process of validating, organizing, protecting, maintaining, and processing scientific data to ensure accessibility, reliability, and quality of data for its users. Now, its users are not just you, right? You are the researcher, you are the primary generator of the data, but the users are also the regulatory agencies to whom you will be ultimately applying. The users are also the reviewers with whom your manuscript will be shared, and they will be consuming the results of your data. And these days, many of the journals actually want you to put up your data in an open source repository where users can download them. So in a essence, everyone who can access your data becomes a user of your data. So you're trying to ensure accessibility, reliability and quality of your research data for everyone. That is what data management is all about. There are a few guides when it comes to the domain of data governance, but like different agencies, ICH, European, MHRA, UK, even ICMR, they have different definitions. But overall, if you see data governance, which is kind of one of the driving forces of data management, is about roles, policies and life cycle control. So it is not that much about the software that you are using. Okay, it is not that much about the data that you have collected, but it is about the policies you have adopted, the life cycle, the everyday things, steps that you have adopted in your research, which are ensuring the data is of high integrity. Every change is traceable when what change was made. The data has been always kept securely, and it has been handled in a lawful and an ethical manner, right from its point of creation to the point of its archival and destruction. Now, good data management is integral if you look at Indian guidelines, the drug and clinical trial rules of 2019 of Government of India. They are also integral if you look at the GCP ICH guidelines, the good clinical practice guidelines. And as early as 6 January 2025, a special section on data governance was actually adopted and made a formal part of the GCP guidelines. It was in a draft till last year, but this year it was made a formal part of the document. And there are multiple, like largely 2 points which talk about, which largely deal with data management, and these days data is all kept electronically, so electronic data capture. So one is the data life cycle elements section of the GCP guidelines, and second is the computerized systems of the GCP guidelines. So I mean, so what are the data life cycle elements? So they start from data capture, right? So what are the controls? Controls means the SOPs or the policies that you have put up, which are ensuring that the data which is going into your system is actually correct, is actually completely verified. So these are the data the first step after you have actually collected or maybe filled up a paper form, and now you are putting it into an electronic system. So are you doing 100% rechecking of all of your data which was put up in the paper and is now being entered? Or are you doing a sample check? Are you doing a dual data entry? So what is the policy that you adopted as part of your study for ensuring correct transcription level errors? Ensuring that not just the data, but the metadata. Metadata means the subject ID, who entered the data, from which device, from computer, from which IP address, at what time, what place the data was entered. So all of this metadata is also captured along with the data as part of the process. And finally, systems, do your electronic systems have any automated inbuilt consistency checks, which we try to adopt as much as possible. But say, for example, if you are using something like Microsoft Excel for doing data entry, very, very difficult to implement. But if you are using designated data capture systems, then you can build in range checks, minimum, maximum checks, things like those. Then a lot of this issue about electronic data collection governance is about audit trailing. So who did what when? So right from, so if you're working in a team, right, very often you are not a solo researcher, but you would have maybe a data entry operator, scientist with you, you would have fellows. So in your data collection system, who are the users who are actually authorized to enter the data? Who are the users who are actually authorized to make changes? Who are the users who are authorized to lock a record or to delete a record? When did a permission that was given to a user changed? So someone, for example, some team member, so does your system capture when their rights in the system were revoked, that now they no longer have the ability to change or edit or make any sort of accessing to your data? So all of those system logs are important from GCP perspective. Change transparencies, audit trailing we have already talked about those. Then sometimes there are data queries which are raised. So this would be especially true if there are multi-centric studies and one center is in charge of your data management and other centers are basically supplying the data, and you have a query. You are asking them, okay, you have entered this value here, is it correct, or why is it this way? So this communication, how is that communication being logged, how is it being recorded? A query is actually considered closed, the query that you have raised with a member partner. Then like the system should not be disabling audit trails. Some of these systems have audit trail capability, but it is disabled, right? You have to explicitly enable it. So whether you have enabled it or not enabled it, all of these things. Timestamps. So you see that in systems, times has been captured, but captured in the UTC coordinates, which is the Greenwich GMT time, right? So, and then you are wondering, okay, I did this work at 9 a.m. Why is it showing the data entry being done at 4:30 a.m. in the morning, right? So, knowing what is the timestamp coordinate system which is being used in your data, the moment you export it, you will know, it will say UTC, or you will realize that there is a 5.5 hour time difference which is there. Then also there are things about reviewing your data. And it's not enough that your system is actually capturing the data. But as an investigator, one is you're doing your data analysis, right? You're doing your interim data analysis, final data analysis, periodic reports, but also user accesses, correctness of user access, team members who have left, have their roles been revoked or not in your data collection system. So those kind of procedures, it's just a one line that you have to write, that we will every, say, month or every 2 months check the correctness of user access permissions in our data collection system, and you have to do it. Tick in the box for you when it comes to compliance with these guidelines. Data corrections I've already talked about as part of audit trail. Another very important is, especially when it comes to multi-centric studies, if you are sharing data, how do you ensure that what was sent by you or what was sent to you is actually what was received by you, right? So one of the ways to go about it would be very simply, you create, first of all is like create a password protected zip file. Okay, create a password protected zip file, and some of these password zipping tools, they also allow you to actually create a SHA-256 kind of a code. Okay, which is a code associated with that file. Okay, and the recipient can actually check, okay, the file that I have received, the received file's code, what was the generated, the code at the generation level. So the authenticity of the file can be verified. So there are tools available for these. Also, like if it is a password protected zip file that you have said, and the file is corrupted, there is some corruption which has happened during data transfer, the file, it will tell you the file is corrupted. Okay.
Can I interrupt you?
Sir.
See, you worked a lot with data entry and doing about complete trial. Can I take one example and explain them? That would be easy for everybody to understand that how it goes on.
Okay. Sir.
It's a big mega study.
Okay.
Just when you explain, that would be more useful for everybody.
Okay. Sir, so I'll tell you the example of the vaccine study that we did. That was a study where we were visiting around 17,000 individuals every week, and we did that for 5 years. And we immunized nearly like 3,000 children with influenza, trivalent influenza vaccine or IPV. So in that study, you can imagine that was being generated, right? 20,000 records every week of surveillance. And among all of them, how many all were developing acute respiratory infections, how many are immunized, their follow-ups, multiple doses. And our data management was being done at UAB, Alabama, Birmingham, right? So that time we did not have REDCap. I was going to come to REDCap to show some of these things. So in that trial, what we were essentially doing was, all of our data was being at that time collected on OMR sheets, scannable OMR sheets. All those data were being scanned over here. And how do we know which form has been scanned? You can take an analogy of how do you know which form data entry has been done. We were simply putting a stamp on that form. Scanned. You can put a stamp, entered. And you know you will not enter that form again in your data collection system. Subsequent to that, like the data that we were sending, the OMR system would work on it, would create a CSV file, and then there was an automated script that was done to analyze that CSV file. And it would say, okay, the age has been read out for this child today as 5 years, right? But last week in the system, the age was 6 years. Why is that? Now that could have been an OMR issue, right? In our case, in your case, it could be a data entry issue. What was 5 earlier, pen was slightly longer, right? It is read at 6 or it is read as 5. So then the UAB team would send us a query that this is the respondent ID, age is listed as this, what is the correct age, right? And so this was the query system that we had with the main central team, and we respond, okay, within a span of say 3 days after checking internally that this is the correct age, and then we would get an acknowledgement that yes, this query is closed. So you have a system, a structured kind of a system of ensuring correctness of data as well as responding and closing of all the queries. And some of these things, I will quickly cover these and I'll show you how to do some of these things in a very easy manner in the presentation. I have some screenshots for that. I've already told you about this issue of data exchange and corruption and ensuring confidentiality is there, right? So this was the same system of password protected zip files that we had used, so that even if someone intercepts a file, they will not be able to access the data in it, because the password was not shared in the email, password was shared through a personal message over SMS or WhatsApp. You also have an important thing which is about data finalization for your analysis. Okay, so if you have say entered your data in Excel and now you're analyzing, you do not want that if you repeat the analysis after say a week or after 10 days, you are working with a different data set and you are now getting different results. I'll tell you how this is important. When we extract our data, for example, from Open Data Kit, one of the systems that we use, sometimes we have to do these corrections, right? So the corrections, just like the age example which I told you. Okay. Now, I have run my analysis today with one particular data set. Okay, is this my final data set? So whatever you extracted from your software, whatever you extracted from your Open Data Kit or Redcat may not be the final version that you use for your actually reporting. Okay, so always make it a practice to create a folder called the analytic data set, what we call, right? 1 sheet, 2 sheet, 3 sheets, whatever number of sheets you have. So this is your analytic data set for this date that you used for all of your analysis going forward. Okay, so that will ensure that whether you run your analysis today, after a month, or after 6 months, if your starting point is the same, you will generate the same results, right? So from research reproducibility point of view, that becomes very important. Another thing which becomes important is obviously following the same analytic steps, but that's a analysis question, not a data management issue, right? Finally, there is also this issue of retention and access. So how we were following in our system was, we had for our paper forms, we had the logged cabinets, we had a log, we had a team, right? So this set of forms have been submitted by this person. Put it in a register in a row, get it signed, done. Once the scanning has been done, again, put it in a register, done. So everything is very, very structured. So you don't, okay, if a problem has happened, who is the person with whom you can discuss and identify where the problem has taken place. Okay. And finally, it's also about destruction, data management. So how do you do it and what is the period? So that's 3 years or 5 years. Typically, we keep it at 5 years. Okay, some basic steps, right? how many of you have only used Excel for data entry? No, only. I'm saying only used Excel. Huh? You have only used Excel, right? Most of, have you, okay, so have you run in with problems with dates, for example? Yes? Very classic thing, right? I mean, you put it, is 01062 for June or 6th of January, right? And the other issues which I have seen often residents coming to us for help when they come out with data analysis is mixed, right? So like weight is written as 20, then 20 kgs, then 20 point something kgs. So consistency, no? And you must remember, like when you are analyzing your data, the consistency in the way data has been entered is critical. Otherwise, you will spend a hell of a lot of time in cleaning up your data sheet. Software tools such as RedCap, which I'll show you, they will help you avoid many of these mistakes. Okay, from a pure analysis point of view, you basically have numeric data, you have text data which can be categorized, dates where formats which I've just discussed become important. You would also be having photo, video, DICOM images, your equipment has generated some waveform data, etc. So these high dimensional data is typically saved just like that in a folder with the patient ID as the folder name or something like that, whichever way you organize. But the key features from that particular data set, you would want to make it as a part of your data entry system, right? For example, ECG various QT interval, what was the QT, etc., right? So based on your high level data that you are, high dimensional data that you have collected, the key features would be part of your data frame of your sheet, whereas the main data will be part of as it is in your, as raw data in your folder structure. You also have, like for example in surveys we have GPS coordinates, for example, that we use for mapping all of our households, our villages where teams have gone. So one thing is how do you manage your data? So how many of you have done follow-up studies? Can I have a raise of hands? You have done. Many of you would have done, right? So you have a baseline visit, you would have a 2-week, a 3-week, a 4-week, you know, something like that, multiple visits. So how do you create columns for, how do you store that data? I can tell you when I started off, I used to store all of that data by putting, okay, day 0, what was the height, weight, symptoms, what was the visual acuity. At 2 weeks after surgery, what is the visual acuity, right eye, left eye. So I'll keep on adding columns for how many visits are happening, right? So that's a classic wide form data storage. That is, every one of us start doing that, okay? And suppose, for example, if it's a trial that you are doing, it's a case control study that you are doing, then some of the residents come to us with 2 sheets, okay? One sheet with cases, the other sheet with controls, right? A better way is you have one sheet, have a column, whether it's a case or a control, okay? But if you're doing a variable number of follow-ups, okay, a better way is basically a long form data collection, okay, in which what you try to do is you try to split your data set into what is the data time and what is the data that is a repeated data that you are collecting. Okay, so you now don't have one sheet, now you have 2 sheets. The first sheet is your baseline data. The second sheet would have one column which is your patient ID. Second column would be your date or whatever is your visit identifier. This is visit 0, visit 2, visit 4. And then you have just one column for your visual acuity, right eye, left eye, IOP, right eye. What is the advantage now? You can have any number of visits that you want associated with each patient, okay? Say for example, what would happen if a patient came to you for an interim follow-up? If you had restricted your number of columns, you would possibly not be able to accommodate that. You would only have 0 week, 2 week, 3 months, right? But in the long form data, you can capture variable number. So if you're expecting that, if you're expecting, you know, ad hoc visits, if you're expecting unannounced visits by the patient and you want to capture that data, then a long form data capture is easier. It's like simpler in form of data organization. It brings in a complexity when you're analyzing your data. But trust me, in any of these statistical analysis tools, it's a very, very easy thing to just do a reshape of the wide to long, at a single command. So it's handled very, very easily. Okay. Now, coming to the meat of it, data entry software, right? So how many have used EpiInfo over here? Raise of hands, no one? Very, very old kind of a software. Epidata, anyone? No, no Epidata. Redcap? Few hands, good. Sorry? Open Data Kit, anyone? We have used Open Data Kit. You have used, ma'am. Excellent. So we have used it in our national trachoma survey with 6 lakh individuals data captured in UDK platform. Open Clinica, anyone? No. So, but these are relatively like rare and common. Open Clinical Trial Master, etc. These are like not free software, but the ones on the left, EpiInfo, Epidata, these are the two cloud. Majority are moving to RedCap, EpiCollect, ODK, these platforms. So let me share you some things about the RedCap platform that will make your life easy. Okay, it's a free software. It's not an open source software, but the Vanderbilt University gives a license for free to institutions and organizations to deploy their own copy of RedCap on their own infrastructure. So I know we have done it. I believe ICMR also has. The universities in India have their own installation of RedCap. It is not open source, but it is free to use. It is capable of compliance to just about any standard, be it HIPAA, be it FISMA standard, CFR Part 11, etc. But it all depends upon how you figure it for your project and for as a server. So there are 2 levels of configuration. Server level we have done. Project level, as project PIs, you have to ensure that it is configured in a secure manner, logging and all is enabled. I'll show you some of these things. It is free for use for Institute Ethics Committee approved projects. Okay, ignore the first URL, this site is down, but the second site, the main RedCap server where you actually log in, is the one which is working. AIIMS faculty can request an account for themselves, obviously, for their staff and their residents, and also for their partners. So if you are a PI and you have, say, partners in AIIMS Kalyani or PGI or CHIPMR, you can request their accounts also be created on the RedCap server. Okay, but the only thing that we say is the accounts are always a named account. We will not create an account such as SR Medicine JIPMR, right? We'll create Dr. So-and-so, senior resident JIPMR. Why? Because as I showed you, as part of GCP guidelines, traceability, like who user did what. So the accounts are always not generic post-based accounts. You all have used Google Form. So it also has an online form designer, not a click and drop, but it's a very much more comprehensive designer. Comes with all your branching logic, right? If gender is this and age is this, then open obstetric history, otherwise skip. All those logics you can build in. Calculated fields, obviously you can do all sorts of calculations. Multiple users per project, right? So, so you can have data entry operators, you can have data managers, you can have data cleaners, you can have data exporters, you can have the PI. Any number of roles you can create these things in my project. It has audit trails, again, which I showed you as part of the GCP compliance you need to do. It also has online surveys in which you just send a link to your participants and they can fill their survey without logging in. It has built-in reports and some descriptive statistics as well. So you start off by creating a RedCap project. It does not create a project immediately. It gives a prompt to us administrators that yes, this PI has requested creation of a project. We get a mail with the IEC letter and we simply say yes, okay, done. So once the project has been created, RedCap, this is the project management dashboard that you have. And it is showing you steps 1 of 7 completed. So it guides you. And at every step of the way, there is a help which is available. Okay, you can just click on this link and it will take you to the official RedCap tutorials where you can learn if you're confused how to do certain things. So my surveys, these are the online surveys. longitudinal data collection with defined, where you say that my patient must have these 4 or 5 follow-ups, okay, when you have defined your time points. So you can use longitudinal data collection, you can enable it. MyCap, which is a mobile Android application for collection of data. So you can imagine collect your data on an Android phone or an iOS. The next step is then designing your data collection instruments, which is like the Google Form Designer, where there is an online design instrument library, okay, wherein there are certain, for example, PHQ, very commonly used in psychiatry surveys for depression screening, right? So there is already a PHQ instrument as part of the REDCap library, which you can directly import, okay, making your life much easier. So, and you can have multiple instruments. As you can see, we have so many instruments as part of this particular project, okay. A REDCap will by itself every patient a serial number 1, 2, 3, 4, 5, 6, 7, 8, and against that serial number, all that patient's data for all the instruments will be entered. And there is no limit to the number of fields that you can have in an instrument. You can get a PDF view of your instrument and give, hard copies. So you can take a blank printout for, backup in case you are not able to do data entry. You can take a filled form printout as well, okay? And obviously you can rename. There are certain features within REDCap wherein you can embed. For example, you have your first form wherein you have entered the UHID of the patient. Now in every form you want to show that UHID. So this is what I've done, right? So in every form, this is not a data entry form, but I'm reflecting data from the baseline demographic into this form, DAS21 form, wherein I have the patient name, UHID, diagnosis, etc. Just so the person who is entering the data will not commit a mistake. Okay, they will know that, okay, I'm entering this patient's data in this form. This is a snapshot of the add new field, and you have various types of fields, including images. So there are also, for example, dentition. There is a whole 32 teeth image which will be visible, do a marking. There are certain validation rules which are available. So variable name is the column name that you see once you are exporting the data. You can say validation as just like Google Forms, whether it's a numeric, whether it's how many decimal places are there, mandatory, no or yes, and identifier. Now this is important. Why? Because we all talk about PHI, protected health information and protected personal So if you mark any field as identifier field, yes. When you are exporting the data, it will ask you, do you want to export data with identifiers or without identifiers? And if you give a person the right to only export data without identifiers, you are now sure that your PII is never going to be leaked as part of your data exports. So you are again ticking a box as part of your ICMS. Okay. Then you obviously have calculated fields. Sir, then you already have access to a data. No? Then you are not doing a data capture. Then you are just doing a data analysis. The whole point of REDCap is electronic data capture. And then you also have auto numbering, and this is repeating instruments. I was talking to you about long form data collection, right? So this is what happens if I say clinical assessment and I make it repeated instrument tick mark, yes. Now I can fill any number of clinical assessments for this patient whenever that patient comes to me. It will be a separate sheet. Oh, sorry, what happened? Sorry. Now this is another thing which is you can set a custom record label. So I told you every patient is given a sequential number 1, 2, 3, 4. But you would want to see your patient as the patient name this, UHID that, right? That makes easier for you to know which patient's record you want to open. So here I've said, okay, patient name. So now this is how I will see each record, each patient list in my master list. I can click on that patient name and view all the records for that particular patient. I can do a secondary unique field. So I will ensure that UHID is always going to be unique. I will never be creating a duplicate record for a UHID in my data space. Then this is a field comment log or data queries, right? If you enable this, you can now put notes against each and every field and say, okay, these are the queries which were raised and were resolved. Okay. Then user rights and permissions. So this is the part where you have all these different things that you can do in one project, and you can say my user will be able to give other user rights, yes or no. Will be able to create a record, yes or no. Rename, delete, lock, exports, etc., etc., right? And this also has a concept of data access groups, wherein you can say that, okay, site A will only be able to access site A's data, not site B's data. So there is a multi-tenancy kind of a thing which is happening. As a PI, you have access to all sites, has access to their site's data, and within their site, the team has access to only that part of that site data for which roles have been given by the site PI. So these are certain minimum roles that we suggest which should be given. And then you will get a dashboard, for example, wherein you will say that, okay, this user has all of these privileges and these are. After you have done all of this, you move your step, you test your data entry, and then you move it to production. So that is the entire 7-step workflow of REDCap. This is like you can click a new record. This is, for example, I am getting, okay, for this record number 13, I have all of these forms, but none of these have been entered as of now. But as I keep entering, these colors will change. And if there are more than one forms, then we will have these overlapping circles after them. And this is how this appears once I showed you, right? Instead of 1, 2, 3, you get the patient identifiers and you get a very nice dashboard. You will immediately know for which patient, which record is missing. I was talking to you about audit trail. What were the changes that were made are automatically a part of the system. Not only that, you also have logs of who changed the project, when and what was done. You have various exports, and you have de-identification options within exports, and you can export to SPSS, data, R, Excel, obviously it's there. So basically, do a data management plan for your study, right? You have to say that, okay, these are the forms which will be done. These are the users who will collect the data. These are the people who will enter the data. This is how data will be entered at this frequency. We will do a single or a dual data entry, or we will do a 10% recheck. This is how corrections will be done in the paper form. This is how corrections will be done in the electronic system. This is how queries will be logged. These are the users who will be able to export the data, and as PI or as the statistician, someone will sign off that this is the final logged data that we have. That will be used for analysis. Do a training system, okay, do a brief orientation, and do not allow anyone to log in and just play around with the system till their training is complete. Keep a training matrix that all of these users who are accessing your data systems have been trained. Okay. Backups. Now backups are very important because now you need to say that, okay, as server administrators, we are taking backups. Okay, but accidents may happen. So as a good practice, always, maybe every day or every week, take a project backup or a data backup and download and keep it on your password protected computer. First validate and only go live after you have all approvals. Have a backup plan. Okay, so one backup plan is about taking backups, the other is how to capture data. Some of our colleagues actually directly capture data in REDCap, but if in one day the REDCap server is down for maintenance, what will you do in that case? So always keep a backup plan ready so that after the fact data entry can be done. You can always call us at redcap our numbers are there on the right on the home page, Dr. Kaushik Sinadev and my numbers, and you can email to us at any point of time if you have anything about the system that is a problem. And no shared accounts. Ensure that every user have their unique account. Otherwise, one person will be saying this, I did not do this. The other person will say, I did not do this, and you'll be saying who did it. Like you'll never be able to get to the bottom of it. Check audit trails occasionally so that you know, okay, not too many changes are happening. If too many changes are happening, then maybe the data entry process itself flawed, right? You need to maybe ask someone to be a bit more vigilant in doing their data entry. Do not email screenshots, etc. Do ensure that you do not leak the personally identifiable information in any of your data sharing. Yeah, I mean, these are things which we have already discussed. One thing about data corrections, for example, we often do it as part of our stata do files as well. So I think with that I will close. You know, data management is critical. These tools can make your life a whole lot easier. So you're welcome to get trained in the CRU trainings that are done on RedCap, create your accounts, play around, you know, and yeah. Thank you.
Any quick question? We are very lucky to have Dr. Vivek with us. Who is an expert of, he takes care of all the electronic data keeping. It's a very tough subject, and one more important thing I must tell you. You know, one quarter of ICMR we heard so far. ICMR is having another arm. Actually, they are giving interface for validation, right? For your validation we are talking. You can be, you might get a little grant also for that. You can be a collaborator for that. I think that tomorrow we are going to discuss more in detail. For all those things, what is he talking about? What is he talking about is going to make a sense. About you are going to use all data in such a type of format. Now you have to pull up your socks. Now you are entering into the next level. There is no chance for errors. Or you know what is he talking about? Audit trail. What is this? CFR Part 11 compliance. My God, it documents every minute information. If you just open it and you change one, you delete it and put something else in the login. You have done this on this time. So there is no point of error in that case, and he explains so beautifully about it. If you want, call him one day to your institution, ask him to talk, so that you know you get more familiarity with this kind of thing. It's very unique.
Sure. Thank you.
Yeah. Sure.
Sure, you can email me at vgupta@aims.edu anytime. Okay, vgupta@aims.edu. Right, sir? Yes, ma'am, please. Right. Ma'am, all of these Google survey form will only tell you capturing data. All these other things are part of RedCap, right? Your teams, users, permissions, exports, audit trailing, all of these. Regarding your first question about importing older data, so there is a process to do it. Okay, first of all, you create a form which matches your Excel sheet. Okay, so you have the same variable, same data format, and then you can do an import. You do an export, and then you put your data in that form, and then you import it, right? Yeah. Any other questions? Thank you, sir. Thank you.
Thank you, Dr. Gupta, for your informative talk on how digital tools can enhance accuracy and efficiency in research data management. I would like to invite Dr. Suchita to present Gupta. We'll now take a short tea break. Please return by 4:30 final session of the day. Also, we have set up a special ICMR pavilion where a team is available to assist you with all your IP queries. You can also avail patent mitra services for patent filing and technology transfer at the pavilion. Thank you. We now come to the final academic session of the day, Research Powered with Statistics and Integrity in Research by Dr. Vishnu Vardhana Rao, ICMR Chair for Medical Statistics and former Director, ICMR, National Institute of Medical Statistics. Dr. Rao has a rich experience in planning, executing and analyzing data for numerous large-scale community and clinical research study in the domains of nutrition and health. He has previously served as the head of the Artificial Intelligence Cell at ICMR and was the administrator for the Clinical Trial Registry of India, CTRI. He has about 150 publications in national and international journals and 20 reports to his credit. Can I please invite Sir?
So, this is the one. So, thank you very much. In spite of the last class, many of you are there. So, two factors are going against me. One is the class is statistics class. Second is it is the last class. In favor of me, since it's the last class, I can annex that time. Okay. So this is what I just want to give you. I don't go into formulas and all, but I definitely I watch you that I will not allow you to slip into the sleep. But so I'll just give like a story how you have to start your research. I think you know him. Of course. So simple as possible, but not simpler. That's what I'm going to do now. So, statistics is the engine that drives research from ideas to evidence. It ensures that findings are not based on assumptions or chance, but on quantifiable, verifiable analysis. That is, you just underline that. So, old wisdom, what old wisdom says, the road to wisdom is, well, it is plain and error, and error again, but less and less and less, and come to some meaningful error-free data. Fine. So this is what I am going to tell. Why I am here, standing before you, and you are listening to me. There are many clinicians, many biotechnologists, many faculties, but I am basically a data scientist and mathematician and statistician. I am talking and you are listening. Why? Because of these things. Patients vary, physicians vary, nurses vary, hospitals vary, measurements vary, disease states vary, immune response varies, drug adherence varies. There are no two individuals in this universe are same. To capture that variation, the science of statistics has got evolved. If there is no variation, everything is homogeneous, the first people who lose the jobs are statisticians because they are not required. So there is a variation. To capture that variation, the science of statistics is required. And also, there is a misconception, skepticism about statistics, even now you people have. There are three kinds of lies, damn lies. Third one I don't want to tell from my mouth, you only read it. But you misuse the statistics, this will become damn lie. Statistics itself is not damn lie. So, therefore, this Benjamin Disraeli told that these are the things, and if at all misuse the statistics, it will become a damn lie. So, for example, now statistics have become a magic word to give us semblance of reality to statements we make. Many people make different statements using statistics. For example, statistics proves that cigarette smoking is bad. According to statistics, males who remain unmarried die 10 years younger. We don't have to verify that. Statistically speaking, tall parents have tall children. Again, you have to verify that. A statistical survey has revealed that a tablet of aspirin every alternate day reduces the risk of second heart attack. I don't know how many you agree. Statistics confirm that an intake of 500 milligrams of vitamin C every day prolongs life by 6 years. Again is a question. And this is very interesting. Statistics are unpacked husband have a greater chance of getting a heart attack. That also you have to verify. So these are some of the what you call semblances by statistics. No, statistics can do anything, but you should do in a right way. Fine. I have been asking this question since 1 or 2 decades. I have been teaching this. What do you know about statistics? No one is smiling here. Everyone is serious. Class. What do you know? No one can tell me what do you know about statistics? Just one word. Very good. He's very theoretical and theoretician man. So, but heart of that, heart who all are sitting here, I know that your heart says this. So you agree. So everyone is laughing. Fine. So then coming to my business, if you torture data long enough, it will confess to anything. That's what I am going to do for another 45 minutes with you, but you have to bear with me. Again a caution, he who accepts statistics indiscriminately will often be duped unnecessarily. But he who distrusts statistics indiscriminately will often be ignorant unnecessarily. Therefore, you have to choose which one do you want. Many people think that average is a simple thing, add it and divide, that will become average. Even interpretation of average is also very important. Many people misinterpret that. And also they take it on the face value of average. For example, if you see, if you take these two people's weight and average it, it is the same weight as you and me. The average weight, they are okay in this world. So therefore, there should be similar things, not the dissimilar things. Average should be similar things, not dissimilar things. Fractions also many people, may not understand sometimes they misinterpret. So in England, also it has happened like that. In England, a Royal Commission reviewing the statistics reported where it is said that the middle class families have on the average of 2.2 children. The minister was very upset with that. How can 2.2 children take birth? One can take, two can take, how 2.2? And scolding everyone, whoever is coming in his way. So no, everyone is very scared about him to meet him also. Then his secretary is very intelligent. Then his secretary told, sir, I have got the report from the commission. I will explain what it is. He explained like this. Finally, they come to a conclusion. The figure of 2.2 children for adult female is in some respect absurd. It is suggested that the middle classes be paid money to increase the average to a rounded and more convenient number. So, but he is happy with that because he wants the only rounded figure. He doesn't understand what is decimal is. So, by virtue of calculation in statistics, he will get the decimal. So, we have to accept that. It doesn't mean that hands have gone, legs, no. It is the number, not the reality. Then, sometimes it is also scaring to interpret some of the things. For example, let us say the risk of having a heart attack in our 50s is 50% higher if you have a high cholesterol. It is very scary. High cholesterol, 50% higher. That sounds pretty bad. Let's say the extra risk of having a heart attack, if you have higher cholesterol of only 2%. That sounds okay to me, but they are the same. If you see the, out of 100 men in their 50s with normal cholesterol, 4 will be expected to have the heart attack. Whereas out of 100 men with high cholesterol, 6 will be expected to have the heart attack. How much more? 2. If you see the rounded numbers or natural frequencies, it's 2. But that can be interpreted as 50% also. So 2 more, 4 and 2 is 50%. So you should be careful in interpretations. Both are same, both are correct only. I have interpreted as based on the percentage. But it has to be realistic, okay? So why we are here? Since morning we are talking about, we want to reduce the error or uncertainty. So statistics is nothing but removing the uncertainty. Nothing else it does. So I just give you, this is not very important, but I wanted to inform you. There is an induction, there is an abduction, there is a deduction. There are the 3 types of uncertainties we will have in our research. But which one is more dangerous and which one is easy and which one you have to take care? Will come deduction. Deduction means, based on some concept you say it is correct. 2 is 5. So, he is asking, Hadi, if you said 2 plus 2, 5, could you prove, say, anything in proportion to like that? He says that, then prove that McTaggart is the Pope. If 2 plus 2 is 5, then 5 is 4. Subtracting 3 from each side is 5 minus 3, 4 minus 3, 2 is 1. McTaggart and Pope are 2, but 2 is 1, therefore McTaggart is the Pope. So this is the deductive kind of thinking, which is abduction. It is more dangerous also sometimes. Abduction means with some pre-assumption, you act. Oh, this fellow doesn't talk well, is boring, headache. That's what your presumption you might have come. So that is the presumption. Dangerous. You can see this. The prince, traveling through his domains, notices a man in cheering crowd who bore striking resemblance to himself. He beckoned him over and asked, "Was your mother in my palace?" No, sir, the man replied, "But my father was." So his assumption was only maids will work in the palaces. So he got the shock. Therefore, abduction is very dangerous. We should not use in our research. Fine. This is the one which we are interested in, induction. Making decisions under uncertainty in an iniquitous situation. For example, did the accused in a given case commit the murder? You will have hypothesis, you will prove, you have forensic report, so many things. The induction. Prediction, it has been continuously reigning in the state, so whatever it is. Testing an hypothesis. Is trial knowledge better than the ibuprofen? Something like that. This is called the induction, which we generally use in our, what you call, research. So therefore, the other two things may be used in some instances, but not in the research. Then I'll start with how the research process starts. Therefore, before you go ahead, you should be careful and you should be clever enough to do statistics. How many say this is right? How many say this is right? At least one. Anyone? Soft hand there. No? Right or wrong? So if you know only algebra, it is right. If you know algebra and trigonometry, it is wrong. Therefore, you should be clever enough to use the statistics. Fine. So what is the research process? When you have to start and when you have to end, so that your publication is on your hand. So this is the 7 steps you have to follow. It's like a Hindu marriage, no? Bride and bridegroom walk 7 steps, they are finished. So what are the 7 steps? Define the problem, specify the objectives, select the design. I am coming to that. Each one I will discuss very briefly, because morning also some information you have got. Calculate the appropriate sample size. This is where the villain is. Many people fail here. Analyze data and finally determine the conclusion. These are the 7 steps. Finally, your research is done. So, problem how to define this is a kind of a very standard way of looking at it. So, how the hypothesis look like? I give some kind of hypothesis, you agree or you can complement with this. Well-formulated hypothesis will both be quantifiable and testable. So, how are the hypothesis look like? I will write. Males over 40 suffering from chronic hypertension, a 100 milligrams daily dose of this new drug lowers diastolic blood pressure on an average 10 mm. So, what is the population here? If you don't have the right population, you will not get the right sample. What is the population here? Men who are over 40 years, blessed with hypertension, that is our population. For them, if you give this tablet, the tension will come down by 10 mm. Average. Suppose they have 140, it will come down to 130. So this is the way you should. So your hypothesis should speak your problem. 50% is done. Then design. Since morning you are talking about designs. Cross-sectional, case control, cohort, this and that. Why should you require a design at all? What is this design? So designer shirts, designer pants also you have seen, designer kurtas. So what is the design actually? Design is situational. For example, in a January month of 15th or so, if you give the clothes, same clothes to Srinagar people and Chennai people, one of them will go. Is it not? Therefore, you have to appropriately give by the temperature. So if we just give Kaddar clothes to Srinagar people and Chennai people same, they cannot tolerate the winter. Similarly, the same clothes which they are wearing on January 15th Srinagar, if you give Chennai, they also. So therefore, appropriate. So that's what design is also like that. Suppose you want a prevalence, one design. Incidence, one design. Cause and effect relation, one design. So therefore, different designs for different problems. Fine. So I need not go much about it, because since morning this has been told by many people. So these are all some of the studies which you already knew about it. But I put schematic representation, whatever the studies we have so far understood or going to understand. Observational studies, I will divide them into. Observation studies means there is no intervention. That is the observation study. I withdrawal and cross-sectional. In the case control, persons with and without the response variable, for example, lung cancer are chosen in factors and smoking that are of interest are studied, so on and so forth. Cross-sectional study. There is a minimum control, there is a partial control. If it is a prospective or longitudinal study, factors of interest are chosen in the presence and in the response variable, and cohort study samples from the same specified defined groups are at different times. And experimental study of the intervention group, there is a minimum control, there is a partial control, there is a complete control. Complete control we call as a RCTs, randomized control trials. This is the one schematically you have to tell these are the different designs. Fine. As morning also in Dr. Geta also that systematic review and meta-analysis is the top, followed by RCT and in vitro test tubes and all. Doesn't mean that they are not required, they are required. So systematic review and meta-analysis is important in the sense that if you are doing RCT, working on RCT and interpreting RCT is only one study. So whereas systematic review is the culmination of 20 or 30 studies of similar objective what you call it, conditions. Therefore, the average always nullifies the error. Therefore, the systematic review and meta-analysis gives you a better what you call output or better what you call estimate, so that the policy. Suppose you see the first RCT 1954 cell polio vaccine trial is almost they have 2 lakhs school children were randomized and their outcome has been measured. The groups should be equivalent except for the factor vaccine being investigated. Extra is vaccine, and so there was many polio cases in the placebo compared to the vaccine group. Could the result be due to the chance or it is really significant? We have to use different t-tests and find. Coming to how do you go about RCT at all? What triggers you to go, or what is the question that you should have to use the RCT? If woman, a friend of a doctor, the woman asked her friend who is also a doctor, current pregnancy. History of neonatal death in her first pregnancy due to prematurity and respiratory distress in the newborn child. Likelihood of premature delivery again. Doctors advise to try a short course of corticosteroids to prevent neonatal death. Will steroids help? That is the lingering problem in the doctor's mind. Whether steroid, if I give steroids, will it work? If or unnecessarily I am giving. So then she decided to go for a kind of a, she doesn't know what that is, that RCT, but she logically put that in a form. The form is like this. The question the doctor has asked herself: mothers at high risk of premature delivery will be her population. Short course of corticosteroids is the intervention. Compared to no steroids is the comparative group or placebo group, whatever you call. Final outcome is prevention of neonatal deaths is our outcome. So therefore, if you take the first letter of everything, that is PICO. That's what PICO is. So when I was delivering in Chennai, Sari is also there. So suddenly, the women scientists are looking at me because PICO is being used for saris. Yeah, saris. So not that PICO. This is the who is the patient or what problem is being addressed. What is the intervention or exposure is I. What is the comparative group C, and what is the outcome or endpoint I. PICO. Fine. And also, the RCT can be now after the pharma industry or the entire medical fraternity, say 10 to 15 years onwards, the RCTs has been subdivided into 4 subgroups. One is test of equality, is just what you have the experimental group, you have the control group, whether they are same or not, you prove. That is what generally we do that. Second was test of non-inferiority. What is this non-inferiority? Why we call it as a non-inferior? It's not inferior, non-inferiority. White people have a very knack way of giving a catchy words. So for example, someone says that, a girl says that I am beautiful. Then another one says that, so what, I am not less than you. But she is not equal, she is not less than you. So therefore, the drug, which is a very superior drug, But other company says that it is not as inferior, is not inferior to that. That's what he says. What does it mean? The inferiority margin which is set by the doctor, saying that if at all the drug is even less than the amount he or she has mentioned, is then that is not going to clinically affect the subject. Therefore, that margin is okay. So most of our Indian clinical trials have a standard drug. Why should I use this non-inferiority drug? Pardon me? Give him a big clap. So economic reasons, most of the time economic reasons. If the drug is 20 rupees, another drug is 500 rupees, but the 20 rupees drug if I take, that is not going to harm me clinically. Because of the conditions of the country, we can give it. Therefore, non-inferiority trials. Superiority trials. No matter what you are, I am beautiful. No one can beat me. So that is generally we don't do. Most of the superiority trials come from Europe or America. Maybe 1 or 2 trials are there in India, but I am not sure. Because that molecule is superior. No one can beat that. Superiority margin. Test of equivalence means demonstrate the difference between the standard treatment is no clinically importance. Means that margin, if at all is there, no problem, go ahead. So I will just pass it. So for these, sample size calculation is different. Difference means the formula is same, but the playing with the mathematics is different, denominator and numerator. Otherwise the formula won't change, concept won't change. Therefore you have different sample size formulas. That is again a different class we can have. Therefore finally, for all the trials, if you want to do a diagnosis kind of a thing, I go for a cross-sectional study. If I want a therapeutic kind of a thing, I go for a RCT. I want a prognosis kind, I go for a cohort. If at all I want to prove cause and effect relation, I go for a cohort or case control. I want to have a descriptive studies like case series or cross-sectional. So, however, more than one study design can be used to answer any given question of a causal association. Fine. So, this is the one. So, these are all some of the guidelines which you all, suppose you want to conduct a observation study, you go for a STROBE guidelines. You want to conduct a clinical trial, RCT, go for a consult statement. You want to conduct a systematic review meta-analysis, go for a PRISMA. And there are some other standards for reporting studies of diagnostic accuracy, STARD. So, there are many guidelines are available, but mostly use 3 guidelines, in fact, STROBE and PRISMA, which you generally conduct those things. So, this I am leaving the slides, you can take. Study population, 4th step, we are going to tie the knot after the 7th step. So, study population is very important. If you don't have the right population, you will not have the right sample. Suppose I want pregnant women, you got all the lactating women, everything goes wrong. So, therefore, you should identify the correct population from the correct sample, then your study will be right. So, I give a dramatic example. I am just dramatizing this. You just see the question. So, United Nations sent to all his 182 member countries asking to solve this question for the solutions of the food shortage faced by third world countries. There is a question UN was sent to 182 countries of the UN chapter countries. Then first it has come to Southeast Asian countries. They read this statement, they stopped at honesty, and they said that in our countries we don't have honesty, we cannot run this study. And it went to China. China also read this statement, they stopped at opinion, and they said that in our country our people do not have any opinion to tell, therefore we also cannot run this study. Then it went to West, and they read the statement, they stopped at solutions, and they said that in our country we do not have any scientific solutions, we only eat whatever we get through petrol, therefore we also cannot run this study, they sent it back. Then it went to Europe. Europe also read this statement, they stopped at shortage. In our countries we do not have any food shortage, so we are also cannot run this study, they sent it back. Finally it went to Big Brother, Trump or America. So America also read this statement, they stopped at third world and said that we don't know what is third world is, entire world is ourselves, therefore we also cannot run this study. Finally the population they did not get. That is the problem if you don't have the right population. Finally, this is the villain. sample size calculation. N number of times I do, N plus 1th time you get the wrong. So this is, you have to practice, you have to work in the real field, then only you will know. Why should I have a sample size at all? Why should I need a sample size? Without that, can't I go ahead with this? Suppose I ask you a question that, can you estimate the anemia prevalence of Delhi under 5 children? Do you know how many under 5 children are there in Delhi? Anyone, any wild guess? How many would be there in Delhi? कितना है? 4 million थोड़ा ज़्यादा बोला आपने, adults को भी ऐड किया, okay ठीक है. So that is 12% you say, 24%, yeah maybe, he's right, sorry. So 12%, 2 crores, 2.4 million approximately children would be there. Can you take blood of 2 million children and estimate? Impossible. Okay. Therefore, if at all I have a method, I take few thousands of children. Instead, I can extract all the characteristics of the 2.4 million children. Am I not successful? I am. Therefore, economic reasons, first reason. Ethical reasons. So I think most of you also know about the animal studies, how it is being performed. Many of you must be knowing. They just do study on 5-6 animals. After the study, they sacrifice. And finally, they found that the study is not very powered study. They just don't publish the results and they just abandon the study. They killed all the animals. Ethical reasons. Third, scientific reasons. There are sound scientific principles, how to calculate sample size, whether it is right or not, this has been proved. So use that. Because of these 3 reasons, we calculate the sample size. Fine. So if you take too big, it requires too many resources. If you take too small, it won't serve the job. Therefore, it should be appropriate and adequate. People also have the misconception that big is beautiful, in the sense, no, you take large sample, yaar, kuch nahi hota. So I just give a funny example. Big is not beautiful. One day there was a fire in waste basket in the office of the Dean of Sciences. In rush a physicist, a chemist and a student. The physicist immediately starts to work on how much energy would be required, all the physics formulas he is using and to douse the fire. The chemist also working on real intelligence he is using. In the meantime, the statistician rushed in and he started lighting remaining baskets of the Dean's office. Then they got annoyed and yelled on him, what you are doing? We are dousing and you are lighting the remaining basket. Then he said that, statistician replies, well, to solve the problem, you obviously need a large sample. Large sample size is mathematically not correct. It will inflate the type 1 error. That is a different theory. So you should have an appropriate sample size. Fine. So this is what we do now. We start with, no, they both are same in the beginning, your data, and you give the intervention, and you found that the means have increased or decreased, whatever it is. Therefore, the change has happened. Why the change has happened? Because of the intervention. Otherwise, if at all this also like this, then there is no change, the intervention has no effect. So this is a testing of hypothesis, no, we call. Then you reject that. First you think that they are not, they are same, but at the end you found that they are not same. You reject that. So then p values come, p is less than 0.05. What is the typical value? If you repeat the experiment 100 times, 95 times you get like this. 5 times you may not get. That's all. As simple as that. But I give a funny example how judiciary and hypothesis testing can be equated. You can say test of significance, a court trial. Null hypothesis, every defendant is innocent until proved guilty. Research design, police investigation. Data test and statistics, evidence and exhibits. Statistical principles, judge's instruction. Statistical decision, a verdict. Type 1 error, conviction of an innocent defendant. That 5% is innocence. They have been convicted because though they are correct, we think them. Because of various reasons, out of sight, your experiment did not yield, our mission is not working, whatever the reason. Type 2 acquittal of a criminal, generally we call as a power, 20%. So the judiciary says that is no single innocent being punished, but 10-20 criminals may get away without punishment. So same is. So diagrammatically if you see, the fun is collecting the evidence means you are giving the intervention. After our intervention, third graph, that has been tilted because one has increased one than the other. Fourth one, you are giving a decision, rejecting the null hypothesis. That's all the testing of hypothesis. Therefore, these are all some of the things. So also 4 types of health analytics generally we do. Descriptive. What is that? Analytical looks data to examine, understand, so on and so forth. Diagnostic analysis goes deeper than descriptive analysis, seeking to understand why and what happened. Predictive analytics relies on historical data and past trends and assumptions to answer questions about what will happen in the future. So identify, then this is the prescriptive analysis. Identify specific actions, an individual or organization, whatever it is. So I am not going to tell you about all the tests, but this battery of tests you have to remember. There are parametric tests, there are non-parametric tests for your data. What do you mean a parametric test? Parametric means where you can quantify mean, SD, correlation, regression. Those are called the parametric test. Non-test means yes or no type, descriptive type, and also you have gave the rankings 1, 2, 3, 4. That is a non-parametric. And also sometimes when the sample size is small, instead of use the non-parametric test. So whenever the assumptions are following, parametric is the best. If not, then I go for a non-parametric. So these are some of the battery of tests: T test, F test, ANOVA, and non-parametric Wilcoxon, and also you heard about Kurzweil-Wallis. These are all some of the known tests which many of you must be using. So therefore, kinds of research approaches, how do you go about it? For example, simple descriptions. You go for, I told you descriptions. Comparison of group means. So comparison of values in the literature. Suppose you have one group, you want to compare with other group, then that is called a one sample t-test. Comparison of 2 groups, you have experiment, you have control, you are comparing that. Comparison of more than 2 groups, that is we call generally ANOVA, analysis of variance, when the groups are more than 2. So when you want to association 2 categorized variables, is there an association between cancer? Or prediction, the values of one variable from another. Can you predict BP from the weight? If the weight increases, BP increases. Suppose you have so much weight, so there are prediction. So determining the effect of interventions, you do the experiments. So these are the kinds of research you have to know. Names of the variables, some of the literature says one value, one name, the other literature gives another name. For example, the variable measures subject response, maybe it is a response variable or dependent variable, both are same. The variable which will explain the response variable, it may be explanatory variable or independent variable. Sometimes researchers intervene and conduct experiments, it may be outcome or response. The type of intervention may be treatment, factor, both are same. Factor is generally categorical and treatment is quantitative. So these are some of the names which you should understand for reading the literature. So this I am skipping because I don't want to show this. Yeah, this you just spend a minute at time. Types of outcome variables determines choice of multivariable analysis. For example, your data is interval data. Interval data means continuous data. Blood pressure, weight, temperature, income, expenditure, creatinine, variables. For that, either I go correlation, I go linear regression, I go ANOVA, or multiple linear regression. These are some of the techniques that can be applied for the interval data. Suppose my data is a dichotomous data, death, happened or not happened, cancer intensive care unit. For that I go for a chi-square test, Fisher's egg, whatever it is, or multiple logistic regression. Generally, you know about logistic regression, you have the dependent variable is yes or no, died or alive, under normal, like that kind of scenario is there, I go for a logistic regression. Ordinal data, stages of cancer, disease, I go for chi-square or proportional odds regression, I go. Nominal data, I go for chi-square data, ANOVA or multinomial logistic regression. Counts data. So number of infections, number of hospital admissions, I go for a Poisson regression or negative binomial. So these are some of the techniques and the type of the data and what you are fine. Now another 3-4 minutes I spend on data quality challenges, which is very important. Therefore, always the dimension of data, I think the previous talk also you came to know, it should have again validity, reliability, completeness. Suppose you have estimated 500 sample for our study, you got only 200. That precision, timeliness and integrity. They are some of the dimensions of data quality we should follow. So keeping this in view, we at NIMS, when I was director, we have come out with the data quality guidelines. I will come to that. Generally, any experiment you conduct, there are only two types of errors you can get. One is non-sampling error, another is sampling error. Other than that, you don't get anything. In the non-sampling, either it is a systematic error or variable error. In the sampling error, mostly you do mistake of the wrong design or sample size. These are only the mistakes. Other than that, you won't do anything. So I'll give one by one. Systematic error or non-sampling generally. Systematic error means you do systematically the same thing, same mistake. I just give a funny example, a doctor and patient conversation. How systematic error can happen, you can see. Doctor says to the patient, you have very serious disease. Of 10 this disease, only one survives. But do not worry. It is lucky you came to me for I have recently had 9 patients with this disease and they all died of it. Will it happen like that? No. First 9 patients will die, 10 will survive. No. People die randomly, not systematically. Do you agree, sir? Yes. So this is the system. These are some of the, I'm just given for funny sake. Systematic means doing the same thing repeatedly. Instrument error, I think many of you fetches data, NFHS surveys. They have used the MQ method for the hemoglobin estimation. So it was proved that in the they are underestimating the hemoglobin level by 0.5 to 0.75 grams. So in turn, that is giving the anemia prevalence, more anemia prevalence because of the instrument error. So therefore, instrument also plays a very important role. Sample size, how sample size plays a role. So I also gone through one of the literature. I found this in a funny example in SAS manual. You just see this, how the sample size is misleading. The cure rate of a drug for a particular disease is 33%, means 1/3. 1/3 of the subjects got cured. That's what the statement says. If you go deep inside, what they got, 1/3 of the mice used in the experiment were cured by the test drug, perfect. They got used. So the statement holds good. Second statement, 1/3 of the test population were unaffected by the drug and remained in the moribund condition. Third statement, the third mouse got away. What is the sample size here? 3. One is in moribund. If based on the one subject, if you say this cure rate is 1/3, then you are misleading. It was misled. Okay. Then I just picked this because it is in favor of Trump only. So if you see this graph, the Democrats are 62%, the Republicans are 54%. So the difference is how much? 8%. But the graph, if you see, there are 4 or 5 fold more than the Republicans. So they might have done that. Biden's aapne kiya ho gaya. So this is, but generally, no, people don't see the numbers. They see the bars. If you see the bar, oh my God, the Democrats won by so much of margin. So where is the, this is also data error. So how the data error has happened? Then what they did and what have we got? They just changed the scale. You can see this. They have started with 53. The number is 62. Instead of that, if they start with 10 or 20 or 0, this is the one. Not much difference, 8%. So this is also misleading. Then what, suppose you already got the data, okay, what you do with that? How do you clean the data? Using again these 7 steps. Always I don't know why 7 steps are synonymously coming for our Hindu marriage. So again, data related also 7 steps. What are they? How good is the data? What should you do? You should do some preparatory analysis, data quality checks and all. What happened to the data? Descriptive. Why and when it has happened? Diagnostic analysis. How did it happen? Cause related analysis. What will happen? Predictive analytics. So these are all the things which you do and minimize the errors for final analysis, so that your data is not 100% correct but close to 100%. And finally, how confident can be reliability based on data. So these are the analytical tools, and you have to do the purpose. Purpose is the question, and if at all you do this, the data ready and your data is clean data. Therefore, the third scenario is you have to data profiling, and the previous speaker also told, checking the data quality, data integration, and big data analytics. Now it is the craze. So previously, 20 years back, people who are wearing white, doctors apron and steth, they used to go on. In the bus stops also, people used to wear steps so that Dr. Sabar. Now it's gone. So what are you doing? I am doing AIR, and I am doing big data. That is a craze now. Whether they know, they don't know, they say, I am doing AI. So that is a craze now. If in our research paper, if you don't put big data and AI, even the reviewer also thinks that you don't know anything. So that is the scenario. So people are using. Therefore, we came out with the data quality guidelines, which is available in the website, in the Google as well as ICMR website. Very, just like a novel you can read. It has got 6 chapters, and each chapter gives how you should take care of the data quality guidelines in different scenarios. Fine. This is the book. Coming to again, I also should talk about something about big data, otherwise you think that this fellow doesn't know anything. So every day you are 2.5 quintillions of data. Can you write those many zeros on your paper? That much data, a day's data, 1 day's data is being created. That is the magnitude of the data available in the worldwide open source. How? In every minute, 98,000 tweets. Already 10-20 tweets from this group might have gone. Last class, they are boring. So tick, 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 tick. These things, no, tweet, you tweet it. Yeah, I'll come within half an hour, everything will be done. Like that, no, the thing means, because the instrument is available, you just tweet it. 98,000 tweets or 1 lakh tweets per minute. That is the magnitude. 7 lakh status reports. Yeah, status reports, I think many people say, I am single here, so what? Why should we say to the world, I am single? I have seen many. I am single. As if the great thing he has done or she has done. So, 7 lakh searches in Google, 168 million emails, total 820 terabytes of data per minute. Can you calculate for a day? You cannot, that much big it is. So that is the magnitude of data, big data. That's why the concept of big data has come. Big data not only in terms of magnitude of big data. Suppose your Apple, phone, it has lot of, it can hold gigabytes of data. The data is very huge. But big data advantage is that we are all used to take the structured data. Means you have got the weight, height, arm circumference, head circumference, income, expenditure. These are standard defined variables. Against that you have the data. Simply take, analyze AP info or red cap, whatever it is. But you have the data, Dr. Sahab scribble here. He has that disease, this is this. Something like that you wrote in terms of text, in terms of some figures, in terms of pictures. How do you analyze that data? Advantage of the big data. Not the big is not beautiful. You can have, your PC can have hold so much of data. So, volume is one of the factor anyhow, because you have very huge memory laptops or desktops. Velocity. In the laptop or desktop, the data, entire data, they will throw like a throw ball. But in the big data concept, it makes into pockets first, then one packet of data goes and again reassembles. That's the concept of LAN kind of a thing. And variety. Variety means we know only structured data, unstructured data, semi-structured data, pictures, films, whatever it is, that we'll analyze. Veracity. While analyzing that, it makes sure that that is correct. And value. Once you do that, that has got lot of value. Therefore, we are struck with structured so far. You can have quasi, you can have unstructured, but unstructured data, you should be little careful and validate the data, then only you can use. Because even one digit, everything will changes. So this is the advantage of big data. Not that it holds lot of information, no. You can have a PC which can hold huge information, but the advantage of this is converting unstructured data into structured data. So this is the one. Basically, we have 3 groups. One is normal, moderate, can you discriminate those children, okay, using some 10-15 variables, their age, their parental education, their nutrition status, based on these 10-15 variables, can you discriminate normal as normal, undernourished as undernourished? If at all that technique is there, can you do that? It's called linear discriminant function statistics. But these AI people are very clever. They took away the technique and they named it as a supervised learning. Very fantastic, no? Supervised learning, which you are listening or hearing, is nothing but a discriminant function analysis or logistic regression. Those two techniques they named as a supervised learning. So what it does, these are three different discriminated three groups, normal, undernourished, mild. But some contamination is there, normal has gone to mild, mild has gone to severe, something like that. So this contamination percentage again it calculates. If it is less than 10% or so, then say that yes, you accept this technique. So this is supervised learning. What is unsupervised learning? Means no one, you are not involved. You just feed the data and ask the AI to come out. So generally we feed the data and it has come out with three different patterns. Pattern 1, pattern 2, pattern 3. Then how it has given? It is given based on the data. How it has given? You heard about Mahalanobis? You know a Mahalanobis? Mahar thought that is d square, Mahalanobis distance will be used in AI. Even in dreams also you might not have thought at that time, 50, 60 years back. So what Mahalanobis distance does is, suppose you have all the subjects here have your weight, your height, your age, your education, everything is being fed in the computer, everyone's. Then the distance what it does is, it takes parameters, compares is 10 parameters. If they are same, what happens to the distance? 0, because your weight minus is height, your height minus is height, like that, no? So both are same in all aspects, the distance is 0. That's why they are called similar. When compared to others, the distance is very away, then they are dissimilar. So the similar things will be in this, dissimilar things will be this. So in this group they are same, when compared to other groups they are different. So that's why it's called a patterns. That they gave a very fancy name, unsupervised learning. That's what it does, computer does. Nothing but they use the cluster analysis. Okay. Then guidelines for reporting of statistics for clinical research. These are some of the guidelines, no? Reporting of design and statistical analysis, consort, inference and p-values. Avoid say p-values 5% is trend. No, you should not say trends and all. Reporting p-values 0.7344 suggests that there is an appreciable difference of p-values between 0.7344 and 0.73446. Multivariable models and diagnostics tests, conclusions and interpretations. These are some of the guidelines for clinical research. Therefore, be clear about the purpose of the experiment. Keep the experimental design simple and therefore clear and powerful. Draw up statistical analysis plan as part of the experimental design. Explore the data visibility before launching into statistical testing. Be very aware of the multiple analysis. Interpret both significance and non-significance with care. This is what morning onwards people are telling this. So, before I conclude, ultimately we also should have the idea for doing anything, even AI, big data, ideas. So idea will change everything. So I just give a funny example. Book is the idea. Book. 3 people became billionaires. So the idea is book. 3 people became billionaires. How and why? I just give them. By writing books, one has become billionaire. Tell me who? Who is that? Huh? Who is that? Harry Potter? Give them big clap, please. Yeah. Okay. By reading books, one has become the billionaire. By reading books, vociferous reader, and he told his in biography. I am telling the gender also. Give him 2 claps. Yes, you are. Finally, by selling books, someone has become a billionaire. So, concept is book. 3 people became billionaires. So, that is where we actually drive in. So, I hope and see after 10 years, one of you as billionaire. Thank you very much. So these are some of the packages which already told. And never give up statistics. Yeah. If you give up, it will harass you. So you don't give up statistics. Statistics, if you fear no, it will fear you more. So you just start doing it, even if it is wrong also. Thank you very much for your patient hearing. Questions?
There cannot be anybody who can make statistics so simple.
Thank you, sir. Thank you, sir.
Any questions?
Any questions, please?
I have a question, sir.
Yes, sir.
Actually, the pad and pad we are very conventionally taught, and we know how to make it, and we have done it on the manual sheets, worked it out and all. Now when we get into it, we get some fancy terms of tails. Can you give a little?
Tails.
Tails.
Yeah.
One tail, two tails.
Yeah. Whatever we test is, we say there is no difference, and we prove there is a difference. So generally what you do, mean one is not equal to mean 2. So then they say that mean 1 is equal to mean 2, alternative. That is to make sure that there is careful about that. We never know whether increases or decreases. Even it is less also, the difference comes, then your experiment might be wrong, but that is, so two tail means, two means are same, two means are different. Fine. Everything is two means only. Suppose you pretty well knew that one mean is definitely you get bigger than more than the other mean. Mean 1 is greater than mean 2. Under those circumstances, that will become one tail only. So the advantage of one tail is that it will, so for becoming significant, the value is less when compared to two tails. That's all. For one tail, 95% confidence is 1.645. For two tails, it is 1.96. So therefore, to become significant for two tails, your outcome or intervention should be more. Delta means the outcome is more compared to one tail. One tail means you already predefined. You know that it is more than that. That's all. It is just a kind of art than a science. But the value is 1.645 for one tail, 1.96 is for two tail. So 1.96 to get, your delta should be more in two tails than one tail. Suppose you take the non-inferiority trial. Non-inferiority trial is always one tail because you know that is non-inferior.
Superiority will also be one tail.
Yes. You know it is one.
Equivalence will be two tails.
Yes. And equal is two tail. Generally we use equal trials. Means we say that mean is not equal to mean 2, and we prove that they are mean equal to mean 2. So mean equal to mean 2 we say, and we prove that mean is not equal to mean 2.
So will it be wrong to do a two-tailed in a superiority or a non-superiority?
No, nothing wrong, but you will lose lot of money, no? When the sample size is more, you will lose money. The very purpose of subdividing the clinical trials is to save the money. Suppose you are doing a cancer trial, on each patient you are spending 25 lakhs example. Suppose 20 patients, if you save also 5 crores. So that is the main and yeah, this is very good question. Actually, it is haunting us also in many of the projects. Basically, there is an outlier analysis. Generally, what they do is minus 3SD plus 3SD, they remove the remaining. That's wrong. Suppose I take hemoglobin value, I can say. His hemoglobin is say more 21 or 22, I don't know. Still, he is very active and surviving. So if you give plus or minus 3SD, that is going almost 16 ko khatam ho raha hai. But inka value, really inka value, the value is 20 plus. So therefore, if you remove that, you are not doing the justice in terms of the natural values. You are just, you can call that as a plastic values or fabricated values. If you take. Never nice sometimes, but it will come, Yeah, it will come. Ma'am, actually, what should you should use your, what you call wisdom also. For example, you also have the ranges, consistency. There are two things which will make you, consistency means, suppose I give a small example, whether you are going to school, first question, yes, no. Second question, how many years of schooling? 9. So he is not going school, but 9 years education you are giving, that's the consistency. Range you already prefix. If the hemoglobin value is less than 7 or more than 18, I will not consider it in the analysis. then you take out all those values. If they are more, then you can see why that has come. Is it a typographical error or data entry error, or the person who took the lab test, it's correct or not? Those also you have to check indirectly. Then only you should make sure that yes, it can be removed. Yeah, you can write, and you said that estimation, suppose hemoglobin estimation correct or not, you can recheck again. Again, that you have the second sample and do that. Second is whether the data entry time only you did a mistake. That also you can check. So these 2 points, if you check, I don't think you can remove. I don't think there is any problem. You can remove that being an outlier.
One more question.
Sir.
Sometimes, what I understood from some of my friends, what they do is suppose they have 4 values they have. 5 with animal diet. This happens because they are forced with animal ethics issues. So what they do is, they take a mean of 4 and they put it as a 5th value. Is it right?
No, it's not. So that value is nothing but the derived value of the 4.
So can we say it's wrong, or it's okay, we accept it?
You see, the 5th value is nothing but the derived value of the 4th one. So that will not accept number, that will not add anything. It is a number. To make the more number, they are doing it. So instead of that, I can do a bootstrapping and I can create.
So bootstrapping we can
is
do.
the best.
Oh, bootstrapping is much better as compared to this. That's a nice idea, sir. Sir, another one is this Euclidean distance. Is this same that of this mega? Is it same?
Very good question for you. It is not exactly same, but some extent same. Euclidean distance doesn't consider the scales. Suppose you have weight in centimeters, height in kgs, hemoglobin in milligram, whatever it is. Then it takes just a value. Mahalanobis distance considers the scale and makes to Z-score, that is the scale invariant. So whenever you have a different scales, use the Mahalanobis. Everything is uniform, then use the Euclidean. That's all you have to. Otherwise, what happened? Scale is different here. So Mahalanobis distance denominator, it takes the standard deviations. Therefore, it will become a Z-score. Z-score is a scale invariant. If at all you think that all the variables are uniform scale, Euclidean is the best.
Sure. These things, information we don't get it at all anywhere.
Thank you.
She asked this question is about the outlier.
Yeah, she asked. I think
Can we take that 3 SD as a value?
95% of the values lie between. So some of the WHO gave me 6 SD also, plus or minus 6 SD. So extreme outliers can be removed. But anyhow, you have to checking is your definitely duty, whether that is real or not.
All of us are doing that online checking to do.
No, that is anyone can do.
We don't know how is it working. I mean, sometimes.
Yeah, sir. Lot of this actually blessing this online technologies. Sometimes they are useful. Sometimes they are very erroneous. Because ultimately your intervention is required because you are the domain expert. So most of the online technologies and online people who are doing are not domain experts. So they do based on some program karke de dete. Yeah, exactly they do. But they don't know, many people don't know anything, but they know only Python. उनको Python ही मालूम है, कुछ नहीं. Coding पता है, कुछ नहीं मालूम है. The domain knowledge is very important. Supplement, complement with that. Thank you.
Anybody else to explain statistics like this? I'm really feeling that you all should have hired me as my teacher when I was
so nice.
So nice.
Thank you so much. Thanks for giving this opportunity again.
Thanks for coming, sir, all the way down here.
Thank you, Dr. Rao, for your insightful session that beautifully underlined the vital role of statistics and integrity in responsible research. I would like to call Dr. Vail Pandian to present the memento to Dr. Rao.
I'll circulate this one to everybody. Please. I know that all of you.
And so from where you get the matter for your
Yeah, that's
As we end today's sessions, let's carry forward the ideas, the inspiration, innovate that we have gathered here. As we close day one, we invite all participants to visit the ICMR Patent Mitra Pavilion. We warmly invite everyone to join us this evening for a networking dinner at the AIIMS swimming pool area, a wonderful opportunity to connect and unwind after an enriching day. After the dinner, the bus to the guest house will leave at 9:15 p.m. On behalf of ICMR and AIIMS, we thank all our esteemed participants, speakers and organizers for making today's sessions insightful and inspiring. This is your host Punya Sharma signing off for the day. Wishing you all a wonderful evening and looking forward to see you all tomorrow. Namaskar and thank you.